A very good morning, distinguished guests, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. May peace be upon us. Distinguished participants, ladies and gentlemen, very warm welcome to each and every one of you. We are gathered here at the Trans Resort Bali for the inauguration and Symposium of Indonesia-China Joint Research and Development Center on Vaccine and Genomics. We are honored to welcome you all on this very special occasion at this hybrid session through Zoom meeting and also YouTube. Ladies and gentlemen, before we start our agenda this morning, kindly please have a seat and be reminded to put your mobile phone into silent mode. And for online participants, please be considerate to change your username with name_institution and keep audio mute when you are not speaking. And if you are comfortable doing so, kindly turn on your video for online participants. Thank you so much. And once again, I would like to welcome all the participants who are joining us online or on site here at the Trans Hotel Resort Bali. Very warm welcome once again to each and every one of you. And you may please have a seat as we would like to commence our event this morning shortly. Thank you, everyone.
Vaksin Collaborating Center atau VOLARE. VOLARE bertujuan untuk menjamin keberlanjutan penguatan ekosistem vaksin yang diharapkan juga akan menjadi platform yang lebih koordinatif, kolaboratif, dan sistematis dalam skema triple helix dalam menjalin sinergi antar stakeholder di bidang vaksin di Indonesia. VOLARE bergerak dalam mengkolaborasikan aspek-aspek riset dan pengembangan manufaktur, penyediaan, harga dan pembiayaan, distribusi, logistik dan rantai suplai, penerimaan dan serapan pasar di bidang vaksin. Vaksin Collaborating Center atau VOLARE. VOLARE bertujuan untuk menjamin keberlanjutan penguatan ekosistem vaksin yang diharapkan juga akan menjadi platform yang lebih koordinatif, kolaboratif, dan sistematis dalam skema triple helix dalam menjalin sinergi antar stakeholder di bidang vaksin di Indonesia. VOLARE bergerak dalam mengkolaborasikan aspek-aspek riset dan pengembangan manufaktur, penyediaan, harga dan pembiayaan, distribusi, logistik dan rantai suplai, penerimaan dan serapan pasar di bidang vaksin. Vaksin Collaborating Center atau VOLARE. VOLARE bertujuan untuk menjamin keberlanjutan penguatan ekosistem vaksin yang diharapkan juga akan menjadi platform yang lebih koordinatif, kolaboratif, dan sistematis dalam skema triple helix dalam menjalin sinergi antar stakeholder di bidang vaksin di Indonesia. VOLARE bergerak dalam mengkolaborasikan aspek-aspek riset dan pengembangan manufaktur, penyediaan, harga dan pembiayaan, distribusi, logistik dan rantai suplai, penerimaan dan serapan pasar di bidang vaksin.
distinguished guests, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. And as we are here in Bali, allow me to greet you in Balinese way. Om Swastiastu, which means that my health and prosperity are always among us. A very good morning and warm welcome to our special event this morning, inauguration and Symposium of Indonesia-China Joint Research and Development Center on Vaccine and Genomics. Please give applause to warm up our event this morning. This event is a hybrid session, ladies and gentlemen, through Zoom meeting and YouTube. Today, 28th of August 2024. First and foremost, let us praise and thank God for His mercy and grace we can gather here on this joyous occasion. And allow me to greet dignitaries here with us through the Zoom meeting. Good morning to Honorable Professor Zhang Linqi, PhD, Chair of Indonesia-China Joint Research and Development Center on Vaccine and Genomics. Good morning, Prof. Thank you for your presence. Mr. Roy Himawan, as from ABT-MKM, Director for Pharmaceutical and Medical Devices Resilience. Professor Dr. Masteria Yunofil Saputra, SSI MSc, Head of Vaccine and Drug Research Center, National Research and Innovation Agency. Welcome to Mr. Tantowi Yahya, President of United in Diversity. We also welcome Mrs. Lu Yinghui, Senior Expert of International Cooperation of Tsinghua University. Mrs. Nimas Mega, Marketing and Operational Director of GT Group United in Diversity. Mr. Michael Arantori, Academic Director of Tsinghua Southeast Asia Center. Good morning, sir. Thank you. And also welcome to all delegates from Tsinghua University, distinguished speakers, distinguished senior officials of the Ministry of Health, Indonesian Food and Drug Authority, National Research and Innovation Agency, and other related institutions. Officials of University of Indonesia, Erlangga University, Bandung Institute of Technology, Gajah Mada University, Pajajaran University, and Udayana University. Also welcome to heads of associations and organizations or representatives, health professionals, and academics, also distinguished ladies and gentlemen. We would like to extend our gratitude and appreciation for the presence on this wonderful occasion. Today's event is hosted and organized by Directorate General of Pharmaceutical and Medical Devices, Ministry of Health, Republic of Indonesia, in collaboration with Tsinghua University, Tsinghua Southeast Asia, and United in Diversity. Distinguished guests, ladies and gentlemen, allow me to introduce myself. My name is Putri Cahaya, and I'm so glad to take a part of this event as your Master of Ceremony. Distinguished participants, ladies and gentlemen, before starting today's event, let us pray together for the success of this event. Pray based on individual belief begins. Done. Thank you. Distinguished participants, ladies and gentlemen, Indonesia and China already had long and great cooperation in many sectors, one of which is the health sector. In 2022, Ministry of Health of the Republic of Indonesia and the Ministry of Science and Technology of the People's Republic of China signed a MOU on the establishment of Indonesia-China Joint Research and Development Center on Vaccine and Genomics. The scopes of activities in this MOU, including joint research and development, training and workshop, talent exchange, as well as encouraging cooperation in the field of vaccine and genomics. This great event, ladies and gentlemen, is the implementation of the MOU, which aims to be a forum for discussion of vaccine and genomics research and development. So for that, I would like to invite everyone to please give big round of applause to our event this morning, our very important event to strengthen our cooperation. Distinguished participants, colleagues, ladies and gentlemen, now to begin our event this morning, let us hear the opening remarks for this important occasion from Director General for Pharmaceutical and Medical Devices, Ministry of Health, Republic of Indonesia, that will be delivered virtually. Ladies and gentlemen, please welcome on screen Mrs. El Rizka Andalusia.
Honorable Professor Ling Sisang, PhD, Chair of Indonesia-China Joint Research and Development Center on Vaccine and Genomics. Distinguished expert and speaker, Mrs. Liu Yinghui, Senior Expert of International Cooperation of Tsinghua University. Mrs. Nimas Mega, Marketing and Operational Director of GT Group United in Diversity. Mr. Michael Aaron Tuori, Academic Director of Tsinghua Southeast Asia Center, delegates from Tsinghua University, senior official of the Indonesia Food and Drug Authority, senior official of the National Research and Innovation Agency, senior official of the Ministry of Health, official of University of Indonesia, Erlangga University, Bandung Institute of Technology, Gajah Mada University, Pajajaran University, Udayana University, heads of association and organization of representative or representatives, health professional and academic. Distinguished participant, colleagues, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, salam sejahtera, Om Swastiastu, Namo Buddhaya, salam kebajikan. On behalf of the Ministry of Health of the Republic of Indonesia, I cordially welcome distinguished participants to the Symposium of the Indonesia-China Joint Research and Development Center on Vaccine and Genomics. Today's event is a significant step toward increasing international cooperation, particularly in important fields such as vaccine and genomic for health. We thank our distinguished colleague from Tsinghua University, led by Professor Ling Sang, and present you a warm welcome to Bali, Indonesia, a vibrant hub where diverse minds converge to collaborate. Distinguished participants, ladies and gentlemen, the advent of the genomics age has fundamentally altered the paradigm of diagnostic, vaccine, and drug development. Genomic approach on medicine and transform people's lives by allowing for faster and precise diagnosis and tailored treatment selection and monitoring. The expanding pool of genomic data and novel genome-based technologies will be fundamental in developing vaccine and medicine, especially for emerging diseases. Indonesia also sees the urgency to improve the vaccine research and manufacturing capacity, not only to serve the routine health program, but also to respond to the next pandemic situation. R&D is the foundation for the development of the capacity to accelerate access to innovative technologies and to build experience among clinicians and researchers with the new modalities. A thriving innovation ecosystem also supports indigenous innovation and best practices for scientific inquiry. Furthermore, an innovation ecosystem will serve as the basis for private investment in life science and thus create economic and health resiliency. Based on that, the Ministry of Health is currently implementing the 6 pillars Indonesian Health Transformation Agenda, including health system resilience transformation, through increasing the availability and access of pharmaceutical, including vaccine and medical devices, and health technology transformation, providing an integrated and inclusive health ecosystem. Indonesia is in the process of pushing it to the next level of healthcare through various strategies, including active membership in global organizations such as Global Fund, CEPI, FINE, and participation in multi-center clinical trial such as TB vaccine, malaria, dengue, and also establishment of clinical research center and clinical research unit, as well as the launch of Biomedical Genome Science Initiative, or BGSI. BGSI aims to advance precision medicine by integrating genomic insight into health services. Our goals are to enhance national disease control and surveillance and support the development of high-quality and effective health products. These goals indeed can be strengthened through cooperation. Distinguished participants, ladies and gentlemen, we recognize that progress and sustainable impact can result from collaborative partnership that pool resources, exchange knowledge and expertise, and enable technology transfer. We are grateful to have a close partnership with Tsinghua University, which will further facilitate access to quality, safe, and effective health products and promote genomic advancement through various activities. In recent years, countries worldwide have progressively implemented precision medicine program. China itself has launched the Precision Medicine Initiative, followed by remarkable transformation by establishing the National Genomic Data Center, or NGDC, providing resources to support the integration of genomic insight into various fields, including vaccine research, leading to significant breakthrough. Despite the challenges posed by the pandemic, China COVID-19 vaccine production capacity has made it a major global supplier, effectively providing vaccine to countries around the world, accounting for 13% of the total volume purchased in 2022, with a large portion distribution to other regions, according to the WHO Global Vaccine Market Report. Distinguished participants, ladies and gentlemen, I welcome today's event as a milestone following the signing of the Joint Action Plan of Memorandum of Understanding between the Ministry of Health of the Republic of Indonesia and the Ministry of Science and Technology of the People's Republic of China in 2022. I believe the cooperation between the Ministry of Health and Tsinghua University will last for many years. At this event, I would like to thank Professor Ling Qisang and the Tsinghua University delegation for making your time and joining us today in Bali. I would also like to thank UID and Tsinghua Southeast Asia Center on campus Kura-Kura, Bali for the collaboration provided. And also, I would like to thank all participants, representative of ministries and agency, hospital, and industry for your participation today. I understand that on today's event, there will be an officiation of Joint Research and Development Center on Vaccine and Genomics. I do hope that this officiation will initiate more and more joint vaccine and genomic research, which is crucial to ensuring widespread access of vaccine and medicine for the population. I look forward to the productive and everlasting Joint Research and Development Center on Vaccine and Genomics, so then we can provide high-quality health products and services for our people. Let's collaborate and make a lasting impact. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Please give applause, ladies and gentlemen, to Mrs. Rizka Andalusia. Thank you for the amazing remarks to begin our event this morning. Distinguished guests, ladies and gentlemen, for the next remarks, let us hear warmest greetings from Chair Indonesia-China Joint Research Center on Vaccine and Genomics. Distinguished participants, colleagues, ladies and gentlemen, please join me in welcoming Professor Chang Linqi, PhD. Please welcome to the stage and please be on podium.
Good morning, ladies and gentlemen. Yeah. Good morning, ladies and gentlemen. This is my great honor to be here today together with our friends and colleagues from Ministry of Health, Tsinghua Southeast Asia Center, Asia Center, and then United in Diversity and Chinese Embassy. Thank you so much for organizing this symposium at a very important and critical juncture facing the health challenges for both countries. Since 2020, when the joint MOU signed by the Ministry of Health, Indonesia, and the Ministry of Science and Technology, China, we have determined to work together to build a vaccine and a genomic joint center here to tackle the most challenging problem, health problem facing both China and Indonesia. And today's focus is part of that endeavor to bring together the experts, professors from both countries to foster communication and identify the key problem areas of collaboration. So over the last 2 years, in particular working with the Royal sitting in the audience and United Diversity, Tsinghua Southeast Asian Center, we have frequent communications, either online or in person. We co-developed a joint action plan to focus on the area that's really critical for health, particularly in vaccine and genomics. Like yesterday, we have already a warm-up discussion in Jakarta with MOH, and we also visited BRIN national laboratories. We have identified many areas that we could pursue together. So today's symposium is critical to move that collaboration a step further, to have a real in-person communications. And then with that communication, we could identify the key collaborative area. From there, we could move fruitful communication and promote our Indonesian and Chinese scientists and the industry partners to focus on an area that truly matters to the health of our two nations. And then thank you so much for your wonderful organization and looking forward for discussion. Thank you.
Thank you so much, Prof. Chang, for your warmest remark and also share a short background of this event or the reason why we are gathered here this morning. Distinguished guests, participants, colleagues, ladies and gentlemen, now we move on to the speech from Tsinghua University that will be represented by senior expert of international cooperation, Tsinghua University. Please welcome on stage Dr. Lu Yinghui.
Sorry, I have a little bit cold. Good morning, everyone. Distinguished Mr. Roy, Professor Zhang Linqi, and dear colleagues, experts, friends from Indonesia and China. It is my great pleasure to attend the symposium to witness the further development of the China-Indonesia Joint Research Center on Vaccine and Genomics. Based on the latest US News ranking, Tsinghua University is ranking number 16 among the best global universities. And it has always been committed to promoting the global innovation and the sustainable development through broad, extensive international collaboration. In the past 5 years, Tsinghua has carried out over 1,300 R&D collaboration projects with over 60 best universities worldwide, like University of Cambridge, NUS, USNW, and also as well as with over 240 outstanding, well-known companies across the broad field of automobile, energy, and retail, and so on. In the field of biomedicine and pharmaceutical companies, we have long-term collaboration with pharmaceutical companies like Bayer, BI, and BMS. On top of that, in 2016, Tsinghua and Gates Foundation, as well as Beijing municipal government, together established the research institute called GHDDI, focusing on the global public health. So as a very important measure of the Belt and Road policy, Tsinghua highly value the collaboration with Indonesia. So we particularly set the research, the international collaboration fund to support the formation of the teams from both sides and to promote the academic exchange and the seminars. So during the past 2 years, I guess many of the scientists sitting here and attend the meetings and the exchange and the visit investigation from to both parts. And last year and this, I think this June, we have Indonesia delegation to Tsinghua to have the investigation to learn what we have in Tsinghua. And today, I think we have a lot of experts and professors sitting together. And it is a great opportunity to talk more about what we can have a good collaboration in the future. So, so personally, it's my second time to visit Indonesia. And the last year, we went to the University of Indonesia and some to visit some very promising biomedical companies. And yesterday, we went to the Institute of Brain, and we saw the very cutting-edge facilities. And so I think it's a very good foundation for both parties to have a very fruitful collaboration. So I hope the collaboration will finally yield a very solid, concrete, and productive results that will benefit the well-being of the people in both countries and even good for the global public health. So I wish the meeting will be a great success. Thank you all.
Thank you, Dr. Lu, for the speech. Distinguished participants, ladies and gentlemen, before we dive into our main topic of our discussion this morning, I would like to invite everyone to take a group photo session together. So I would like to request all distinguished participants, colleagues, and all of you are gathered here. Please all rise, please stand up, and photo will be taken with our committee from the stage. So I would like to invite everyone to please all stand up and face the camera that we provide on the stage. Yes. This is also part to boost our mood and energy before we dive into our main topic of discussion this morning. We have to check. All of you are captured in one frame. Can we start? Photographer on stage? Yes? Okay, allow me to do the countdown. 1, 2, 3. Smile. Okay, big smile. Another pose, please. Yes, you can change into the other pose. Okay. Health greetings. Yes. Okay. 3, 2, 1. One more photo. Yes. Again, 1, 2, 3. Is it enough? Okay, thank you so much everyone for joining the group photo session to capture our moment of togetherness. Please have a seat. Distinguished participants, ladies and gentlemen, to continue our agenda today, we will enter the first plenary forum, which will discuss about vaccine ecosystem for research and development from Indonesia's perspective. And to guide this session, ladies and gentlemen, I respectfully invite Deputy Director for Health Biotechnology Policy. Kindly please welcome on stage Mr. Kindi Adam, SSI and Biotech, as the moderator of this session. Please welcome. Yes, Mr. Kindi, stage is yours.
Thank you, MC. Good evening, ladies and gentlemen, distinguished guests and speakers. Good afternoon. Good morning. Yes, good morning for this session. Yeah. In case it's dark, there's no window. I think it's already evening. So hopefully that you are having a wonderful day, wonderful night tonight. And on behalf of organizing committee, I would like to extend a warm welcome to all of you all to Bali. And it's a pleasure to have you all here in this symposium. For the next agenda, I'm honored to become the host for this first topics. Here will be with the speakers. I would like to invite our speakers to join me at the stage. First, Mr. Roy Himawan from Ministry of Health of Indonesia. And the second is Professor Masteria from BRIN. Please give applause. And the third, we have Mr. Andreas Doni from Etana Biotechnologies. And the last speakers will be invited soon because we're still waiting for him. It's Professor Budiman Bella from University of Indonesia. Before we begin, I would like to remind everyone that each panelist will have 10 to 15 minutes for the presentation, and we will have a time for discussions, and you can ask the questions in the discussion session. So for the first, I would like to introduce our first panelist for today, Mr. Roy Himawan, with the topic of policy regulation on research and development and also market shaping of vaccine in Indonesia. Short introduction. Maybe all of you here already know Mr. Roy Himawan, but I still will read the introduction for him. Mr. Roy Himawan, he have a strong educational background with bachelor in professional degree in pharmacy from Gajah Mada University, and also Master of Public Health from University of Indonesia. He have an extensive academic qualification and solid foundation for his career in public health and also pharmaceutical management. Mr. Himawan currently serves as Director of Pharmaceutical and Medical Devices Resilience at the Ministry of Health, Republic of Indonesia, with over a decade of experience in various strategic roles. And now he's been instrumental in shaping national policy and program aimed to enhance access to essential medicine and also healthcare services. And I believe that his expertise and leadership in this field will be a respected figure in the national and internationally for tomorrow. Please welcome Mr. Roy Himawan. Time is yours.
So, thank you, Pak Kindi, for the exciting introduction. And can I do stand presentation? Thank you, Pak. So, good morning, colleague, and especially good morning to Pak Tonto Yahya. Thank you very much, Pak, for making your time present here. Professor Linky, thank you very much. I think it is our second anniversary, Professor, for this kind of meeting because we had conducted twice. So today I would like to share to colleagues and Also, I would like to welcome Professor Erlina. Thank you very much, Prof, for making your time representing the IMERI today. So I would like to update on the colleague on what happened in the space of policy, with regard to research and development, and how do we make that policy work in place, complement it by the market shaping of vaccines. Because in practice, in the reality, everybody will understand, or whenever you do research, when you do development, you will come to an end with the product. And the product itself should be go to the market. So whenever there is no access to market, the R&D will still only be R&D, not will be impact to the population. So in the next stage, this is on the next slide, we will see that the background is clear, that we had a challenging situation now, where we see the timeline between pandemic event is going shorter and shorter. So as we may see on our history record, in the last start from 1920, in the period of 70 years, we only have 6 pandemic situation on that 70 years. But starting from 1990, in the term of 30 years, we already have 10 pandemic. It makes clear that this situation will initiate the global tension to do a managing action to risk out global supply chain disruption. Because the pandemic is going more frequently, so every country will do their best to secure the supply chain. That's why many attention now shift from efficiency to the self-resiliency. So understanding this, we are being forward by the national situation in Indonesia, where the ecosystem to do and to provide the resilience is still lag behind. So the next slide, we will see that on the next slide, we will see that we have the capacity. We have the capacity to do research. We have the capacity from researchers, from funding, from industry, and we need to do something for this component to be more elevated to the next level. So next slide. We also see that in the space of ecosystem, on the stage of developing the product, we already had the potential actors. We already had the agency, all the potential that we have, and now we need to collaborate and to do something more focused, to bring the resiliency and to lift up the R&D of the product Indonesia. Next slide. So this is what we have the data, that from the developing vaccine, we see that the capacity that we have are immensely big. In the clinical stage, in the exploratory stage, on the preclinical stage, that we see most of the trial what happened there. As you see from the slide, the platform of the vaccine are being very various, started from the traditional conventional platform like inactivated, up until the newest technology like mRNA. We also looking this platform to be more contributor to the health system by making them directed to the disease focus. Next slide. And with regard to the capacity itself, we do see an increasing number. We do see increasing capacity to manufacture the vaccine in the last 3 years. So it become challenging for us now how to make this capacity work. How to make this capacity is still on the place whenever the country need that manufacturing capacity. Next slide. So this is what we see from the small picture that we have on 2022. We do small research, we do small collecting data action, talking to our researchers, and we see from that activity, even though we have several capacity on research, but we still have lack and gap on the researching activity. We still have not sufficient researchers in every university, and also we still have low productivity on our activity on research. This is complemented by fully regulated products that make the developing of product being little bit stringent, little bit hard to pass through, but everybody are looking on it. So that's why the strategy that we take in the Ministry of Health with all the ministries is to build the capacity to do research. is to bring on the focus and the coordinating center to do clinical research by establishing clinical research unit and center. We also bring the capability and capacity to our local players. We also see that incentives on research and development should be in place. That's why it will be complemented by collaborative network to make the efficiency in the place. Next slide. So this is what we take on the regulation. That point, that standpoint, we take to the regulation. So for the very big achievement that we have last year, we just established new law on health, law number 17 on 2023, which remain as a fundamental regulation, which bring the most basic guidance for all people, not only from public, but also from private, that can use this law as a guidance. Where in the law, it stated that one of the work to do to increase the production capacity, to have ecosystem, is doing the promoting and making partnership with global agencies. And by understanding this law that put big point on resilience, we bring on the government regulation. We develop the implementing regulation that we call government regulation number 28 this year, established this year, that make a history as the fastest government regulation being finalized as implementation of the law. You may check from the timeline, we able to finish the regulation not more than 14 months after the law being enacted, that providing you sense this is a big motivation from the government to do activity that focus on resilience. Some actions have been pointed on the regulations like supply chain governance. We will manage the supply chain in more integrated way. There's also pointed on national health security to R&D. So we request and we will direct the R&D activity directed to the national health policy on security. We also will prioritize the local use, the local production use, local product use to enhance our capacity. And to be able to deliver that points, they will be providing incentives, they will be built a research ecosystem and a risk mitigation related to the product. Next slide. So we would like to go too deep on the law itself. So in the law on health, we're going more deep that prioritizing the use and fulfillment of domestically produced product as a guidance of the regulation. So this sentence will make a signal for everybody that Indonesia will put priority on our local product. So that's why we encourage any active participation, collaboration that elevate our national capacity so that it will align to the market access. In the government regulation, as implementing regulation of that law, we also put the local use as a mandate for any related health participant or health stakeholders on providing quality, safety, and useful product in Indonesia. So we see on developing the health services, we prioritize on local product. We take the measures of uptake level of domestic components as a mark that you are using the product that locally produced in Indonesia. So this is the sentence, the regulation that we put to market, to shape the market of our local production, including vaccines. On the next slide, not only on the level of those regulations, but on the level of implementing or technical regulation, there is also mandate to maximize the use of local product. So in presidential instruction level, on decision of Minister of Health level, we put a same frequency, a same mandate to enhance the use of local production. So this is what we are using now to shape the market, to direct the market, so then the R&D activity that result in product may be utilized more to fulfill the need of the country. Next slide. So that's why in this slide, we make the collaboration center. Previously, you may see from the video that we are joining effort from researchers, from regulatory agency, from industries as a network together to collect the potential and to do a synergetic effort. We invite colleagues from researchers and hospitals to join us on the collaboration that we call Volare. You may recall this as a title from a song, famous song, I guess, Volare. So Pak Tantowi know very well about this song. And this song means that we're going to fly together. We're going to fly together to elevate the ecosystem with researchers, with manufacturers, with stakeholders to elevate the Indonesia ecosystem of life science, including vaccines. So next slide. So this is one of our achievements that we believe it is not easy to elevate the ecosystem. That's why we work with the big hands. We work with many agencies, and we do hope today activity with Tsinghua University will add another value of our collaboration. We had experience working with BMGF, with CEPI, with MPP, and now we look experience to more collaborations with Tsinghua University to add the value of collaboration and to elevate the research and manufacturing capacity of the nation. So I will end my presentation by the next slide that telling us that this is the beginning of our journey to living up the ecosystem Indonesia. To do this, we need collaborative efforts, and that's why we look forward for this forum as a continued effort on collaborating to increase our local capacity and of vaccine, R&D, and manufacturing in Indonesia. Thank you very much, and good morning.
Yeah, thank you very much, Pak Roy, for your presentation. It's very interesting presentation for today, and I'm sure that you have something in mind to ask for Pak Roy, but please hold because we will go to the next speakers for this morning. Professor Masteria Yunofil Saputra from National Research and Innovation Agency, with the topic is BRIN's role in research and development and long-term planning for vaccine, and also opportunity for collaboration with Tsinghua University. A brief introduction for Professor Masteria. He have an academic background in biotechnology and earned his master and doctoral degree in Marche. Is that right? Spelling? Yeah. Marche Polytechnic University in Italy. Following his undergraduate studies in chemistry at the University of Andalas and also in 2023, he was conferred as a research professor by BRIN. This is reflecting that his significant contribution in this area. And also, Prof. Masteria now serves as head of the Vaccine and Drug Research Center at the BRIN, and he have a very extensive experience in coordinating and leading research program, especially in biotechnology. Please welcome, ladies and gentlemen, Prof. Masteria. Please.
Thank you. Thank you very much. Good morning, everybody. I would like to thank you to Pak Roy and Directorate Pharmalkes for organizing this such wonderful meeting today. And Pak Tantowi, terima kasih. And all of the audience in this meeting today. And first of all, I would like to say thank you. I apologize for all of the Tsinghua delegation because I was in Pontianak yesterday and you attend to BRIN. Yeah, and yeah, I'm so sorry for I'm not attend your meeting with BRIN yesterday. So as moderator mentioned, my name is Masteria Yunofil Saputra. Right now, I'm the head of Research Center of Vaccine and Drug. So next, maybe you can. So as you know that maybe some of my colleagues presents about our research organization for health. So we are BRIN is we are the technical research in the BRIN. So BRIN has two policy, one of the scientific authority, one of the policy authority. So one of for the scientific research of the health, we call it Research Organization for Health. So the chair of the Professor Dr. Indi Damayanti, and Research Center of Vaccine and Drug is one of the 7 research center in Research Organization for Health. So I'm the head of Research Center of Vaccine and Drug. So one of our main tasks is doing a research with vaccine, biopharmaceuticals, and also drugs. Next. So this is our strategic plan of health in 2025-2029. So because 2024 almost end of in 3 months, in 2 months maybe. So in strategic plan of our research, we have the 6 main project in our research organization right now. So first is vaccine and drugs. So this is very important right now. Indonesia since the pandemic COVID-19. So Indonesian governments aware about how we make the accelerating of research and innovation, especially for drugs and vaccines. So we not demand from the other country. So the rest is precision and regenerative medicine. So we are talking about genomic, artificial intelligence, everything. So public health and medical device and disease detection. So medical device also, for example, like we developing also molecular antibody also for the disease detection, and also infectious disease is one health study that we are studying right now in our research organization, and herbal medicine and phytopharmaca. I think we cooperating also with some Chinese university also for phytopharmaca and biopharmaceuticals. Next. And this is 10th our project right now in BRIN 2025-2029. So yes, one thing that we adding in our strategic plan is sport medicine technology. So this is the new platform that we're working right now in BRIN 2025-2019. But mostly, yes, we are working with drugs, vaccines. Also, we're working with genome. This is very important. We support also Ministry of Health for the genomic studies also in Indonesia. So next, this is our research center. So we have in our research center, we have 6 groups. Okay. 6 group. So first, the cell biology and disease mechanisms. This is mostly related with drugs. So we're working with mechanism of action, some of drugs, and certain activity relationship of some of drug repurposing also study. We're working also in our research group, and nanomedicine, because right now is mostly some of the drug like using the liposome, nanovilosome, and everything. We're studying also in our research center right now, and we cooperation also with some of Indonesia partnership in US also, and we're working at marine natural products, because Indonesia is large biodiversity of marine, so we're focusing also in our research center is about the marine natural product, working mostly related for anti-cancers. And this is vaccine. We're talking about the vaccines today. So vaccine design and gene delivery and biosimilar. This is our focusing on our research center, and drug design and discovery. So we're using the artificial intelligence also. We're using how to design the structural activity of the compounds, make the new synthesis and everything, and doing the cell disease mechanism for looking the structural activity. And this is diagnostic. What we are doing is what we see here is we're focusing with infectious disease, cancer immunology, immunotherapy also. So we're developing also CAR T cells also in our research center, and tissue engineering and monoclonal antibody and stem cells also what we're doing. Next. So vaccine research in our research center for vaccine drugs, mostly we are working with 2 related diseases right now. So TB, tuberculosis, and dengue. So what we are focusing on, because Indonesia is increasing of demands of the patient for the dengue and TB. That's what we are focusing. But maybe we are related difference with the others platform. So we working with, maybe I will talk later in my presentations. Next. So this is what we see here with the vaccine type immunization process. You see there is some of the DNA, mRNA protein, viral vector, and you see the platform recombinant protein. We're focusing on recombinant protein here, also in Asia, but we have the collaboration also University of Queensland and Sanofi in Australia, focusing with mRNA tuberculosis. We started this year with Sanofi and University of Queensland for TB, also for mRNA. So what we are focusing in Indonesia is, you see that there is the sum of the platform, the bacterial, yeast, and mammalian cells. But we are focusing in Indonesia right now. The plan is tobacco and the microalgae. Why? Because Indonesia is large amount of the tobacco. So we cooperation with some university, some of the industrial partner in Madura for the greenhouse tobacco and microalgae. So why is microalgae? I will tell you later. Next. So we're focusing our design vaccine for microalgae protein. Okay. So I think there is 2 industry right now that producing there is one of industry that producing the microalgae based on the vaccine right now, I think from Australia, yeah. And why we starting the microalgae? Because we have the 10,000 collection of microalgae and in Indonesia, in our brains. And microalgae we producing using photobioreactor, okay? They cultivated using the carbon dioxide, okay? So they need the carbon dioxide for growing, and we planning to how to next target our deforestation in Indonesia. See, so we make the some of the large later. Our ambitious idea is to make large plantation of photobioreactor of microalgae to trapping of the carbon, to collaboration with industrial partner later. So that's what our target in our ambitious target with microalgae. And we focusing with dengue. So we already start the construction serotype 1 and 2 microalgae, and we plans, we already have the photobioreactor 200 liter in our research center. So growing with some of the microalgae that already construct with some of the dengue serotype 1 and serotype 2. Next. And this is one is the vaccine that we're working is PLP tuberculosis antigen vaccine. We hopefully 2026 we already have some formulations and doing some animal experiments. But this project starting in 2023. Next, and this is one thing that what the new technology platform that we study in our research center, silkworm. Yeah. In China, there is Silk Road. So we met to the Silk Road between Indonesia and China with silkworm. So why the silkworm? The silkworm is have the, if we growing the protein in silkworm, there are large amount of protein that growing in silkworm than other to bacteria or the mammalian cells. So that's why focusing in the silkworm. And Indonesia, there is some of the plantation of the some of the agriculture of silkworm. So it's such as in Bogor, Sukabumi, and the others. So we started project with the silkworm with some of the with one of the startup company named Sutra Pro. They are based in the Bogor. So we started our project first foot mouse disease, but this is for animal cattle vaccines. What we are doing right now, and we already have the finish of the prototype. And right now we study the clinical trial or preclinical trial with large animal with the cow and with the with the vaccination with the cow. And this is what we're doing right now with, I forget the name of the English of the animal, but with large animal, what we are doing right now. So later, next year, we want to commercialize these vaccines. So this is 2024, we are finishing doing the clinical trial in 2025. We ambitious to commercialize the foot mouse disease vaccines. Next. And this is what we are doing right now, avian influenza vaccines also. So we did the zoonotic study between animals and human, right? That's avian influenza is very important right now, and one of the large cases in Indonesia also. So we base it on with silkworm. So why we use silkworm? Because in animal, because we can is edible to eat for the silkworm. So also microalgae is edible to eat. So what we are ambitious studies is how to make the vaccine to edible to eat. That's why we started with microalgae and the silkworm. What we are doing right now. Next. This is what we are doing is recombinant vaccine for recombinant development of the adjuvant for what we're doing. Next. And next. Yeah, you see, you already see facilities, right? From the yesterday, you already see the cryo-EM, you already see animal biosafety laboratory, you already see biosafety laboratory, level 3 in breed, you already see the cryo-EM and genomic laboratories there. And I think you attend the GMP facilities that we have also in Cibinong. So this is our facility that support our research in vaccine and especially vaccine and drugs in brain. So we can collaboration with others partners, some industry, also some university in around the world. Yeah, not yeah. We open the collaboration with the Tsinghua together, and yeah, we collaborate also with Ministry of Health. Next, thank you very much.
Thank you, Professor Masteria, for your impressive presentation. That this is what we like about research, because we can open any opportunity, and imagination is the limitation. So for the next speakers, we will have here Mr. Andreas Doni from Etana Biotechnologies. He will deliver the topics about current capacity and future expectation for vaccine ecosystem in the research and development industry. This is also including the collaboration with Tsinghua University. Brief introduction to Mr. Andreas Doni. He is a graduate from Wageningen University from the Netherlands, and he also now one of the figure in biotechnology industry in Indonesia, and also executive in PT Etana Biotechnologies Indonesia. And he's playing an important role in the driving innovation solution in the field of biopharmaceutical in Indonesia with PT Etana Biotechnology. Please, Pak Andreas.
Thank you, Pak Indi. Distinguished Bapak Tantowi Yahya, distinguished Professor Zhang Lingge from Tsinghua University, distinguished Professor Elina Burhan. Good morning, Prof. Elina, and distinguished all the participants here, and also Pak Roy. Thank you for happening today's event. Very wonderful. We can become one here in Indonesia. So today I'm on behalf of Indonesian Association of the Pharmaceutical Company. I will try to deliver what's about the current capacity and the future expectation for the vaccine ecosystem and the research and development in the industry field. So next. So before we talk about the biopharmaceutical things, we have to look about the condition of Indonesia. So now Indonesia have 279.1 million population. This is the world fourth most populous country in the world. And also, this is the most populous Muslim majority country in the world. So it means if we develop one product here, we will sell the product in all over the world, especially the Muslim country. The total population of the Muslim country is about 1.8 billion population all over the world. So this is very big size of the market, especially for the vaccine and the biologic product. Mostly of the Muslim country, they don't have like the good facility to produce and to fulfill their needs of the biological product and also vaccine for their population. So this is also the chance for the Indonesia and also China in the future. We can work together to fulfill the market of the Muslim country, especially with the halal market product. So we can see also the GDP is still very optimistic. This is 4.94% year on year. So this is quite high with the expectation until 5.05. And the price increased like 115.40 from CPI. And the inflation is not too high, 2.28% annually. And the financial, we have 134.9 billion foreign exchange reserve, and also the lending rate is quite stable in 6.5%, and the customer confidence 121. So it means the buying capability of the Indonesian population is still quite confident and high. So this is quite promising market for the pharmaceutical company here. Next. So this is very strong encouragement from the government. Why? Because from the President Jokowi, he already has very big commitment for the pharmaceutical industry with launching the Indonesian government has institutionalized the changes of the promote local manufacturing of the pharmaceutical product. So all the pharmaceutical product that will sell in Indonesia, the government encouraged to produce locally here. So this is very, very good event. So Indonesia, through Ministry of Health, Indonesian FDA, and also Tsinghua University can make a collaboration to produce the product and to do the research locally in Indonesia. So this is in line with the direction of the President Joko Widodo. With the decree number 6, 2018, it called accelerating development of medical device industry and also drug. So the government is already have the very good way to increase the development of the medical device and the drug in Indonesia. And also with the law of number 33, 2014 about the pharmaceutical product must obtain the halal certification. So this is also the chance to develop the new product of the biologic product that has the halal certification from Indonesia. So this is a good promising for all over the world, especially for the Muslim countries. So with the local requirement, the drug local content will give priority to get them listed in product tender over imported product. So the local content regulation will give more incentive for the local manufacturer here. So the minimum value for the local content requirement is 40%. So there is no official guideline published, but government has already started using the local content requirement criteria in public tender. So all the public tender in Indonesia now is already based on the local content requirement. So if we will produce someone or something, or especially the biopharmaceutical product, the local content will be the most important part that we have to consider together. Next. So this is the pillars of the health transformation that's published by the Ministry of Health. So for the biotechnology, we supporting the increase of resilience of pharma and medical device sectors. So the government already has the target to domestic production of 14 routine vaccine of top 10 drug and top 10 medical devices. And the government already has the target of the health technology transformation. Next. So this is also still very high in Indonesia, the condition of the cancer. So the cancer can be handled by the vaccine and also the biopharmaceutical product. So this is the condition in Indonesia. The top 5 most frequent cancer is the breast cancer, lung cancer, cervical cancer, colorectal cancer, and the liver cancer. So all of this now already has the new therapy, both with the vaccine for the prevention and for the curative, we can use the either chemotherapy and also the biological product. So for our knowledge, in Indonesia we have 228 industry. 228 industry. Most of them is the chemical industry, pharmaceutical industry, the chemical. So just 5 in Indonesia, we do the biological product production. So it's about 97%, all of them is the chemical. But now the condition in the world is changes. 65% of the economical of the pharmaceutical industry taken by the biological product, including the vaccine. So now this is the right time for Indonesia to change this, to switch the industry from the chemical pharmaceutical industry to the biological industry. So this is very, very good time for us to discuss. Next. So this is also because of the global prevalence of the tropical disease. It is changes because of the climate change. We can see increase of the rainfall, increase of the temperature. It can make the increasing of the malaria, dengue fever, and many tropical disease. So it should be tackled by the product of the biopharmaceutical and also the vaccine. Next. So from the perspective of industry, this is the challenge for the biopharmaceutical and therapy development in Indonesia. The first one, the challenge is the capital and infrastructure. To develop one pharmaceutical industry, we need 10 times, at least 10 times capital compared to the chemical pharmaceutical industry. This is very big chance for us, like for Itana Biotechnology. We know our investor is coming from overseas. Why? Because the investor from domestic, they don't really brief enough to invest in the biopharmaceutical sector because there are the people said this is the green field. So there are no development yet in this field. So this is the chance for us to develop ecosystem in the biopharmaceutical industry. And the second one is the inadequate infrastructure for the vaccine production and also genomic research. So from the human resource perspective, we have lack of the new and specialist knowledge of the capabilities. This is also should be fulfilled by the Indonesian countries. And also we have lack sufficient expert and trained personnel in the field because for us, for industry, if we hire someone from the university, so in fact, there are not ready directly to use. We have to train for them at least 1 or 2 years. But for the some product, we have also to send our employee to overseas to just learn in the production side. So for the regulatory framework, our FDA also still catch up very fast to get catch up with the another standard in the world. And also this is also the big problem for Indonesia, the ecosystem and supply chains. So unavailability of the local material, equipment, and services, while inefficient custom process delay the import critical material and equipment lead to higher cost and longer lag time. This is also one of the biggest problem for the industry. This is also another problem, the testing and evaluation. We can produce the product here, but for the testing and evaluation, we have the limited testing capacity, and also we have challenges with the evaluation standard and also high cost of testing because what? Because the ecosystem and supply chains, we have still the problem on that. So for example, for the testing price, for the release one batch of the biopharmaceutical product, the price in Indonesia is 5 times higher than China. So if we can solve this problem, so it can make the local production or local product here is more competitive. Next. So I think we can do the collaborative measure on the increasing local capability. The first one, what we have to increase in Indonesia is development of the local testing and evaluation capacity. So we can just empower the incubation and local CRO, and then develop the local animal subject breeding capacity in vivo tests for the release of the pharmaceutical product here. So we don't have to send it again to the overseas. So it will be efficient either for the cost and also for the time. And maybe we can just expanding biopharmaceutical industry in Indonesia and has increased the need for the routine in vivo animal testing in Indonesia. So the second one, we can just also increase the effectivity for the local clinical trial. So this is also the field we can increase together. And we can build and bridge resource Indonesia and also China. So we can just develop the human resource development through degree and also non-degree program, joint research program, short course and also training. And also we can also do the bridging outstanding of Indonesia and China industry, because the people to people contact is very important. If not, there will not any strong cooperation between these two countries. So between the academician and investor along Tsinghua ecosystem for the better competitiveness of Indonesia vaccine, genomic industry, and also development. From this event, Tsinghua already know the condition of Indonesia. You can bridge us to the many investor in China to develop the lab, to develop the product, to develop the R&D. So this is very important between Indonesia and also China through Tsinghua University. Next. So, yeah, I think this is not a short time to develop a biopharmaceutical product, but this is the beginning, just the beginning. So I think we need the long-term strategy and policy support for the vaccine and genomic development. The first one, we need to develop the research and also development. We need acceleration of the development of R&D and also ecosystem and technology transfer facilitation. So for the effectiveness, so we have to has the hub for the technology transfer and facilitation for the vaccine clinical trial, sensor facilitation, we also need. And also for the production side, we need the simplification from the Indonesia FDA for the licensure process. And this is very important, the fiscal and non-fiscal incentive for local production and use for the local raw material. And also we need technical facilitation also for the local manufacturing, especially for your export from Tsinghua to Indonesia. And this is also very important for the business side, for the market assurance for the local product. So we can substitution of the imported product. So for the government, they already do the freeze unfreeze mechanism for the procurement from the local content and the imported content, and then implementation of the local content, PKDN, and affirmative action to increase the use of the domestic product. Next. Okay, that's all my presentation. I hope it can be used for next discussion. Thank you, Shishe.
Thank you, Pak Doni. He emphasized the unique position of Indonesia in the Muslim countries, and especially that Indonesia have a pharmaceutical industry. And if we in Indonesia and also together, we can grow the vaccine and also biopharmaceutical industry in Indonesia, and also can be used in the another Muslim countries. I would like to inform you that Professor Budiman Bella cannot join us for this session. Maybe we will have another chance to Professor Budiman Bella to deliver his topics. And now, thank you very much, Pak Himawan again, and Prof. Masteria, and also Pak Doni for your presentation. Now we go to the next agenda is questions and answer sessions for these first topics. So we invite if there is any questions from the audience for Pak Bonanza, Pak Himawan, or Pak Doni, or Professor Masteri, please. Yes.
Good morning. Thank you for your interesting talks. My name is Dani from BRIN, and I would like to ask to Professor Mastria. My question is, you developed the silkworm as the host for protein production. My question is, did you use local strain or commercial strain of the silkworm? And the second is, as far as I understand that I also work with silkworm, and we need to sterilize the medium and also the product, because sometimes contain buccal virus. And how to solve this in your laboratory, or if you want to produce in large scale, how to solve this? Thank you very much.
Mr. You want to answer?
We work together with the farmer from in Bogor. There is the farmer silkworm in Bogor, and we have collaboration with Ministry of Environments. Yeah, in Ministry of Environments, they have the large farmer of the silkworm there in Sukabumi also. So we use the silkworm strands local Indonesia. So I will tell you something that we already compare the Indonesian silkworm with Japan silkworm, because previously we were working collaboration with Shizuoka University using the silkworm, and you see there is some of the company in Japan's name is Kaiko. So Kaiko is one of the pioneer of silkworm in the world, I think, yeah, because then they already have the approval from Japan's authority for the pig influenza or something like that, pig vaccine, something like that. So they asked us also to use the Indonesian silkworm, because what their look is, the Indonesian silkworm is most producing of the protein than the Japan silkworm. And right now we already have 5 collection of the strand silkworm from Indonesia. we starting also to do some of the host of transfection using the protein right now. But this is locally strain originally from Indonesia. So we already have it. So we don't export the silkworm, import the silkworm from other country. So the silkworm from Indonesia. The second is, this is a good question. Why, how to sterilize? So right now, if you go to the Serpong, right now in the next week, we already make this collaboration with Sutra Pro, some small production of the silkworm. So in ELC, Indonesian Life Science Center in Serpong, in KSTB Habibie. So that's startup company already rent some of the space there, and they are already starting to growing of the silkworm using of the standard. And one thing that's interesting with the silkworm, we don't need the reactor. We just need the incubator. So it's more cheaper than bacteria. It's more cheaper than cell. It's more cheaper than yeast, because we just need the bioreactor. We don't need more from sample from media, everything. Because what we are doing is transfer the baculovirus, not in silkworm, but in I forget the name, in the pupa from silkworm. So we don't need to worry about the sterilization. So because we don't need the whole bioreactor, but in all of our study in our project, we working in the clean room, in biosafety laboratory level 3 and standard of GLP laboratory. Thank you.
Okay, could you please tell us about the yield? I mean, as far as I understand, from one pupa, we only get like 0.4 milliliters of serum.
We can discuss later. I have the
Okay.
presentation, then I will show to you. No
Thank
problem.
you. Okay.
So we already have made this some business plan about the silkworm from the beginning to under the production. That I will tell you. It's much more cheaper than bacteria and cell. My guarantee to you. Thank you. Yeah.
Okay, thank you.
Okay, thank you, Professor and Mr. Dani. It's very interesting question there. The startup on silkworm. We invite another questions. Maybe our colleagues from Sinhua, or yes, please.
Thank you. I'll follow up on the silkworm question. I asked your colleague yesterday, since the edible, your stress on the importance of edible vaccines, the protein made in silkworm when you eat it, what's the likelihood that can be digested by our proteases in the stomach? So if that can be digested, then and that may lose the antigenicity. So how you overcome that problem so we could have an immunity generated during this process?
Yes. We collaborate. We discussing with our team about that. How to the edible vaccine, not only with silkworm, also with microalgae. So first, we talking. We starting right now to using some adjuvants, and some of the maybe some of nanotechnology from like a liposome or something like that. That to maintain the bioavailability and the bioavailability of this edible vaccine. So we aware about that, but what we are doing in FMD vaccines, yeah, we already tested with guinea pig. We tested with guinea pig. We tested with the mouse also.
Edibles.
And the immunogenicity of the silkworm is high. And right now we tested also with the cattle, some of the cattle animals right now, and we still don't have any data with that because we already start in last months. We started in June, and with some of the kettles, yeah. But later, if I have the data with that, we can discuss with professors. Yeah, I think that's very interesting. So because we already got some question from us also for industrial partners. We discussed it with some of industrial partner for animal vaccine in Indonesia. And one of the question, how to maintain the digestion of that, because
Yes.
there will be antigenicity. So
This is a very interesting strategy.
yes.
But it's also important to develop immunological assays to
Yes.
evaluate
Yes.
how good immunogenicity how good is protect from infections or how long this immunity could last.
Yes,
So
we
those
can
are
work
the-
together and collaboration
Those are the
together
key
with.
questions, so we could go further after during the break.
Yeah, we can work and together we can collaboration together with it. Yeah, I think
Thank you.
this is very fun, interesting. And yeah, why our ambitious strategy with the silkworm is because right now we are decreasing the farmer of the silkworm in Indonesia, and the price is so higher in Indonesia. Then one thing is we work with thinking about the farmer in Indonesia also, and that's what one thing what we are focusing. And the second is the technology of the silkworm is already new also in the world. I think only Japan and maybe some of the researchers in China are working with the silkworm, and they are producing the large amount of the protein than the bacteria than the cells. And if you're working with protein recombinant, that's a really fantastic host of the platform that we working for the developing of the vaccine later. So one the problem with the silkworm is we still don't have the standardization standard GMP production of silkworm. Only one industry partner, I forget the name in Spain. They're using baculovirus also, but they're using the strain of the silkworm in Europe, and they already have one one approval of the vaccine, but also for animal from European agency, and one is from Japan's Kaiko. Kaiko, I forget the name. I think the name is Kaiko. Then you can see in the, and that's why in Indonesia have the large strand of the silkworm. And what we are doing right now, what we are doing a research where we find that large amount of the Indonesian silkworm have large amount of protein than the others, Japan silkworm and than the European silkworm. That's what our ambition is. Yeah. And we can follow up the collaboration with how to the maintain the immunogenicity and everything. Yes. Thank you, Professor.
Thank you.
Thank you, Professor. And thank you, Professor Masteria. Another question for the speakers. It's very interesting. Yeah, Professor Erlina, please.
Thank you. My question is to Roy Himawan. So, during COVID pandemic in 2020, the vaccine ecosystem and research development is really widening and price. And there are a lot of collaboration of pharma company, and also researchers from university and scientists collaborate to develop a new research, not only in vaccine, but also in new drugs such as monoclonal antibody, etc. So from my experience, we face a lot of challenges in making this clinical trial to be one of the good quality. Regarding to your position, there are challenges in regulation. So what have been done, established in order to have a good comprehensive regulation between Ministry of Health, BPOM, and also the MTA committee, because those three bodies is really the bodies that need to facilitate the love of clinical trial. So, yeah, please tell us what have been developed and done to help the scientists, because the regulation is really a challenge. Thank you.
Yeah, thank you, Professor Alina, for the good questions, and thank you very much for your kind and enthusiasm on the ecosystem of research of vaccines and biological product in Indonesia. You are right, Prof, that Indonesia faces challenge, and we do need to do something to accelerate the ecosystem development in Indonesia. So this also being realized by the minister, and he see that some the bureaucracy and the form of how the license being provided to the researchers is challenging. So then what do we observe from his direction is we need to revamp the law. We need to reinvigorate and recalibrate the law. So then we came with the law number 17 in 2023 that put all the sectors, that put all the stakeholders on the same page to develop this ecosystem. That's why on the law itself, we may see several chapters specially designed to move every stakeholders, including MTA committee, including Indonesian FDA, to work on the same page on elevating the ecosystem. So that's one thing that we have changed, because in previous health law, we may not see this attention. We may not see this focus, and by this new law, we have new guidance. So the second one, Prof, the Ministry of Health already realized that we need to accelerate that process, even though we have the law on the page, on the table, but we need to provide instruments and to have some programs and actions to accelerate the ecosystem. So then the first thing is we do benchmark, Prof. We do benchmark with the best international practices on how managing clinical trials better. Later on, Bu Indri will also explain us about what is had been done on the field of clinical trials. So then, based on this benchmark, we see to be centralized, the arrangement of clinical trials will be the most significant step, Prof. So we identify several factors that may elevate the trial ecosystem, and making it centralized, it will be the most significant one. So then the ministry assign and form clinical research center and establish more than 17 clinical research units across hospitals. So then any arrangement needed on trial will be more easy to execute it. That's the first thing, Prof, that put as an instrument to implementing the regulation. The next thing that are developed now, that we renew the MTA committee. We renew the MTA committee. We change the structure. We invite more and more practitioners like Prof and others who daily involved in trials to join the committee. So then the committee will have the same perspective, the same understanding on managing the trials. Previously, we see the committee consists of people who merely not on daily basis work on clinical trials, so they didn't know what is the relevance of this clause, of this clause, of this clause. So then by having it reinvented now, having it more people work in clinical trial on daily basis, they will understand how to manage the agreement, how to secure the needed aspect on sample without making burden on speed, on access, on quality trial, Prof. So then by having this, we are sure that we will have progress on this one, Prof. So as that we have jointly experience on managing the TB candidate vaccine on the trial, Prof. So that's the thing that we can reply on your question, Prof. Thank you very much, Prof.
Thank you, Pak Roy and Prof. Erlina, for the question and answer. Ladies and gentlemen, now we are already on the end of the first session for today. I would like to just a bit conclude the topic of discussion of this morning. Professor Pak Roy Himawan already mentioned about the focus of Indonesian government on the local production, and especially on the vaccine and also biopharmaceutical products in Indonesia. And Professor Masteria already mentioned about all of the research topics already being done in BRIN, and also the future research topic. And interesting topic is already talked before about the silkworm for vaccines. And maybe it's not only a Silk Road initiative, maybe silkworm initiative between Indonesia and China. And Pak Doni also mentioned and emphasized about Indonesian unique position in vaccine and also biopharmaceutical industry in Muslim countries, and the point of view of industrial sector about that. Before I close, I would like to read you about a poems, Indonesian poems. We call it pantun. Yes. Usually we use this in the closing of the topic sessions. So for Indonesian colleagues, you can help me with the cakep, right? And for Chinese colleague, you can follow. So the pantun is the waves embrace in Bali golden sand. Mountain rise and sky are blue. Cakep. In collaboration, hand in hand. Cakep, yeah. Together we make dreams come true. Right. Thank you very much for this session, and I offer to MC Ibu Putri.
Thank you so much. Please give applause to our amazing speakers and also amazing moderator. What a beautiful poem. Yes, Mr. Kindi. And speakers and moderator, please remain on stage as we will continue to take a picture together with speakers and also moderator. Please be upstanding in front of the table. Yes, we will continue with photo session with all speakers and also moderators. So please, photo video are ready. Yes, gentlemen on stage, please face the camera. 1, 2, 3. Smile. Yes, let's show our spirit and energy. Yes, to strengthen our collaboration. 1, 2, 3, smile. Okay, thank you so much, speakers and moderator, for amazing session that we have been through together. Please return to your seat. Thank you so much. Distinguished participants, ladies and gentlemen, now let us proceeding to the next plenary session, the second plenary forum that will elaborate about vaccine ecosystem for research and development from Tsinghua's perspective. And to guide this session, I would like to invite distinguished moderator. She is member of the Infection and Immunology Cluster, Indonesian Medical Education and Research Institute. So please welcome on stage Professor Dr. Erlina Burhan, MSc SPP. Please welcome on stage our distinguished moderator, Prof. Erlina. Well, ladies and gentlemen, now I will let the stage to our moderator, Professor Erlina. Stage is yours, Prof.
Good morning, everyone. Thank you for the opportunity. Pak Tantowi Yahya, terhormat, Professor Lang Vikin, and also all the audience and participants. Let us proceed to the second plenary. We have heard our Indonesian expert talking about vaccine ecosystem and also research and development. Now let's proceed to the second plenary, and we will hear from our expert from Tsinghua University. Without further ado, let me call upon Dr. Yu Guo Chan, Dr. Zhang Changzai, Dr. Fan Shilong, and Dr. Deng Haitang. So doctors ready to share experience and also your expertise, yeah? Okay, good. More than ready, okay. Okay, let me introduce to you our first speaker, Dr. Yu Guo Chan. Yeah. Dr. Yu is from the Department of Pharmacy, Tsinghua Hospital. He had experience working at the National Institute of Health, USA, and also joined Tsinghua University in 2020. And three years after that, he selected as the most cited researcher. And then also the top 2% of top scientists in the world out of 100,000 scientists. I hope I cover all your CVs. Okay, please present.
Okay, thank you. Good morning, ladies and gentlemen. It's my great honor to introduce the fundamental research in my lab. Today, I want to share several stories about the lipid nanoparticle for mRNA vaccines and therapeutics. So I'm Guo Chanyu from Tsinghua University. Next. Okay, last year, the Nobel Prize was awarded to Professor Joe Weissman and Dr. Kathleen for their contribution to the mRNA modification, which can reduce the side effects of the mRNA so that we can use this technology for the preparation of mRNA vaccine during the pandemic. And compared with the small RNA such as sRNA and ASO, mRNA can express the protein inside the cells. These proteins can be used as the antigens and the antibodies. So we can use mRNA for the treatment of cancer and genetic disorders. And next, please. Next, please, okay. However, the mRNA molecules cannot be used directly because it can be degraded in the body. So there are several physiological barriers we needed to overcome. We needed to modify the mRNA through chemical modification so that we can increase the stability in vivo. And we can also use suitable delivery systems to deliver the mRNA into the body and into the cells. In my lab, we are focused on the delivery systems because I stay in Department of Chemistry. So please, next. We can roughly divide the delivery system into two parts. The first one is the viral vectors. These vectors are always highly effective, but the safety is a problem. The other one is the non-viral vectors, such as the polymers, the lipid, and the exosomes. Among these materials, we choose lipid nanoparticles as the delivery system for the preparation of vaccines by the Moderna and Pfizer because they are quite cheap. They can prepare easily. Next. However, the mRNA vaccine based on the lipid nanoparticles still face some limitations. The first limitation is the high liver accumulation. After intramuscular injection, a lot of vaccines will get into the liver and it will induce the side effects. And we can also see some reports about the side effects after vaccination. And the next is. And the second problem is about the efficiency. For mRNA vaccine, it takes several steps before they can work. And the final efficiency are always very low, maybe lower than 1%. So we need to increase the injection dose. For the infectious vaccines, it's okay. We only needed to receive 2 or 3 doses. But for cancer treatment, we needed to increase the dose, maybe for one person, for one injection, maybe the injection dose is around 1 milligram RNA. So the injection dose is very high. And for the Pfizer vaccines for melanoma, the patient needed to receive 10 injections. So if we increase the injection very high, and the side effect will cannot be ignored. Please, next one. So in my lab, we are focused on the design and synthesize new lipid, and we want to prepare new formulations which can targeting the cells, the organs, so we we can prepare more precise lipid nanoparticles and among vaccines, and we can also incorporate the functional groups into the lipid through chemical modification, so we can increase the efficacy of our vaccines, and we can use our formulations for the treatment of cancer and other disease. And the next one. The first story I want to share is about the albumin hitchhiking. For the traditional LNP, there is a very special lipid. We call it PEG lipid. It can stabilize the LNP, but it will detach from the LNP after injection. And a lot of protein, especially for the ApoE protein, can absorb on the surface of the LNP. And they can across the blood vessel and get into the bloodstream and then get into the liver. Here we replace the PEG lipid by a very special lipid. We call it a THU lipid. This lipid is very special. We have a purple group. This group can complex with the albumin in the body. So we can avoid the liver accumulation, and we can increase the accumulation in the lymph nodes. And the next one. Next one. Okay. The mechanism behind this phenomenon is about the protein corona. As I said before, the purple group can complex with the albumin, so we can change the protein on the surface of the nanoparticles. And for our nanoparticles, they can stuck around the blood vessel. They cannot get into the bloodstream, while it can be very easily get into the lymphatic vessels. So we can increase the accumulation in the lymph nodes rather than the liver. Okay, next. So we test our formulation in mice, rabbit, and pig, and especially for the PET imaging of the pig, we can find that the accumulation in the liver can be avoided if we use our formulation. Okay, next, please. So because of the high lymph node accumulation, we can prepare cancer vaccines. So first, we prepare the cervical cancer vaccines, and compared with the commercially used LNP, our vaccines displayed some advantages. We can prevention and treat the HPV-based tumor. Next one. And we can also use this platform to prepare other cancer vaccines for colon cancer and oral cancer. And we also test the combination with chemotherapy and immune checkpoint blockade. And we can find the advantages of our vaccines. Next, please. And we can also use this platform for the preparation of infectious disease, such as the flu and the COVID-19. And we also test the efficacy and find some advantages. Okay. So next story is about the inhaled lipid nanoparticle. We are find suitable lipid nanoparticles for pulmonary mRNA delivery, because we want to use our mRNA technology for the treatment of fibrosis or the lung cancer. So we needed to find suitable lipid nanoparticles. Okay, next. So for inhaled lipid nanoparticles, we needed to overcome a barrier. We call it MUX. The MUX is a gel-like liquid containing a lot of proteins and polymers, which formed a network-like structure. So we mimic the migration behavior of our lipid nanoparticles in the MUX, and we find that the size is very important. The nanoparticles is around 200 nanometers is the best size, because we can balance the stability and the migration in the MUX. The next, we also needed to optimize the charge to increase the transfection ability of our lipid nanoparticles. And so we optimize the ratio of each lipid components, and we find the best one on the corner. And we also test our lipid nanoparticles in the rabbit and in the pig, and we can find the efficiency is quite good. Next. And with this LNP in hand, we can use these nanoparticles for the preparation of mRNA vaccines, and we can also use our platform to deliver the mRNA encoding the cytokines, so we can use them to treat the lung cancer, and we can find that both of them are quite good. Next one. Okay, the third story is about the pancreatic targeting lipid nanoparticles. As we know that the pancreatic cancer, the treatment is very challenging, and the 5-year survival rate is very low. So we want to find a new lipid, a new particle which can selectively deliver our drugs or our mRNA into the pancreas. So we almost test around 1,000 lipid nanoparticles, and we find a very special lipid which can deliver our mRNA very specifically into the pancreas. And we also test on mouse, pig, and non-human primates, and especially for the monkey, we can find very good targeting ability. Okay, next one. So we can use our nanoparticles to deliver mRNA. Here we use the interleukin-2 based mRNA. We can deliver the mRNA very special, very selective into the pancreas, and we can change the therapeutic effect, and we can combine our strategy with CAR T therapy and the mRNA vaccines, and we can find it's quite good. The next one. The fourth story is about the mRNA-based cytokine therapy. As we know that the cytokine are already widely used in the treatment of cancer, for example, the interleukin-2, but the clinical application is also limited by the short half-life, the side effects. So here we find a new way that we can change the therapy. We put the LNP encapsulating the mRNA encoding the interleukin-2 cytokines and put the LNP into the gel. So we can inject the gel in the tumor site, and we can express the cytokine in the tumor. Okay, next one. Okay, this is the anti-tumor study, and we use our formulation for the post-surgical glioblastoma treatment. After surgery, we put the gel in the tumor site, and we can prevention the tumor relapse after surgery. And the last one. Okay, we can also lyophilize the gel, and so that we can store our gel at room temperature for a long time, and we can use as an implant after surgery. So it's very easy to use. Okay. Okay. The next one. Okay, at last. Thank you. Thanks for your attention.
Thank you, Dr. Yu Guochan. You will get so many questions later on. Okay. Our next speaker will be Dr. Chang Zai. Dr. Chang Zai is Associate Director of Laboratory Animal Resources Center, School of Life Science, and he is a senior engineer. And he actually doing a lot of activities to support animal research. But to make it short, I would like to mention that he has been continuously evaluated as excellent in the annual assessment organized by the School of Life Science at Tsinghua University for many years. Excellent for many years. So, Mr. Excellent, please deliver your excellent presentation.
Thank you.
Thank you. I'm an associate director of the Laboratory Animal Resources Center and service for Tsinghua University. The next, the topic is for content. Next, I took to support our animal welfare and the special technology. Okay, our member come from multiple department of Tsinghua University. Next, history of and qualifications. Our center built in 27 and 9. We have our facility have ALAC accreditation and CNAS accreditations. We have used the animal cages, namely 47 cages, and the mouse, the kind of animals is mice, rats, and ferrets, and rabbits. Next, in China, the animal activity are be licensed by the government and for laboratory use and production. Next, this is a very design and a total area. We have 41. We have 41 cages in total. We have first one, the purchase animal experiment area, and another facility is genetically engineering mutant mice breeding and experiments. This is our field wash center and barrier animal facility. Next, the fresh air machine room and tennis work. Next is our facility is IVC 100 and the automatic drink facility. Next is of chart organizing chart of a lot. We have next. We have 5. Next, we have 5 platforms provide multiple service and animal breeding, the mice, and we can do animal import and export. A facility maintenance to provide temperature and animal control maintenance and requires and embryo micro manipulation. We can do transgenic kill mice and the phenotyping center, and we can do botanic the instrument training and the usage, and the vet care and the quality animal quality control and animal welfare. Next, each example is embryos micro manipulation. I can do IVF based different service and microaggression and micro injection best Cas9, CRISPR-Cas9 knockout, and the ESL injection and different development stage, and the cryopreservation of embryo and the spore. And we have B6 mice production. Next, this is an example of IVF for rapid mice. We have 1 male mice can produce 1,000 puppies. Next, multiple different injection ratio. Next, phenotyping center, we have 40 plus behavior test, including sensor to sensor, motoring, and emotion, social rhythm, and learning and coordination. Next. So we have 10,000 cases through the mice software to remote monitoring mice breeding data. The student can didn't need to go to the annual facility. Next. Our vet care, including animal environment, water, and the feed quality control. We have to lots of things. The room are classified for different interests. The mice, rats, guinea pigs, rabbits, rats. We have another training system for researchers ensure the faculty and the students can start animal deployments within 2 weeks. We have lots of training. Next. We have another training system for staff. The staff knows the latest development has mutual understanding between platforms. Next, we have 5 platforms that we have must improve our technique. So we have continuous high quality development through the SNES accreditation. Next, thanks. That's all.
Thank you, Dr. Chang. And our next speaker is Dr. Fan Shilong. Dr. Fan Shilong is the director of the Life Science Facility Office, School of Life Science, and also has experience working in Cambridge University, UK, and then returned to Tsinghua back home, and then do a lot of things again to make it short because it's very long CV. Yeah. His mainly research work includes providing macromolecular crystallography facility and technical services for structural biology researchers at Tsinghua University, and as well as life science research of whole China based on the high-end equipment and technology advances of the National Protein Research Center. Please, Dr. Silong, present your presentation for 15 minutes.
Thank you. It's a very great honor I can come here to show our research center. Actually, my presentation is considered 3 parts. The biggest part is introduce our protein research center. Another part is about the structural biology in the Tsinghua University. And the last part is a real case about the structural biology adolescents. So next. So this yesterday we visited another institute in Indonesia. I really consider this. We are very similar. We are all research center. We are very fresh research center. Actually, in our protein research center, we don't have very long history. We set up this program at 2008, and our first funding is coming from 2012. And actually in the 2015, the first funding has been finished. We set up the original research center. We call it Protein Research Technology Center. After that, we got more funding from our local government, our university. So actually, to 2023, we're still building up our research center. Next. So our mission at the beginning, our mission is very clear. We will provide the facility service and the technology service for our university. Not only our university, also for the whole country in China, and also currently we also provide our service to the all over the world. Actually, we currently have some user coming from outside China, from Hong Kong, America, some country like that. Yeah, so currently we have established standard automatic and genetic to enhance the level of the protein structure analysis. Because at the beginning, our dean is Professor Shi Gong, so he is focused on the structure biology. So at the beginning, our research center, our major research is about structure biology. But actually, after several years building, we still can have a lot of facility for the whole protein science. So next, please. Yes, this is the overview of our national center. So we are one of 9 national project for large research infrastructure. Actually, we are the only one about the life science. So currently, we focus on the structural biology. We mainly also use cryo-EM as a major research tool and integrate some other methods, include the crystallographer, MR, and also we noticed that the protein is mostly important. So we set up all the facility about the functional and the protein, and we need analysis of the function of this protein. Yeah, please next. Yeah, this is our organized. Actually, we belong to the Tsinghua University, where all our human resources is belong to the life science. We currently have 10 subunits. The biggest one, the bigger part is a cryo-EM facility, and also we have another crystallography platform, MR platform, and also we need to check the function of this protein if it's fully functional and about the protein interaction. So we have a mass spec about computer facility and a cell image facility. And because COVID-19, we also set up a public P2 laboratory for the whole university. Yeah. This is our lab area. Our major equipment are located at the biomedical science building. Actually, we have some other equipment located in another 3 buildings, include the life science building, medical science building, and the biotech building. This is some picture gallery about our equipment, include the X-ray crystallography facility, the cryo-EM facility, and some others, include the mass spec. Yeah, please. Next. As a team member, we have a large crew within our team member, and in recent years, we got a very stable team members. As I mentioned, the equipment is not that difficult to buy it. If we got enough funding, we can buy all kinds of facility we can have. But the team member is mostly important than anything else. So we can see in recent 2 or 3 years, our team member is very stable. And also the distribution of our staff is also has different ground. Most of them have the doctorate degree, and we have 24 people have a doctorate degree, and the master's degree is 29. So this is the major technical support of our core facility. The largest part is cryo-EM. Actually, we have 4 Titan Cross in our research center. And totally, we have 10 electrical microscope and 4 dual beam microscopes. Yeah, please. And also, we have the totally full functional facility about the crystallographer and the MR facility. Next, please. And also, some supporting systems include the mass spec and the and Professor Tekana will introduce our mass spec facility next. Please, next one. So we are not only just provide a service, we also encourage our staff to do some innovation development. In the 2023, actually, in our research center, we have our own instruction function development. We have 19 development, and also we writing the protocol for our new user. We can see here we can We have some customer experiment training protocol. We have 95 protocol for our new user. And also we have our own corporate. In 2023, we have another two commercial one. The corporate we oscillate to industry user. And well, I need to mention here, although we have such a lot of equipment in our research center, we have very major instrument reservation system and a training system. The training system is very important. Currently in our research center, we worked all the time. And this means it's 24 hours per day and for the whole year. So we separate our user into 4 brands: new user, primary user, and intermediate user, and a very advanced user. We let this user work at a different working time. Maybe for the new user and the primary user, we must supervise by our technician. But for the intermediate user and advanced user, they can use all the equipment by themselves. Yeah. So for this purpose, we have a lot of training course and also a lot of workshop. We have big workshop for the background, introduction principle of all the equipment. We have also some very tiny workshop when we 2 or 3 users per time. We can show them how to operate this equipment hand by hand. Yeah, next please. This is a typical case pulled by the protein center. We saw as results as we are not only just provide the structure biologists, but also some immunity research in Tsinghua University. Next, please. So this is a achievement supported by the protein center. Only in 2023, we support 251 research papers, include 16 papers in the Nature, Science, and Cell. So next part, I'll introduce structure biology in the Tsinghua University. Actually, in the Tsinghua University, we have some very famous structure biology scientists in our university, something like such as Professor Shi Gong, our Wang Hongwei, Cui Senfang, Wang Xinquan, and Li Sai. The below is a very great achievement for this structure biology scientist, including the whole process of the splicing, especially in the COVID-19 period. We have solved the first crystal structure of the COVID-19 and the whole virus of the COVID-19. Next. So for the macromolecular structure biology, actually, we currently have 3 major methods for the structure research. One is cryo-EM, which is much more popular at this moment. We publish a lot of papers with cryo-EM, but for the crystallography, it's also very major technology to solve the structure. And for the NMR, also, we can use this method to check the dynamic conformation chain of the protein. So next. So the future for the structure biology should be combined with all this method to do this structure analysis. These 2 examples from the Function Yang's group and the ChIGS group. They all use different methods, something like a BioSAX, NMR, crystallographer, especially in the ChIGS group, which we published in the Nature 2021. You use a cryo-EM to solve the whole structure with a large complex and use crystallographer to get much more detail about the single protein. So this outstanding is both with our structure facilities, something like the splicing and sodium channel and the COVID-19, and also we developed our own method by our research center. Next, please. And also with this support, we support another, we have 2 commercial antibody drugs on the market. One is for the COVID-19, another one for the PD-1 antibody. So the last section, I mentioned some structure research biology of the vaccine. For the MR vaccine, they need, normally they express the component of protein, but problem is coming that if this protein is fully functional, if there's any mutation or the conformation is functional. So this one, we have a lot of methods to make sure this protein we expressed from the mRNA vaccine is fully functional. But the structural biology will give us a visible result. It's very strong evidence to suppose that the mRNA vaccine expresses a fully functional protein. So actually, there has two cases in my platform. These two users coming from the industry. Because confidential, I didn't mention, I cannot tell which company, but it's a real case. First one is RSV vaccine is from MR to the vaccine. So for the MR, the whole process, normally we got the MR vaccine from the company and then this transfection, the vaccine into the mammalian cell, and then it will express the protein. The single peptide will lead this RSV protein, the F protein, into the surface of the cell, and it will form tetramer on the surface. That means it's fully functional, but if it really form the tetramer, so the structure biology will give you this result. So when we get this case, we check the PDB library. We find there's two kinds of structure in the PDB. One is crystallographer, one is the protein. So we take the both. One is we purify this protein and for the crystal screening, and at the same time we use the protein to check which way is the quickest. Please next. So for the negative stain, we will find that the protein we purify is very good. This single particle is very clear on the negative stain. After the check, we found that this protein, the purity, the homology is very good. So we use protein to collect data on the cell structure. Please next. In this case, it shows that if the sample is good, the cryo-EM is much more quick than the crystallography, even with dollars of crystal. But for the cryo, it's very quick. We only use 3 days to collect data and no more than 1 week. We first solve the whole structure of the RSV. This result shows that this product is very good. We superimpose this structure of our protein with structure in the PDB library. The RMSD is below to the 0.5. So this is the case for the RSV MR vaccine. Please next. Another case, next. Another case is mRNA vaccine for the COVID-19. They use this mRNA vaccine to express the spike protein. So the same technical approach work. We do it at both times. One is for crystallographer, one is for the cryo-EM. But next. And at this moment, we do really got the crystal, and also we got the diffraction pattern from this crystal. But the difference is not that we need to optimize and collect more data to solve the structure. But for the cryo-EM, we check your negative spin, we find out the single particle is also very good. So we show the 2D classified to show that the spike number is really from the tetramer. And then they use this result to get on the market, whatever. Yeah. So that's it. So maybe we can some opportunity to cover this. Thank you.
Thank you, Dr. Silo. Our next speaker is Dr. Deng Haiteng. Dr. Deng Haiteng is a professor from Tsinghua University. He got his PhD at the Pennsylvania State University, and he was the director of mass spectrometry facility at Albert Einstein University, and also at the University of Nebraska and Rockefeller University. And his research interests focus on development and application of proteomic, metabolomic, and chemical biology technique, understanding the function and mechanism of redox biology in aging and neurodegeneration disease, and development of novel therapeutic intervention and diagnostic biomarker for aging-associated disease. And over the 5 years, this is very remarkable, he has published 135 papers in peer-reviewed journal. The time is yours, Professor. 15 minutes only.
Thank you. Thank you very much for the introduction. It's a great pleasure to give a brief introduction about our proteomics facility. We are part of the Protein Biotechnology Center. Next slide. Yeah, the purpose to establish facility, especially the core facility, is due to the instrumentation sometimes is very expensive for specific lab, and also need a big space for accommodation those equipment. Also need specific skills to maintain all the operation of the facility or the instrument. So this is why we need to establish those mass spectrometry or structural biology facility for all the faculties in the biomedical research. Next slide, please. Yeah, of course, the competition of the genome at the 204. So right now we are in the era of the functional genomics. So proteomics and other kind of omics technology has become the popular approach to studying the biomedicine, especially to establish the bridge between the genotype and the phenotype. Next slide, please. So to sequence the protein actually is quite difficult. The first protein to be sequenced is insulin. It was sequenced by Fred Sanger. It's taken 10 years to sequence this protein. It's very small protein. It's only 53 residues. For this achievement, he got Nobel Prize in 1958. So it's very difficult to sequence the proteins. Next slide, please. But mass spectrometry actually is the ideal tool for sequencing the protein. You can sequence the insulin, for example, B chain, within a couple seconds. You can sequence them, and also you can measure the molecule weight of the insulin. And of course, the mass spectrometry has become the major tool for many applications, including the food chemistry, food industry, and also environmental monitoring. Next slide, please. Of course, the development of mass spectrometry has been almost 120 years. It has a big impact on all the science development, including nuclear physics and also chemistry, material science. The application of mass spectrometry to the protein sequence actually is made possible by a bigger breakthrough by Jiang Fen. It's the introduction of the electrospray ionization. Next slide, please. Yeah, the mass spec is quite simple. So you can see that the mass spec has 4 major parts. One is the ionization. The second part is the mass analyzer to separate the mass according to mass to charge ratio. And also you have the detector and the data system. Jiang Fen's work actually is published 40 years ago. So this year actually marked his big discovery on the electrospray. So you can see the electrospray is a very simple device. It's a metal capillary. So if you apply the high voltage to the metal capillary, you can transfer sample ions, for example, protein, from the solution phase to the gas phase, and to be sequenced and detected by the mass spectrometry. Next slide, please. Currently, we have 7 types of different, mass analyzer. 2 of them belong to the Alexander Markov, a Russian original Russian scientist. I guess for the probably near future, he will got a Nobel Prize for this discovery. He single-handedly developed this mass spectrometer called Orbitrap. So right now, Orbitrap also become the major working force for the protein sequence. Next slide, please. He also developed another kind of mass analyzer called Astro. Was actually put to the market, last year.
So right now we have those all kind of hybridized, mass spectrometers for protein sequence. Of course, the high end of the mass spectrometer is quite expensive. One equipment it costs like $2 million US. So it's a big investment for the protein, sequence analysis. Of course, we have also timsTOF mass analyzer from, broker company. So right now, about 3 or 4 companies occupied all the mass spectrometry, market. Next slide. Yeah, of course, with all those kind of mass analyzer, mass spectrometers, right now we can do pretty high throughput proteomics analysis. Within 10 minutes, you can sequence like 10,000 proteins from cell, from tissue, or from organ. Then you can do very high accuracy of quantitation. Also, sensitivity is pretty good. For example, you can identify 5,000 or more proteins from a single cell. Each single cell you only have 100 picogram of proteins. So that the sensitivity and the throughput is pretty high right now for the proteomics approach. Next slide, please. Of course, this is only the beginning to understanding the genome functional genes. The reason is because we only have less than 20,000 of coding genes. But for the proteins, we have more protein forms because the protein was after transcription, translation. Then you have splicing, you have post-translation and modification. So we thinking for each cell, you have up to 1 million different protein forms. Of course, for human being, we have 36 trillion cells. So we estimate we have about 50 million of different protein forms. So this is just the beginning to understanding the genome functions by using the proteomics. Next slide, please. Of course, just like cell, actually each cell is quite unique. So each protein is also quite unique. For example, for the blood cell, the lifetime is about 120 days. So each blood cell, actually, they have specific characters, so they are unique. Protein is the same. For example, the milk protein, the lifetime is 30 minutes in the cell. So they have different modification. So each protein is also alive and also have different protein form, actually different PTMs. Next slide, please. Of course, right now we are studying, for example, the life cycle of proteins, including proteins in the blood. For example, for the albumin, we know that's the most abundant protein in serum. For the albumin, their lifetime was controlled by those modifications, for example, oxidation, the C-terminal trimming, and also glycation. You have sugar addition to the albumin. So right now we are in the edge to start functions of the gene product like those albumin, those milk, those kind of proteins. Next slide, please. Of course, one of the main applications of the proteomics is to identify some biomarkers. For example, we can use only 1 drop of blood. We can predict this person's biological age. If your biological age measured by this 1 drop of blood is larger than your chronic age, that means you're aging faster. In that case, you really need to do some intervention to slow down the aging. Of course, within this 20 proteins list, you can predict the biological aging. Some of them is very, is very famous. For example, GDF15. Lin Qi has been talking about how to make antibody against this GDF15. So in the future, we may have some antibody to kill, to at least slow down the aging process. Next slide, please.
Of course, the proteomics has been used to studying the pathogen host interactions. Of course, all kind of questions we can answer from the pathogen host interaction. Next slide, please. For example, if you have a virus infection into the cell, within a couple of days, we can do a quantitative proteomics. So we can get all the changes of protein. So this is very classically to study HCMV infection in the fibroblast. So you can identify not only the host protein and also all the virus protein presented in the surface. Those are potential, antigens to make antibody or make the, vaccine. Next slide, please. Also, you can understanding the mechanism about the pathogen host interactions. Next slide, please. Of course, you can also identify, for example, you have a virus, you want to know virus will, which receptor enter the cell. So this is one of the famous example by a Chinese scientist, Wen Li. He identified actually HBV, the receptor for HBV enter the host. Slide, please. Also, we can do using the mass spectrometry to characterize antibodies, including quality control, or we can find out all the, for example, all the variable region of the antibody, including all the modification, for example, glycosylation, and also we can identify what factors influence this life cycle of the antibody in the blood or in the organ. Next slide, please. Also, we can identify some, natural product, the target of the natural product. From this identification, actually, you can identify, you can establish a new target for the therapy. For example, we know the senescence cell is the major contributing factor for the aging-related disease, including tumor, including some neurodegeneration disease. So if you can selectively clearance those senescence cells, you can slow down the aging. So James Kirkland found out quercetin, a flavonoid, can selectively eliminate all the senescence cell. So we use so-called the thermal proteomics. Slide, please. We can identify the quercetin, the target for the quercetin, for example, this LDH1B1, 1I2, the targets for those quercetin. Next slide, please. Also, we can identify other kind of flavonoids. We found out all the flavonoids has some kind of tendency to slow down the aging by selectively eliminate, senescence cells. So one of the compound we identify is called DMC. Those DMC can eliminate senescence cell and also slow down the aging, at least in the animal models. I think next slide, please. Of course, using those plant extract is actually you don't have very targeted clearance of senescence cell. If you want to identify those specific targets for clearance of senescence cell, then you can use the so-called surface proteomics. You can identify, for example, in the senescence cells, you have B2M, you have UPIR, you have DPP4, those kind of proteins highly expressed on the surface of senescence cell. You can use CAR T cells, or you can use ADCs to target those kind of targets then eliminate those senescence cells. Next slide, please. Of course, from those identification, you also can identify some kind of cells may be very harmful to the body, to the, contribute to the aging process. In that case, it's, you can develop some, anti-aging vaccines. Those anti-aging vaccines actually is the newest addition to the vaccine list. For example, you can use GPNMB as the target to eliminate GPNMB express the cell, then you can slow down the aging. Next slide, please. Also, for example, you can use CD153 to eliminate all the senescence T cells, or using ILT to eliminate all the ILT PT3 or express cell, then decrease the lipid in the blood. So those all methods actually is the newest addition to the vaccine list. Next slide, please. Of course, at the Tsinghua, we have a proteomics center. The proteomics center, the major task is mainly to help all the faculties like Lin Qi, like Xinquan, to do their research. We can do all kinds of from protein identification to thermal protein profiling to antibody sequence. And also we provide training for the new staff member in different institutions. Next slide, please. Of course, maintain a facility actually is quite difficult. We need the Tsinghua University actually invest a significant amount of money to maintain all the staff member salary and also the service contract for the instrumentation. I guess if you have any question, you can ask Dr. Fan or Xinquan, because they are actually to make all the policies about which center, which facility they are supposed to invest more money. Okay, my time is up. Next slide. Yeah, thank you very much.
Thank you, Professor Haiter. Anti-aging vaccine, very interested. Yeah, I'm very interested. Okay, ladies and gentlemen, we have 20 minutes for a discussion. So I will take a few questions. Any questions from the audience? Oh. Okay, I will take a question from Zoom. Yeah, this is a question from Ibu Weni from the Ministry of Health Office. I think staff of Pak Roy Himawan. To Dr. Van Shilong. As you previously explained, the Protein Center is the only life science center among China universities. My question is whether this center has any certification like as ISO or GMP standard. Thank you.
Actually, we don't have any certification for that because if you actually for the qualification, it doesn't matter. We fully fit for the certification. But if we apply for the GMP certification, this equipment is just working for that function. We cannot provide our service to the whole scientific research. So we don't have a lot of research, just for that.
Of course, so that is possible to operate activities in your institution or in China in general without the laboratory having certification. Is that correct?
Yes, out of our research center, there's a lot of commercial CRO company or something like that. They have certification for the GMP. But for us, our purpose is to provide service for the scientific research. And if we have more internal content working for the industry, for GMP normally is for the industry. But for the scientific research, we do some very primary and innovation research, not guarantees. Keep this in.
So GMP is actually only for the
only for the
industry company,
yeah,
but not for the basic science laboratory. Okay. So, Pak Roy, maybe we can learn here that GMP is only for the industrial company, but not for the basic science laboratory. Is there also apply in Indonesia? Can someone deliver microphone to Pak Roy, please?
Yeah, thank you, Prof. Lina, for the question. And we do learn from China how to manage the facility, Prof. But what do we have here that we need to ensure the product that will go to clinical trial will have to meet the standard of safety and quality. That's why the authority in Indonesia put the GMP as a level that should be fulfilled by the lab. But I will be happy if colleagues from Indonesian FDA that attend also here today, Prof. They also attend here today, may also be able to explain more. But to what we see now that if we want to make the product move to the clinical trial, we have to make sure the product has to meet the safety and quality level. So ensuring that product, while they have no registration approval for a while, they need to ensure the facility that made of the product should meet some criteria that we use as GMP. But I would like to be happy to invite colleague from Indonesian FDA, Pak Dio atau
Dio
colleague.
from Bipom.
Yeah, please Pak Dio.
Okay, please, Pak Wildan.
Okay, thank you for the question about regarding in GMP implementation for clinical trial. I think in Indonesia we also regulate for clinical trial drugs. It's also the facility should be implemented of GMP standard. But for the certification is voluntary because for clinical trial facility, it can be like what we think that it should be implemented the GMP aspect, but for certification, you can submit to us, and also it's only applied for clinical trial phase 1 and phase 2, but for phase 3, it need a GMP certification. Okay, I think that's all that I can explain to you all. Thank you.
Thank you. So any other question? Yes, Pak Doni.
Thank you for the chance. For all the speaker, I would like to ask regarding the commercialization of your research into the commercial product. So maybe you can share, maybe one of you or two of you can share because all of you is complement each other from the animal and then from the instrument and also for the basic research of the product of the vaccine itself. So could you tell us about how your experience to develop the product, especially vaccine and biologic product from the scratch, from the research to be the commercialized product? Because for us in Indonesia, this is being the bottleneck in Indonesia. We already has one of the vaccine product that developed from the scratch in Indonesia on the time of the COVID era because of the condition. But I think you have more advanced experience to develop one commercialized product, and maybe you can tell to all of us about your experience and what the key to make the product can be commercialized, either from the research perspective and also for the business perspective, because you also have many connection with the business in China, and maybe you can just give us advice how to boost the condition like in China and bring it to Indonesia. Thank you.
I think the question is very clear. Like to answer. Okay, Professor Haida.
Okay, let me try to answer your question. It's a very important question for the basic research scientists like we are. We provide service, we provide some basic, for example, target for intervention. Actually, I guess we all have some connections with business partners, but we cannot do business ourselves, I guess. Yeah, so in that case, if we identify something, probably new target, or immediately probably I will go to Linqi. Yeah, because Linqi has more connections with the business uh, industry. And also we will test, for example, to do some preclinical studies. At least we can finish that. After preclinical studies, I guess the company will be, if any company will be interested, they will have a big investment on those kind of vaccine or drugs. Probably Linqi is better person to answer.
Donnie, this is a great question. First of all, Tsinghua University has a very clear and transparent rules of regulation of product development, from transferring from the laboratory to industry. So that has built a good foundation mechanism for this development. For the professors sitting in the audience, playing very key role in the very early period of innovation, because for the mRNA, for the animals, for the structure, for the proteomic testing, we say if you have a product you want to push into commercial development, you want to make sure all the technology available at Tsinghua give you a correct signal, a go signal. Without their characterization, we have no ability to judge whether our product, protein, mRNA, vector, are good enough for commercial development. So although we haven't got, or we don't need at a GMP level of qualification, but their judgment is going to give us a confidence to go or not to go. So I think in that context, we can collaborate with you, with the BRAIN, and with the other institution here to streamline the early period of qualification, quality control. So when you move into the commercial field, Professor Li sitting here from the China vaccine release, she has a system of commercial related rules and regulations. So we'll hear, she is our advisor. We have been working together a long time. So in the commercial development, we will follow the China's rules, regulation, how we could use that to push into human trials and then into the market. So you could see the connection between the professor here and the transparent mechanism, as well as the China regulatory authority, is the key thing for us to develop this or streamline the development of the product. I'm not sure whether I'm clear or not. That's why this communication is so important, because vaccine development is a system. It is a system project. It's not just like a one fragment. That's why we need to connect dots together. So Tsinghua certainly innovation and the preclinical, and then the Chinese regulatory authority is more of a regulatory authority to make sure your product is safe and efficacious. And for the, I know what the Don is trying to get at is to how we could transplant this system into here, into the vaccine evaluation the platform. So with this capacity, we could do it from the preclinical, clinical, and then the whole system to work with you and with the Ministry of Health to do it here. Okay, Danny? Okay, good. 李老师，你想说两句吗？不要。
Okay, okay, please.
Thank you so much. My name is Indra Diansha. I'm from Biopharma, and I have a question for Dr. Yu and Dr. Chang, Dr. Fan. First of all, I would like to say congratulations to Dr. Yu. It's very impressive data. And as we know that mRNA vaccine has a lot of, is protected by IP to certain sub company, the one that currently are available. So I would like to understand more about the lipid that you are using. Is it public domain or including the formulation as well? And the second one is, I would like to also understand about the safety of this THU lipid, because we know that there is some drawback on the current available mRNA vaccines where the PEG components can induce some certain adverse event. So do you have any data to show that this lipid is safer or better compared to the current existing one? And the third one is, I wonder also about the stability of your formulations, whether do you already have some data regarding of that? And for the Dr. Fan, do you have any plan for to bring your mRNA RSV vaccines for the clinical developments? And also, I might miss about the information whether these mRNA vaccines is expressed as the natural perfusions structures or because this is mRNA vaccine, we don't really care about the structure itself. Thank you so much.
Okay, Dr. Yu, would you like to answer the third question?
Okay, you mean the safety of our lipid nanoparticle, right?
Yes.
Okay, we use the building block is very safe, and we use chemical modification, and we conjugate the building blocks together. So the safety is very good. And we also test on the large animal models, such as the monkey, and we already verified the safety.
Do you have any plan to bring this formulation into clinical development?
For the pancreatic cancer the study, we plan to initiate an IIT study this year or next year on the safety and efficacy on human patient. Thank you.
Sorry, Dr. Yu, so you are going to do a clinical trial. Okay. Is it possible for Indonesian researcher here to be involved? Okay, thank you. So you listen, everyone, that the Indonesian researcher can involve in this clinical trial later, next year or next year. So, okay.
Sorry, I think I have one more question for Dr. Yu that I haven't follow up. Regarding the IP, is this formulation going to be public domain or specific to for single?
We have filed the patent already. Yeah.
Dr.
Yeah, I answer something about the structure of this license created. And actually, if you try to get this license approved on the market, you don't need to provide the structure to the FDA or CFDA or whatever. But when you try to purify this protein from the mRNA license, you will find it's quite different from different companies. I just show you the best result from one of them. Actually, I collect several data from different companies from different mRNA license for the same protein, something like RS3. And when you purify the protein, try to get the structure, you'll find the hormone is quite different from different ones. So from this, we can provide some very strong evidence.
Can you make your microphone
Sorry,
closer?
sorry. Just during this process, you can give a response to this industry company, tell them that vaccine is not that good or good enough to apply the certification from the FDA or CFDA, whatever. Yeah. So, and also the structure can show that it really form very stable tetramer of RSA or F protein or the spike protein. It's just like approved for the quality. Yeah, it's not necessary to apply for the FDA, but it's really very strong evidence to show them this protein is stable.
Okay. I think I want to
Good afternoon. So my name is Robbie. I'm from Etana. So what I would like to ask is about to continue the previous question regarding the connecting the dots for the research. How do you do the transfer of the projects between one department to another department inside the Tsinghua University? Because we already talked about transferring that outside to the industry, but I haven't seen like from the preclinical and inside the ecosystem of the Tsinghua, since I think it's a lot of department inside.
Yeah, anyone can answer that internal networking?
Yeah, so of course, our facilities right now, we are in the same building. So this is depends on the research, on the professor, on PI, if they have intention to use different facility to do the characterization. So we will provide this characterization. So actually, I guess the answer is depends on, the PI, his intention to, for example, if they can develop some new product.
In the practice, Professor, before you do any research, do you have a plan funds for collaboration between your internal department and have the algorithm being established? It's not like if you are interested, but from the beginning you already plan it.
Oh, okay. Okay, that is very good questions. Yeah, of course, for the basic research, we try to answer some scientific questions. From those scientific questions, the PI already have a plan, if this one discovery can be eventually, put into the practice. So I guess, this is the circulation of the information will be, quite often between those faculties. For example, Xinquan and Vinci, they have a lot of collaboration. They were talking about their project. So we have internal this kind of meetings to discuss the development. Of course, we always have a leader, who will determine if this project will go on.
A project leader.
Project leader, yes, yeah. So in China, we have a lot of, this project already, grant. So we have a leader, basically this leader will determine, which product, which invention eventually will become.
So when you mention about grant,
yes,
does Tsinghua support the project itself or getting grant from the government or other donors?
Yes, we have different ways to, have this kind of, grant. Of course, the major part is from the government. Tsinghua probably have the highest, budget from the government. Also, the Tsinghua, the different schools, like my school, life science, they got donations from bigger companies. Some of them is for the basic research, some of them is for development, what kind of drug they are interested in. So we have a lot of collaboration with these big pharma companies. Yes.
Okay, majority of the budget come from the government.
Yes.
Okay, Pak Roy, majority of the budget come from the government.
Yes.
And second from pharma company.
From pharma.
Donnie, pharma company. Okay. And they support their research institution.
Yes.
Like BRIN. Okay.
Yeah.
BRIN asked Pak Roy and Donnie. Okay. Yes, please.
The topic of research of the idea of research between commercialized product and the research interest of researcher sometimes is different. Yeah. So how, in experience of the Chinese researcher, to bridging with the interest with the commercialized interest and with your idea of research? Sometimes, yeah, for example, governments make the roadmap like that, and pharmaceutical company, they are looking of the, for example, benefits or the commercialized product. But in our idea or on our interest is different. Sometimes we have the ideology working with the new technology, working with the new idea, new platform, how to bridging with that with the industry. Just a question like that. Thank you.
It's a very difficult question to answer, but I will try. Okay, I guess the government, especially Chinese government right now, investment big funds for development newest technology. For example, for computation, for example, like brain-like computation, quantum computation. So Chinese government is more interested in this kind of areas. But just like you mentioned, pharmaceutical company, they will have immediately interest, income from those kind of things. So I guess for research like me, I guess it's money driven. So whatever I can get money from government, I will do the government project, or from company, we will do the company project. Of course, the biggest task or responsibility for us actually is to answer some scientific questions. So we are scientific question driven. So this is different from government. They want to form organized research, and also from company, they are interested driven research. Of course, Linky, please help me out.
I think being a professor and researcher, our main theme is to education and research on campus. But as Professor Deng said, all of professors sitting here, I think many of from Indonesia have to be multi-talented. You cannot focus just on one thing, because the expectation from the university and from the government are way beyond single job. So all the professors here, while they're focusing on teaching and research, but they also have obligation to serve their own career development into collaboration with the industry, collaboration with international organizations like this, and then to promote the networking and to promote the knowledge and technology. So in that context, I think we'll all be motivated or somewhat pressured to diverse or to take full advantage of our skills, knowledge, and collaborative network. So in that context, I am sure that's why they're here, right? So we were, during this process, we'll expand and take full advantage of what we have learned, what Tsinghua has and what Indonesia has. And that's a big job. I mean, but that's a very rewarding, challenging job. Is that right? I'm not sure, Ilana, you are a clinician, but you have been driven into discussion, vaccine, diagnosis, treatment. I think you would agree with me. Right?
Yes,
Okay.
100% agree. So Professor modified here, compromise, collaborate with many stakeholders, and be nice to the regulatory.
That's right. That's right. Thank you. Thank you.
So, any other? Okay, yeah, 2 more. So we have 20 questions.
Okay, thank you very much. My name is Christian from the Etana Biotechnology. So I would like to ask to Dr. Chang Zai. So actually, it's very amazing that you've been able to manage that huge of the animal facility. And then I see there is a huge increasing of the usage of the animal facility, which is also established, started from 2007 until 2022. That's already facing a lot of how to say, a lot of faith. Right? So my question is, in order to manage the increasing usage of the animal itself and also the numerous of the variants of the type of the animal itself. So what is the and how is the contribution of the governments and also internal of the researchers, how you manage and then the contribution of the government in order to, how to say, to push the increasing of the mice as yearly started from 2007 until 2002 that you've been able to manage a huge of the variants of the mice itself. Thank you.
Okay, this is the last question, sir. So please answer.
Prof. Elina, I'm sorry, excuse me. Before Dr. Chang answer that question, I have I think it's a little bit similar with the previous question regarding the animals. So if it's possible, I can ask the question to Dr. Chang as well. Yeah, I'm Dio from Indonesian FDA. One of the big challenges that we faced for the evaluation of the vaccine or biological product is the animal testing. So we have to maintain the animal facilities that is quite expensive as well. And right now, as we know that WHO, they release more guidelines regarding the 3R, reduce, replacement, and replace of the animal for testing. I would like to know, or maybe you can share your experience. Do you have like a program or a roadmap to implement the 3R regarding the animal testing? Because and what do you think about the reduce or replacement of the animal using in vitro instead of in vivo? Do you have any experience or do you have plan to do that in the near future, probably? Thank you.
So I take the question because my last words, be nice to the regulatory. Actually, the time is up, but okay. So, you ready? Yeah.
You talk about the 3Rs. Yeah. The 3R have in China we have 100 more regulations and the national standard for 3Rs and animal feed, animal use. So the FDA, WHO have reduced the cosmetics. So, but the drugs have a stricter use animal.
So can you answer about the roadmap I was asking before? Second comment.
So I'm sorry, just as he mentioned, for the drug development, we still need these animals. For domestic, maybe we don't need use the mice or something else. We can use cell, live cell, or something else. But for the drug development, in our China, there's still some institutes that are focused on finding some way to without animals to do some research about without mice, right? Yeah, but currently we still have a lot of users for drug discovery in city.
So maybe during the break session, we can directly discuss, yeah, because our time is up. And So a big applause to all the speakers. And just before go down, I would like to say one pantun in bahasa. Ya, Profesor Rinci dan Pak Tantowi di Bali ingin meneliti vaksin dari daun sirih. Sampai di sini, terima kasih.
What an amazing poem, Pantun, Prof. Thank you for leading this session amazingly. Also, please give applause, ladies and gentlemen, to all the speakers. This is going to be a very insightful session. And same as the previous plenary session, I would like to invite the speakers and moderator. You will please be upstanding in front of the table as we will continue with the taking picture with all of you, speakers and also moderator. Thank you so much. And I'm sure, ladies and gentlemen, you can continue your further discussion with the speakers later on during the lunch break session in a casual way. So after this, you will have a lunch break session and we will continue our plenary around 1 p.m. Bali time. So please take your time, everyone. All right? Prof. Chang joining the photo session. 1, 2, 3. Smile. Pak Tantowi, if you don't mind, could you please join Prof. Erlina for the photo session? Please remain on stage. This thing is on stage. I would like to invite to join the photo session Bapak Tantowi Yahya, the president of UID. Special request, Pak, from Prof. All right, please face the camera. Excellencies on stage. 1, 2, 3. Okay, one more from handphone. Okay, V for vaccine. All right, 1, 2, 3. Okay, good. Thank you so much. Please return to your seat on the floor. Speakers, moderator, Prof. Chang, Bapak Tantowi Yahya. Yes, we have concluded the second of plenary session. Thank you so much, speakers and moderator. As I mentioned previously, distinguished guests, ladies and gentlemen, now we will have lunch break session. For all participants who are joining us here on site at the Trans Resort Hotel Bari, the lunch already served at the ground floor restaurant in front of the lobby. So once again, you may please enjoy the lunch that we have provided at the restaurant, at the ground floor restaurant in front of the lobby. If you want to pray, we also provide you the praying room or musola, ladies and gentlemen. It is located at ground floor as well. You may please follow the signage that we already provided special to each and every one of you to make you easier to find the praying room or musola. And once again, can I remind you, we will start our next panel discussion at 1 p.m. Bali time. It's around 45 minutes from now. So please take your time and please be on time for the next hour session, plenary session. And also for online participants, thank you so much for joining us. Please don't go anywhere. I just let you to enjoy your lunch break and you will be invited again to our next plenary session at 1 p.m. Bali time. So ladies and gentlemen, enjoy your lunch. Selamat makan, bon appetit, and I'll be back soon for the next plenary session. Thank you. Have a good lunch, everyone.
Paling jendela di kamarku melihat seberkas cahaya hingga terjaga segera dari tidurku malam itu dan mimpi yang mesra. Ketika aku membuka jendela, sinar mentari menyambut pagi, memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Dari balik jendela di kamar menyelinap seberkas cahaya hingga ku terjaga segera dari tidurku malam itu dan mimpi yang mesra. Ketika aku membuka jendela, sinar mentari menyambut pagi, memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini dan sepanjang hari betapa bahagia. Salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Tanpa terasa siang akan menjelang, hari pun berganti, aku pun berlalu. Berikan salam, selamat pagi, betapa bahagia. Dari balik jendela di kamar menyelinap seberkas cahaya hingga terjaga segera dari tidurku malam itu dan mimpi yang mesra ketika aku membuka jendela sinar mentari menyambut pagi memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Dari balik jendela di kamar menyelinap seberkas cahaya hingga ku terjaga segera dari tidurku malam itu dan mimpi yang mesra ketika aku membuka jendela sinar mentari menyambut pagi memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini dan sepanjang hari betapa bahagia. Salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Tanpa terasa siang akan menjelang hari pun berganti aku pun berlalu memberikan salam selamat pagi betapa bahagia. Paling jendela di kamarku menyelinap seberkas cahaya hingga ku terjaga segera dari tidurku malam itu dan mimpi yang mesra ketika aku membuka jendela sinar mentari menyambut pagi memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Dari balik jendela di kamar menyelinap seberkas cahaya hingga ku terjaga segera dari tidurku malam itu dan mimpi yang mesra ketika aku membuka jendela sinar mentari menyambut pagi memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini dan sepanjang hari betapa bahagia. Salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Tanpa terasa siang akan menjelang hari pun berganti aku berlalu memberikan salam selamat pagi betapa bahagia. Dari balik jendela di kamar menyelinap seberkas cahaya hingga terjaga segera dari tidurku malam itu dan mimpi yang mesra ketika aku membuka jendela sinar mentari menyambut pagi memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Dari balik jendela di kamar menyelinap seberkas cahaya hingga terjaga segera dari tidurku malam itu dan mimpi yang mesra ketika aku membuka jendela sinar mentari menyambut pagi memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini dan sepanjang hari betapa bahagia. Salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia tanpa terasa siang akan menjelang hari pun berganti aku berlalu memberikan salam selamat pagi betapa bahagia. Dari balik jendela di kamarku menyelinap seberkas cahaya. Hingga ku terjaga segera dari tidurku malam itu dan mimpi yang mesra. Ketika aku membuka jendela, sinar mentari menyambut pagi, memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Dari balik jendela di kamar menyelinap seberkas cahaya. Hingga ku terjaga segera dari tidurku malam itu dan mimpi yang mesra. Ketika aku membuka jendela, sinar mentari menyambut pagi, memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini dan sepanjang hari betapa bahagia. Salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Tanpa terasa siang akan berjalan, hari pun berganti, aku pun berlalu. Memberikan salam, selamat pagi, betapa bahagia. Balik jendela di kamar menyelinap seberkas cahaya hingga terjaga segera dari tidurku malam itu dan mimpi yang mesra ketika aku membuka jendela sinar mentari menyambut pagi memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Dari balik jendela di kamar menyelinap seberkas cahaya hingga ku terjaga segera dari tidurku malam itu dan mimpi yang mesra ketika aku membuka jendela sinar mentari menyambut pagi memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini dan sepanjang hari betapa bahagia. Salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia tanpa terasa siang akan menjelang hari pun berganti aku pun berlalu burung-burung memberikan salam selamat pagi betapa bahagia. Dari balik jendela di kamarku menyelinap seberkas cahaya. Hingga ku terjaga segera dari tidurku malam itu dan mimpi yang mesra. Ketika aku membuka jendela, sinar mentari menyambut pagi, memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Dari balik jendela di kamarku menyelinap seberkas cahaya. Hingga ku terjaga segera dari tidurku malam itu dan mimpi yang mesra. Ketika aku membuka jendela, sinar mentari menyambut pagi, memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini dan sepanjang hari betapa bahagia. Salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Tanpa terasa siang akan menjelang, hari pun berganti, aku pun berlalu. Memberikan salam, selamat pagi, betapa bahagia. Balik jendela di kamar menyelinap sebekas cahaya hingga ku terjaga segera dari tidurku malam itu dan mimpi yang mesra ketika aku membuka jendela sinar mentari menyambut pagi memberikan salam dalam kesejukan dan indahnya pagi. Seandainya suasana pagi ini kan sepanjang hari betapa bahagia. Dari balik jendela di kamar menyelinap sebekas cahaya hingga ku terjaga segera.
Immediate steps are needed to scale up preventative, diagnostic, and treatment services for tuberculosis to break the chain of transmission and reduce the case of multi-drug resistant tuberculosis. Indonesia is also experiencing a rise in non-communicable diseases such as cancer, diabetes, and stroke. And together, these diseases put significant pressure on our health system. Every human has a unique DNA sequence, and those differences can influence our susceptibility to disease and our response to treatment. Understanding these will help us improve prevention, diagnosis, and treatment through precision medicine. Biomedical and Genome Science Initiative, BGSI, is Indonesia's first national initiative for precision medicine. With priorities towards 5 high-burden diseases and a focus on exploiting genomics to improve our well-being and help us through the aging process. BGSI will support the routine use of genomic information to improve public health using an integrated platform that brings together 3 services units: biobank, registry, and bioinformatics. A biobank is a collection of biological samples such as blood or DNA and health information that is stored in specialized facilities and kept in controlled conditions for future use. Our biobank will collect samples for cancer, stroke, diabetes, TB, and psoriasis patients, as well as from healthy volunteers. A registry is a structured data repository that curates information for individuals that are included in a specific population, including information about the disease, clinical information related to each patient, demographic information, and others where this information can be used for clinical or policy purpose. Our registry will connect this information with the samples stored in our biobank, thus ensuring that these samples can be used for further research and innovation. Imagine being able to use samples collected today for research in 10 years' time. BGSI's Bioinformatics Service Unit will focus on supporting the primary and secondary analysis of the whole genome sequencing data which would then be used to assess gene variation and expression, and other information generated from next-generation sequencing facilities spread throughout Indonesia. BGSI will work closely with 6 hubs, each targeting different diseases, with the support of 7 national hospitals. BGSI will coordinate the spread of 48 next-generation sequencers to Indonesia's research institutions to increase enthusiasm for biogenomic research. Indonesia has begun its journey towards the establishment of a national genomic sequence reference with a plan to collect 10,000 human genome sequences over the next 2 years, as a collaborative effort to support the complete mapping of disease-specific variants in our population. This project requires us to leapfrog several technological aspects, including next-generation sequencing, bioinformatics pipeline, methods for genotyping single nucleotide polymorphisms, and high-performance computing. Those technologies will discover precise medication for each individual based on their genetic profile to make the best therapeutic choice by facilitating predictions on benefits and/or potential side effects after taking a particular medicine. This can only be realized through inclusive participation and collaboration of various stakeholders. We require your support to ensure we can transform healthcare in Indonesia towards the use of precision diagnostics for more personalized treatment and prevention to improve public health. Indonesia ranks 3rd in tuberculosis burden in the world, with an estimated 824,000 cases and 93,000 deaths per year. Immediate steps are needed to scale up preventative, diagnostic, and treatment services for tuberculosis to break the chain of transmission and reduce the case of multi-drug resistant tuberculosis. Indonesia is also experiencing a rise in non-communicable diseases such as cancer, diabetes, and stroke. And together, these diseases put significant pressure on our health system. Every human has a unique DNA sequence, and those differences can influence our susceptibility to disease and our response to treatment. Understanding these will help us improve prevention, diagnosis, and treatment through precision medicine. Biomedical and Genome Science Initiative, BGSI, is Indonesia's first national initiative for precision medicine. With priorities towards 5 high-burden diseases and a focus on exploiting genomics to improve our well-being and help us through the aging process. BGSI will support the routine use of genomic information to improve public health using an integrated platform that brings together 3 services units: biobank, registry, and bioinformatics. A biobank is a collection of biological samples such as blood or DNA and health information that is stored in specialized facilities and kept in controlled conditions for future use. Our biobank will collect samples for cancer, stroke, diabetes.
Distinguished participants, ladies and gentlemen, as we would like to continue our next plenary session, I would like to invite for those of you who are still outside at the foyer area, you may please move your way and re-enter the ballroom and kindly have a seat as we would like to begin our event shortly. And also for the online participants, you may please back in front of your screen as we will continue the next plenary session shortly. Thank you.
Thus ensuring that these samples can be used for further research and innovation. Imagine being able to use samples collected today for research in 10 years' time. BGSI's Bioinformatics Service Unit will focus on supporting the primary and secondary analysis of the whole genome sequencing data, which would then be used to assess gene variation and expression, and other information generated from next-generation sequencing facilities spread throughout Indonesia. BGSI will work closely with 6 hubs, each targeting different diseases, with the support of 7 national hospitals. BGSI will coordinate the spread of 48 next-generation sequencers to Indonesia's research institutions to increase enthusiasm for biogenomic research. Indonesia has begun its journey towards the establishment of a national genomic sequence reference with a plan to collect 10,000 human genome sequences over the next 2 years, as a collaborative effort to support the complete mapping of disease-specific variants in our population. This project requires us to leapfrog several technological aspects, including next-generation sequencing, bioinformatics pipeline, methods for genotyping single nucleotide polymorphisms, and high-performance computing. Those technologies will discover precise medication for each individual based on their genetic profile to make the best therapeutic choice by facilitating predictions on benefits and/or potential side effects after taking a particular medicine. This can only be realized through inclusive participation and collaboration of various stakeholders. We require your support to ensure we can transform healthcare in Indonesia towards the use of precision diagnostics for more personalized treatment and prevention to improve public health. Indonesia ranks third in tuberculosis burden in the world, with an estimated 824,000 cases and 93,000 deaths per year. Immediate steps are needed to scale up preventative, diagnostic, and treatment services for tuberculosis to break the chain of transmission and reduce the case.
Vaksin Collaborating Center atau VOLARE. VOLARE bertujuan untuk menjamin keberlanjutan penguatan ekosistem vaksin yang diharapkan juga akan menjadi platform yang lebih koordinatif, kolaboratif, dan sistematis dalam skema triple helix dalam menjalin sinergi antar stakeholder di bidang vaksin di Indonesia. VOLARE bergerak dalam mengkolaborasikan aspek-aspek riset dan pengembangan manufaktur, penyediaan, harga dan pembiayaan, distribusi, logistik dan rantai supply, penerimaan dan serapan pasar di bidang vaksin.
Indonesia ranks third in tuberculosis burden in the world, with an estimated 824,000 cases and 93,000 deaths per year. Immediate steps are needed to scale up preventative, diagnostic, and treatment services for tuberculosis to break the chain of transmission and reduce the case of multi-drug resistant tuberculosis. Indonesia is also experiencing a rise in non-communicable diseases such as cancer, diabetes, and stroke. And together, these diseases put significant pressure on our health system. Every human has a unique DNA sequence, and those differences can influence our susceptibility to disease and our response to treatment. Understanding these will help us improve prevention, diagnosis, and treatment through precision medicine. Biomedical and Genome Science Initiative BGSI is Indonesia's first national initiative for precision medicine. With priorities towards 5 high-burden diseases and a focus on exploiting genomics to improve our well-being and help us through the aging process. BGSI will support the routine use of genomic information to improve public health using an integrated platform that brings together 3 services units: biobank, registry, and bioinformatics. A biobank is a collection of biological samples such as blood or DNA and health information that is stored in specialized facilities and kept in controlled conditions for future use. Our biobank will collect samples for cancer, stroke, diabetes, TB, and psoriasis patients, as well as from healthy volunteers. A registry is a structured data repository that curates information for individuals that are included in a specific population, including information about the disease, clinical information related to each patient, demographic information, and others where this information can be used for clinical or policy purpose. Our registry will connect this information with the samples stored in our biobank, thus ensuring that these samples can be used for further research and innovation. Imagine being able to use samples collected today for research in 10 years' time. BGSI's Bioinformatics Service Unit will focus on supporting the primary and secondary analysis of the whole genome sequencing data, which would then be used to assess gene variation and expression, and other information generated from next-generation sequencing facilities spread throughout Indonesia. BGSI will work closely with 6 hubs, each targeting different diseases, with the support of 7 national hospitals. BGSI will coordinate the spread of 48 next-generation sequencers to Indonesia's research institutions to increase enthusiasm for biogenomic research. Indonesia has begun its journey towards the establishment of a national genomic sequence reference with a plan to collect 10,000 human genome sequences over the next 2 years, as a collaborative effort to support the complete mapping of disease-specific variants in our population. This project requires us to leapfrog several technological aspects, including next-generation sequencing, bioinformatics pipeline, methods for genotyping single nucleotide polymorphisms, and high-performance computing. Those technologies will discover precise medication for each individual based on their genetic profile to make the best therapeutic choice by facilitating predictions on benefits and/or potential side effects after taking a particular medicine. This can only be realized through inclusive participation and collaboration of various stakeholders. We require your support to ensure we can transform healthcare in Indonesia towards the use of precision diagnostics for more personalized treatment and prevention to improve public health.
Vaksin Collaborating Center atau VOLARE. VOLARE bertujuan untuk menjamin keberlanjutan penguatan ekosistem vaksin yang diharapkan juga akan menjadi platform yang lebih koordinatif, kolaboratif, dan sistematis dalam skema triple helix dalam menjalin sinergi antar stakeholder di bidang vaksin di Indonesia. VOLARE bergerak dalam mengkolaborasikan aspek-aspek riset dan pengembangan manufaktur, penyediaan, harga dan pembiayaan, distribusi, logistik dan rantai supply, penerimaan dan serapan pasar di bidang vaksin.
Indonesia ranks 3rd in tuberculosis burden in the world, with an estimated 824,000 cases and 93,000 deaths per year. Immediate steps are needed to scale up preventative, diagnostic, and treatment services for tuberculosis to break the chain of transmission and reduce the case of multi-drug resistant tuberculosis. Indonesia is also experiencing a rise in non-communicable diseases such as cancer, diabetes, and stroke. And together, these diseases put significant pressure on our health system. Every human has a unique DNA sequence, and those differences can influence our susceptibility to disease and our response to treatment. Understanding these will help us improve prevention, diagnosis, and treatment through precision medicine. Biomedical and Genome Science Initiative BGSI is Indonesia's first national initiative for precision medicine. With priorities towards 5 high-burden diseases and a focus on exploiting genomics to improve our well-being and help us through the aging process. BGSI will support the routine use of genomic information to improve public health using an integrated platform that brings together 3 services units: biobank, registry, and bioinformatics. A biobank is a collection of biological samples such as blood or DNA and health information that is stored in specialized facilities and kept in controlled conditions for future use. Our biobank will collect samples for cancer, stroke, diabetes, TB, and psoriasis patients, as well as from healthy volunteers. A registry is a structured data repository that curates information for individuals that are included in a specific population, including information about the disease, clinical information related to each patient, demographic information, and others where this information can be used for clinical or policy purpose. Our registry will connect this information with the samples stored in our biobank, thus ensuring that these samples can be used for further research and innovation. Imagine being able to use samples collected today for research in 10 years' time. BGSI's Bioinformatics Service Unit will focus on supporting the primary and secondary analysis of the whole genome sequencing data, which would then be used to assess gene variation and expression, and other information generated from next-generation sequencing facilities spread throughout Indonesia. BGSI will work closely with 6 hubs, each targeting different diseases, with the support of 7 national hospitals. BGSI will coordinate the spread of 48 next-generation sequencers to Indonesia's research institutions to increase enthusiasm for biogenomic research. Indonesia has begun its journey towards the establishment of a national genomic sequence reference with a plan to collect 10,000 human genome sequences over the next 2 years, as a collaborative effort to support the complete mapping of disease-specific variants in our population. This project requires us to leapfrog several technological aspects, including next-generation sequencing, bioinformatics pipeline, methods for genotyping single nucleotide polymorphism.
Welcome back, distinguished guests, ladies and gentlemen, my beloved offline and also online participants. Thank you for your presence. Thank you for staying with us. And before we continue to our next plenary session, I have an important message to each and every one of you. Distinguished guests, ladies and gentlemen, after we conclude the entire agendas of the symposium, we will move to our next segment of our program. It is inauguration of Indonesia-China Joint Research and Development Center on Vaccine and Genomics that will be located at UID Tsinghua, Southeast Asia. So after the whole agendas, after we conclude the symposium, ladies and gentlemen, there will be another event that will be held at the UID Tsinghua, Southeast Asia. For further details information, I will inform you later at the closing segment of the symposium. Now, let us continue our next plenary session. Are you ready? I cannot hear your voice. I know after the lunchtime, there'll always be a critical time. So let's bring back our energy by having big round of applause before we start the event. Again, louder, please. Thank you so much, ladies and gentlemen. Now we will enter the third plenary session on the theme of genomic medicine in disease prevention and management, current trends and future directions. And to guide this session, I would like to invite our distinguished moderator, senior researcher at Research Center for Genomic Engineering, National Research and Innovation Agency. Please welcome Dr. Dani Permana, SSI, MSI to the stage. Please give applause to our moderator, Dr. Dani Permana. Yes, Dr. Dani, I will leave the stage to you. Stage is yours.
Okay, thank you very much for the MC. And welcome back. And good afternoon, everyone. Distinguished ladies and gentlemen. And in this occasion, I would like to start the third plenary forum. And I will invite one by one of our speakers. And as you can see here, we have 6 chairs. So that's mean we have 6 speakers. So this will be a very long journey for you and also for me. So yes, let's start. Okay, the first speaker is, I would like to invite Indri Roslamiati, MSc APT. please come to the stage. And the second is Uni Rukminiati M. Biomed. Please welcome. The third speaker will be Dr. Luh Inta Prilandari. Please come to the stage. The next is Professor Dr. Wang Xingquan. The fifth is Professor Dr. Ding Xiang. And the last but not the least, Dr. Liu Xin. Please come to the stage. Okay, because we started this section a little bit late, so I will just start this section. So first speaker will be Indri, and I will just briefly read her CV. Currently, she is a PhD candidate at the University Medical Center Utrecht University in research focus in infection prevention and control and antimicrobial resistance. And currently, she is acting director of the National Biomedical and Genome Science Institute, Directorate of Health Science, Ministry of Health, Republic of Indonesia. And if you are ready, you can start. You have 15 minutes. Present your presentation.
So thank you for the moderator. I think I will just go down a bit. Oh, okay, maybe here. It's okay. Okay, so good day, everyone. So today I would like to share regarding our initiative called the Biomedical Genome Science Initiatives. So it was initiative 2 years ago, but now it's becoming a unit under the Ministry of Health. So slide. So as we know, the Indonesia has quite a big population, right? It's around 280 millions. And most of the data for the development of the medical devices or development of the drugs is based on Caucasian populations. Then we're thinking like, why don't we have our own database so that all the medications, like the medical devices and everything that we will develop will be based on our data, and it will become more precise. And also we can give the better quality for the services for the patients and for the community. So therefore, this is like, what do we need? So the first step will be that we need the set data genome populations that based on Indonesians. And the result will be the precision of medicine. So the precision medicine is that one of the approach that can make the patients have the treatment a bit better. So we can adjust the medications to the patient based on their genetics. So that is one of the purpose. And then also we can also reduce the time of the treatment, because that if the medicine that we are having now is that one diseases can be given one drugs with the same doses for everybody, right? But if you have the precision medicine, and then you can give it to personal person, and also the result will become better. So slide. And this is what we're having at the moment. So now the drugs that we have is one fits all. But for the precision medicine, it will be based on individual. So therefore, thank you. Therefore, we will reduce the side effect and improve the outcome. So the patient, hopefully, they will get the drugs that suitable to their genetics. Size. So this is the example. So you know that there's one drug called clopidogrel. So this is the drug for stroke, which is now becoming the second drug that is purchased by the Indonesian government. But however, there was this publication saying that the Asian populations, they are quite resistant with the clopidogrel. It's around 28%. So therefore, we would like to find out, how about the Indonesian population? So what we're doing at the moment is that we would like to see how the enzyme for these medications called C2C19 in Indonesian population, do they have like poor metabolism, or do they have like normal metabolism, or the rapid metabolism? What it will affect? It will affect the dose. For example, if most of Indonesian, they will have the poor metabolism. So therefore, it's not effective anymore. So this can be one of the study that we can give to the government to take a look on their treatment, if they can change the drugs, the priority of the drugs. Slide. Yeah, but for that, I'm not gonna say we have the barriers, but we have also the challenges, right? So the challenges gonna be, the first is that I said before about the availability of the data genomics for Indonesian populations. So therefore, we are doing this. And the second one is more about the knowledge and skill gap. So I think this one can be achieved or can be solved by the collaboration that we are having at the moment. So I heard that Tsinghua has some skills that we need in bioinformatics, in analyzing the data. So I think this forum is going to be very beneficial for us. And also the infrastructure. So for the infrastructures, I think Indonesia, now we are quite, I'm not going to say advanced, but I think you said you already saw in the video that we already have 64 genomic sequencing machines all over Indonesia. So that can be a start. And then the awareness gap. I think this is the homework all for us. So we need to engage the community. We need to educate the clinicians. We need to have like a proper understanding about the genetic data, which is provided by the result from the sequencing. Because like in the end of the day, I think the clinicians will be the one that using this data. So we need to inform them well. Slide. Okay, so what the government's doing? So what is the Ministry of Health do? So last year, we launched the Biomedical Genome Science Initiative to cover all those problems that I already said. And also, we are establishing the Clinical Research Center. So I heard that in the morning, there were several questions regarding the clinical trials. So the Clinical Research Center will be the one-stop center for all the clinical trials in Indonesia. Slide. And these are the starting of the program. So we have some catastrophic diseases that still become a high burden in Indonesia. So we have tuberculosis, we still have stroke, cardiovascular. So from these challenges, so therefore, we have, second slide, we have this. So to how to solve those problems is with the BGSI. And what we have is not only the data, but we have the series of the infrastructures and also the system that can support everything. So the first one, we have the biobank, and then we also have the registry of the demographic data and the data for the participants. And then we also have the sequencing services and then the bioinformatics capacity. And slide. And this is the roadmap that we have. So this year is the year of the proof of concept. We started 2022. So hopefully in the end of this year, we can get 10,000 samples. And then in 2029, hopefully we can get 100,000 samples. So this is the roadmap. And not also the collecting of the samples and the data, but we also, I think, the main key is to develop to hospitals so that they can develop the services based on genomics that can give the patients the treatment based on the precision medicine. Slide. And these are the hubs that we have. So BGSI, we are working together with the 10 practical hospitals. So we have the infectious diseases focuses, that we have 2 hospitals, Persahabatan and then Sulianti Saroso, and then we have cancer, Dharmais, we have diabetes, in RSCM, rare disease in Sarjito, and then maternal and baby in Harapan Kita Hospital, cardiovascular in Harapan Kita Hospital for heart, and then for the stroke in Rumah Sakit PON, anti-aging nutrition and wellness in Murah Hospital. Dr. Inta, I think you will talk about this a bit. And then the last hubs that joining is the mental health in the Marzuki Madi. So these are our network. Slide. And as I said before that we are BGSI and we have hospitals, right? So the hospitals, they are doing the targeted sequencing. And in central BGSI, we're doing the whole genome sequencing. So these are the capacity that we have or what we are doing at the moment. So in hubs, they are doing the targeted sequencing, which is based on their center of excellence. So I think based on those focus of diseases. For example, like in Dharmais, so you're focused on a cancer. So you have certain panels for the screening of the cancer. And also, for example, in RCM, they are now developing the panel for detecting the MODI, those kind of things. And what we're doing in BGSI, we're doing the whole genome sequencings, and also we are coordinating all the hubs. Slide. And these are the detail of the recruitment that we are hoping to be done in 2024. So it consists of the target of these diseases based on the hospitals. Slide. And we are not only developing all the services, all the data, but I think the most important thing is the ecosystem, is the health ecosystem. So we can see here, they are all linked. So all the biobank, all the data of the participants for the demographic data, the clinical data, and also the genomics data is all linked, but they are in the different platforms. Slide. And in addition to the building the ecosystem, but we are also trying to not enhancing, but more to give a trigger to ecosystem to develop the new medical devices, research development on drugs that based on our data. So therefore, we will have like a data sharing platform. So here we can see that on the upside is the central BGSI, which is in Bebe Binomica. And then we have all the hubs there, which is the hubs. They also have satellite and spokes to collect the samples. So that we send the samples to all the hubs, and the hubs will do the targeted sequencing, and they will have their own biobanks. And on the other hand, the samples will be also sent to Central BGSI to be sequenced there for the whole genome sequencing, and also to be stored in our biobank. And all the systems are linked. So I think this is one of the networking that we have. And then for the data sharing platforms, everybody which is pharmaceutical company, startup, academia, a research institute, they can use our data. Of course, with the system that I can send us a protocol, and then we will review, and if you guys are eligible, then we can give the data based on what you want, and you can play all in the cloud. Because like regarding to our law, our health law, that the data cannot be go outside, so it should be here. So we have our own cloud. Slide. And this is what I said before, that we have the registry platform, and we have the biobank platform, we have the research data platform, and lastly, we will have the Satu DNA. So Satu DNA is the pharmacogenomics report of each participant. So hopefully it can be in the health app of Satu Sehat. So it's like the features there, so you can go in and then you can see about the pharmacogenomics data of each participant. Slide. And these are the progress that we're having at the moment. So we already recruited around 7,500 participants in 10 months. And then we already sequenced around 3,500 samples. And then the sample which is stored in our biobank is now, I think, around 6,000 something. And then we already analyzed 85% of it until the secondary analysis. So I think this is quite an achievement for us. Because like our minister always say, if we ask when is the target, and then he will say yesterday. So are you guys sleepy or not? No, okay. Okay. Next. And this is one of the activities that is conducted by our hubs, which is based on the TB hubs. So what we are doing is first we done, as you know, the WHO, they endorse several RDT, right? With rapid communication. So what we're doing now, we implemented those in those two hospitals. The samples will be 7,600 patients, and we are not working by ourselves, but we also have the networking with the unit within the Ministry of Health, and also the health district in health district and health provinces, and also university and hospitals not under the Ministry of Health. So we are trying to make everybody in the same loop, because otherwise the data will be scattered and then everybody will do everything by themselves and they're not connected. So that is the idea. And the second one is we're going to do the whole genome sequencing of the MTB. And for this, we also had collaboration with the BioCast and other DST labs around Indonesia. And this program is integrated with the national TB program from the CDC, Indonesia. And the third one is the human whole genome sequencing that will be collected for 1,000 patients that will be conducted in BB Binomika or National BGSI. Slide. And these are the networking that I told you before. So we have now, we focuses on the hub of BGSI hospital as a proof of concept. But of course, if this proof of concept is succeed, then it can be replicated to other hospitals as well. And then we're focused on sample only from the region that has a high resistance of TB. Slide. And this is, I told you that one of the key aim is to provide services in hospitals, right? So these are the services that already have in our hospitals, house BGSI. So one is in Dharma East, one is in Harapan Kita. So they already implement the precision medicine-based services for the eye care. I think it's around 113 panels of cancer, and it's already running. And also in Harapan Kita, they tested the NIPT. And for the RSCM or Cipta Mangunkusumo, they still develop the panel for the MOGI. And slide. Yeah, so I think innovations never stops, right? So every day, there are always new innovations, and it's very dynamic. And of course, we have to be in cope with that. So how are we going to use the technology? How are we going to use the innovations? So therefore, there are several things that we need to keep in mind. So for example, the artificial intelligence. So the artificial intelligence now is becoming one of the tools that will be, it's not going to replace the doctors, but it's going to be like the one of the backup, or one of the tools to make the doctors better in diagnosing. So slide, and also the research and development. So this is for the other initiatives. So we have the Clinical Research Center. So the Ministry of Health have the clinical research centers, I think 4 or 5 months ago. And then the clinical center is in Binomika, and we have the hospitals called the Clinical Research Unit. So currently it's the 17 hospitals, but we will expand into 37 hospitals, which is our vertical hospital. So the CRC, what we're going to do is that we are coordinating the clinical trials. So we're trying to negotiate with sponsors, with industry, how, if you want to do clinical trials. So we will help you with the regulations, what do you need if you want to do clinical trials in Indonesia, and also to make a capacity building for all the CRU. And the CRU will conduct the clinical trials, and they also they have their own center of excellence. They also can develop their roadmap, and they also need to, I think this is the hardest part, because as the clinician, the hospital, mostly they're doing services, right? But how to make the research environment is becoming familiar with the clinicians in hospitals. I think that's a big job for us. Slide. And to strengthening the clinical trial system here, what do we need? Of course, the first is the engagement from the community, from the patients. So how we tell them, how we educate them, so they are aware that the clinical trials is actually very beneficial for them, because like they can get their medicine at the first place, unmet medicine, and it can help them for like certain diseases. For example, for now, for the rare diseases, we haven't got any, we have the medications, but the gene therapy is very expensive, those kind of things. And then for the government, so we need to simplify the regulations. For example, what we are doing now is for the material transfers agreement, we have the transformations. So hopefully next month we will launch the digitalizations of the MTA submissions. So all will be transparent, also with the how you can check your application. So it's like a live report. It's like when you're doing the shopping online. So if it's packed, if it's sent, if it's accepted. So we will have that for the NTA applications. And also for the clinical, for the ethical committee, because we know that for those research or like clinical trials that have multiple centers, so now what we do is that we need to apply to each of the ethical committee in the hospitals. So we are trying to simplify that by having the National Ethical Committee. So the National Ethical Committee will apply, the ethical approval will apply to all the sites of the clinical trial. So I hope that will make your life easier, right? And also the hospitals, they need to have a commitment. I think the commitment from the leader is the most important thing. And also how to convince the clinicians to do research. And then other sponsors, other stakeholders for sure, like CRO, industry, the central lab, pharmaceutical company, the startup, and also the professional associations. I think this is my last slide. So thank you very much. And then I will give it back to the moderator.
Thank you very much for your very interesting talks. And then we will go to the next presenter, is Uniati and Biomed. Here, she received her master in biomedics from the Faculty of Medicine, the University of Indonesia. And currently she is public analyst and lab scientist, National Health Biology Laboratory, Ministry of Health, Republic of Indonesia. And this, you can start if you are ready. Thank you.
Good afternoon. First of all, apologize that our head could not attend this event because at the same time there was a launching of public health laboratory. And today I would talk about genomic strategy in infectious disease surveillance and outbreak management. Next slide. I divide this presentation to three sections. The first, introduction, public health laboratory. It is a new structure in Ministry of Health. And the second about our lab, National Health Laboratory, Biology Laboratory, or in Indonesia we call BB Lab Biokes. And the last is about genomic surveillance. As we know that Ministry of Health is committed to implementing health system transformation, and in laboratory we focus on the improving capacity and capability of primary care. This is the first pillars of the primary care transformation. Next slide. And for the public health laboratories, we have 5 tiers from tier 1 and until tier 5. Tier 1 is laboratory in primary healthcare. And we have 2 tier 5 national laboratory. The first is our lab, BB Lab Biokes, focusing for the specimen, clinical specimen. And the second is BB Lab Kesling in Salatiga. They are focused on environment factor and reservoir. The aim of the laboratory testing is for screening, for diagnosis, follow-up, and surveillance, also quality assurance, and also the research development to disease prevention and control. And next. As I said before, that this is distribution of public health laboratory in Indonesia. In regional 4, we divide to 11 regional that cover all of Indonesia. For example, for the Aceh and North Sumatra, there's cover region 1 coordinator by Balai Litbangkes Aceh, and for the Kalimantan covered by BBTKL PP Banjarbaru. Next slide. And also we hope not only collaborating network with with the other health public laboratory, but we hope we can collaboration with another laboratories such as research or veterinary, environmental, biopharma, and so on. Next slide. In a public health laboratory, we have a basic standard service that we have, we must can do examination. There is 81 examination that divide to 8 category. The first, we examination about highest burden of disease and screening for 14 disease. This is such as diabetes, tuberculosis. And then also we do the examination of infectious disease and potential outbreak disease such as maybe Ebola, unknown disease, and also environmental health risk factor and factor risk factor animal transmitted disease is for the pathogen detection in animal transmit disease, and drugs toxicology, and also monitoring of drug resistant such as antimicrobial resistant. Next. Next. BB lab vehicles established based on the Ministry of Health Regulation number 26 on 2023. We have 14 main function, including the laboratory-based surveillance of disease, health risk factor, and respond to extraordinary event, pandemics, and disaster. This is a history of our laboratory. From 1975 until 2021, our laboratory is under National Institute of Health Research and Development, Litbangkes. The main function are research and development. We have biosafety level 3 facilities for the culture of virus and for the research. But after 2021, that government have policy that all of the research institute merged to be one research institute in Green. So we changed the name of our laboratory to be a Laboratorium Nasional Prof. Sri Umiyati under National Health Development Policy. That's a main function is for a national reference center and do examination for the laboratory for the new emerging and re-emerging infectious disease for policy assessment, application, and surveillance. And since the 1st January in this year, we also move again to the Directorate General of Public Health and change our name again to be Balai Besar Laboratorium Biologi Kesehatan or BBLBKES. The main function are as National Reference Lab, Center for Infectious and Potential Outbreak Disease and Surveillance, and also make a quality assurance for the public health laboratory. Next. This is activity that we have done. First, contributed to international network for WHO or CDC. We are our lab is National Influenza Center, and we examination the polio, measles, rubella, and also examination of vitamin A or retinol and zinc, and do tuberculosis drug resistance survey, and also support the national scale laboratory survey such as basic health survey, sero survey COVID-19 that we want to know the antibody of in the community, and then strengthen national laboratory network that we making a training of laboratory biosafety officer. Next. And also our lab is a reference laboratory for whole genome sequencing for the COVID-19 and unknown disease, and in cooperation related to COVID vaccine clinical trials. We provide biosafety level 3 for the industry of pharmaceutical industry, and cooperation related to drugs development and validation test of RDT for COVID, and now still process in for malaria. And also we open to student internship and research. Next. Next. I think it's the same with BJSI. This is next generation, the distribution of next generation sequencing machine in Indonesia. The owner is from different institution from Ministry of Health, and then local government, private sector, and so on. Next. This is an example that what we have done and the benefit of genomic surveillance. The first, we monitoring for COVID-19 variant start from 2020 until to 2024, and we support to government to make a policy to relaxing health protocol, and transitioning pandemic to endemic is based on the pattern of the circulating virus. And also we integrating with influenza and COVID-19 sentinel surveillance for the community case and hospitalized case in based on with WHO guideline, and so we can monitoring of variants virus can be continued. Next slide. This is also monitoring of Mpox variants that we know that monkey Mpox virus in Indonesia belongs to the group West Africa clade 2 lineage B1 and B13. It is preparedness for region and for clade 1, and now we prepare to phase of the outbreak of clade 1. That's we know for the clade now is transmitting not only by sexual transmission, but also can transmission by contact. It's and already found in Thailand this month. Next. And this is a monitoring of influenza variant, part of global influenza surveillance response that we have that in the global influenza circulation, the pattern is increased from the end of the year to the beginning of the year during the winter. But in Indonesia, the pattern appear throughout the year. And from the mid of 2023 until to early 2024, the influenza virus circulating in Indonesia and the world is subtype S3N2 and lineage B factor. This is the last this is the last set of the vaccine. And for Indonesia virus is do not specifically cluster with their closest neighboring country. Next. We also mapping in dengue based on the phylogenetic tree. We obtained the dengue virus from Indonesia is grouped in let's box is one group with the Singapore and Malaysia. And also next. And also we do in 2017 until 2018, the national we have a national drug resistant tuberculosis survey to measure the burden of anti-tuberculosis drug resistant, and we find that in Indonesia, lineage 4 is Euro-American is dominant lineage, followed by lineage 2 ASEAN Beijing, and then lineage 1 and lineage 3 are limited only in East Africa, but it's still found in Indonesia. And this year we also support BJSI to do the whole genome sequencing tuberculosis and also targeted sequencing. Next. This is this is we also find when there is any case in acute kidney injury in child, maybe last year, we don't know what happened in that children. We also do the metagenomic from the sample from the children, and we found this, but at the end we found that the injury caused by the drugs toxic for from drugs, not from the pathogen. Next one. It is the last slide. We analysis the pathogen genomic surveillance situation in Indonesia now. It is I'm sorry, it's too small, that we have the strength, we have many machine next generation sequencing in Indonesia, and also have national epidemic potential disease. Also, we have WHO global strategy genomic surveillance, but in financial and human resource, we still need the advanced capabilities for the bioinformatics data analysis, because we don't maybe still we don't have the bioinformatics to help to data analysis. And also, as we know that genomic surveillance is high cost, need so many infrastructure, technology, and also training. It is also to be opportunity to us to make a collaboration in sharing knowledge in bioinformatics, and also training to how to analyze the data. I think, and also we have trace about potential misuse of genome data, including maybe bioterrorism, and also for the privacy and ethical issue. Thank you. I think it's the last slide. Thank you very much.
Thank you very much for your interesting talks. And we are going to the third presenter. Will be Dr. Luh Inta Prilandari. She is currently a clinical microbiologist at the Murni Teguh Hospital. Sorry. Ngurah General Hospital, Bali, and also in Prodia Healthcare. She received clinical doctor from the University of Udayana and clinical microbiology residence from University of Indonesia. Time is yours, please.
Okay, thank you very much for a brief introductions. I'm Inta. I'm from Ngurah Hospital. First of all, I want to apologize. My medical director should be present the presentation here, but he's in another things to do in the same time. So I will represent from Ngurah Hospital. I will present about how the implementation of genomic surveillance, genomic approach in Ngurah Hospital. Slide. So as continued from the explanation from Bu Indri about the BGSI, Ngurah Hospital is one of the BGSI hub in aging, wellness, and nutrition. So as mentioned before, that BGSI have about 8 or 9 focus disease and 10 hospital as hubs. And Ngurah Hospital is one of the hubs in aging, nutrition, and wellness. Next slide. So for this program, currently we have ongoing research project in pharmacogenomic analysis of methotrexate in psoriasis vulgaris. And we also preparing for upcoming project in term of obesity, in precision nutrition in obesity. So this project is to support the wellness, aging, nutrition, and wellness aspects in our hubs. Next slide. So the aims and the output of this project is, of course, to support the program of the Ministry of Health in implementation of precision medicine in hospitals, especially in Rumah Sakit Ngurah. Slide. So I want to briefly explain about our first project, our ongoing project. So about the pharmacogenomic analysis of methotrexate in psoriasis vulgaris. So this is a novel approach for precision medicine in Indonesia. Slide. This is about the psoriasis vulgaris. So psoriasis is autoimmune inflammatory of the skin. Yeah. So it's like a chronic inflammation caused the skin became it's like a redness and itch, scales, and it can affect all the skin all over the body. So this is the at the left side is the figure of the psoriasis. In Indonesia, the prevalence of psoriasis is 0.3%. It's not a big, it's not a high number. And about the psoriasis, the severity about the psoriasis is based on the total body surface that affect. So it can be differ in a mild psoriasis if less than or equal to 3% of body surface area is affected, moderate 3 until 10%, and if more than 10%, we call it severe psoriasis. Mostly of the psoriasis cases is mild, so more than 50% is mild psoriasis. But this is like a chronic recidivity, so it can recurrent based on the risk factor, and it's very affecting the patient. Next slide. So this is the current psoriasis vulgaris treatment guideline in Indonesia. So based on the severity grade of psoriasis, a mild, moderate, and severe, there's options for treatment from topical phototherapy, systemic treatment, which is non-biological, and biological systemic treatment. So, so for the mild psoriasis, the options of the therapy is topical and phototherapy. But for moderate and severe, there is options of systemic treatment, which contains of non-biological and biological treatment. A non-biological treatment, systemic treatment that currently used for treatment guideline in Indonesia is methotrexate. And the biological systemic treatment will be given if the first line is failed or the patient is in contraindication of receiving the first line. So next slide. So the problem is about the response of the treatment is different. So for now, currently, methotrexate is a primary therapy of psoriasis vulgaris. But the problem is the result more than 50% no improvement, and more than 50% have a side effect of methotrexate. As we know that methotrexate is drugs also currently used for cancer and autoimmune disease. So the side effects is like vomiting, diarrhea, mild secretions, and others serious side effects. So the current situations is methotrexate therapy was given for all psoriasis patients, but the treatment response and the side effect is different among patients. And with this flow or the current guideline in Indonesia, the biological agent was given too late. So other complications of psoriasis will be developed in patients such as diabetes, hypertension, and also heart diseases. Next slide. The next is about the biological agent availability in Indonesia. So first problem is the flow chart is like a general for psoriasis patient, you should get this first line no matter conditions, and if failed, you will move to the biological agent. But the availability in Indonesia is also limited. So and also for national insurance coverage, it's also different. So for interleukin 17, secukinumab, it's widely available in Indonesia. So in Jakarta, Sumatra, Kalimantan, it's most widely available. And this secukinumab is the only biologic agent covered by the national health insurance for psoriasis currently. Besides the secukinumab, there's several biologic agents also that available in Indonesia, but in limited area and with the more expensive cost. Next slide. So based on that, from the literature and the experience, research, they found that the gene treatment response is related with gene. So there are several genes that currently identified associated with the therapeutic efficacy for methotrexate and biologic agent. So we searched the literature and research and found that several genes, about 20 genes, polymorphism that relate with the treatment response of psoriasis. Next. So the importance of the genetic testing, because the genetic might alter the therapeutic approach. So the genetic testing help to improve the approach treatment. So it can decrease the systemic inflammatory response and of course decrease the risk comorbidity. So for the aim for the importance of genetic testing is for precision medicine in the case of psoriasis vulgaris. So the early of a biological agent may be given. Sorry for the disturbance. So next. So we do, this is the flow of the research. So the research is about analysis of genetic role in personalized treatment for psoriasis vulgaris, focused in methotrexate as the first line agent and the biological agent, interleukin 17. So there is two phases. For phase 1 is case control. So we do the validation of relevant genes that we already seek in literature and from previous research from Caucasian mostly population. We want to see the relevant gene for Indonesian populations. And for phase 2, we will run the research in larger sample size. And the aim of this study is to develop the recommendation of treatment guidelines of psoriasis vulgaris in Indonesia. And of course, we want to simplify the examination. So the research is conducted by targeted next generation sequencing. So we do the sequence of the target gene that is related with the response of psoriasis. So if we found the relevant genes for Indonesian population, we hope that we can develop the commercial PCR-based panel for psoriasis that can widely used and more cheap than sequencing. Next. This is the inclusion of the subject. So we run the research in the study in our hospital. The subject also, the samples, the specimen of the subject also sent to the Ministry of Health, to the Central National BGSI to support the project of Indonesian Human Genome Projects. So the benefit will be for all Indonesian. Next. And for upcoming project, we also for support our hubs as aging, nutrition, and wellness. We want to explore about precision nutrition and obesity. Next. So as we know that the prevalence obesity in Indonesia is based on the Riskersdas 2018 is quite high. So in female it's 44% and male is more than 20%. Next slide. So obesity is now considered as disease and it can complicate and develop to more serious disease such as diabetes, dyslipidemia, cancer, mood, mental health, heart disease, reproductive liver, and all systemics. This problem that caused by obesity, of course, became a global health problem that can cause also of hospitalizations, and of course, it will be a big burden of national healthcare system. Next. So from what we plan to do, we will do the genomic approach of nutrition therapy in patient obesity. So we want to change the paradigm of a curative become preventive. As we know that there is no one size fits all, or in choosing the nutrition therapy that fits for every patient will be based on the genomic profile of each patient. Next. So this is the our plan. We already in the first phase. This is our research plan in obesity plan. We want to toward the Center of Excellence Obesity. So we hope that Sangla Hospital will also become a center of obesity in the future. For now, we are doing health surveillance for hospital employee based on anthropometry. We are analyzing the data. Based on that data, we will have the data, the status, the nutrition status of hospital employee, and for the nutrition status of obesity will be also examined with bioelectrical impedance analysis. And after that, move to genomic using the microarray with, we want to see the genomic profile of patient obesity, focus on vitamin D and focus on deficiency of vitamin D and deficiency of antioxidant. So and after that, we will get the precision nutrition therapy based on BIA and based on genomic profile. This is the roadmap of our plan. We hope that it can end with the center care of obesity in our hospital. This is my last slide, but we also have a lot of challenge in doing the genomic approach in the hospital setting, as we know that research culture in the hospital is orientation for the services, so it's very difficult to change for that culture. So it's like the challenge from the human resource, the culture, also for cost, because we know that the cost for genomic testing is very expensive, and the most problem is the data. So we are lack of data analysis. So genomic data is very big. So it's what we are lack of. Okay, thank you. It's last of my presentation. Thank you very much for your attention.
Thank you very much for your interesting presentation. And we are going to the next presenter, Professor Dr. Wang Xingquan. He received his PhD from the Institute of Biophysics at the Chinese Academy of Sciences in 2000. After that, he spent 3 years as a research associate at the same institute and 5 years as a postdoc at Stanford University. Professor Wang's research groups employs structural biology, biochemicals, and related functional methods to investigate protein complexes involved in virus-host interaction. Professor, time is yours.
Thank you for the introduction, and also thank Lin Qi and the Indonesian organizers for inviting me to attend this event. Today, I will give you a brief introduction about the utilization of the structural information in the immunity design and vaccine development. Next, please. I'm a structural biologist. The scientific goal of the structural biology is to understand the functional mechanism of biomicromolecules by determining their three-dimensional structures. With the knowledge of the high resolution structures such as the antigens and the antigen antibody complex, the structure guided vaccine development has been emerging and making significant progress over the last decade. Next, please. So how can the structures help in the vaccine design, in the immunity design and the vaccine development? So in the basic research step, we can determine the structure of the antigen and its dynamics. And also we can map the neutralizing epitopes. And we can also discover the mechanism of the neutralization of the antibodies. So in the engineering steps, the structural information can be utilized to identify the neutralization domains or to stabilize the antigen conformations and with other purposes. I will give two excellent and successful examples of the structural guided immunity design. Next, please. So the first example is about the SARS-2 vaccine. So for nearly all the mRNA vaccines targeting the spike glycoproteins of the SARS-2, two protein mutations were introduced to stabilize the pre-fusion of the spike glycoprotein. So, so you can see the aquarium pictures. You can see it's for the MERS spike Y type and also the MERS spike with two protein mutations. You can see the difference of the particles. The particles in the red square are in the post-fusion conformation, while you can clearly see the difference between after introducing these two protein mutations. Nearly all the spikes are in the pre-fusion stage, just reducing the numbers of the spikes in the post-fusion stage. So the mechanism is to just inhibit the contact helix formation during the pre-fusion to post-fusion transition stage. So to stabilize the spike protein in the pre-fusion stage. So this discovery was made by my previous PhD student, Wang Lianshuang, when he did his postdoc in the United States. Next, please. So the second example is about the RSV. So Dr. Shilong Fan mentioned this in the morning. Actually, by determining and analyzing the complex structure of the RSV F fusion protein with the potent antibodies, so the scientists can introduce the mutations in the F protein to stabilize and preserve the neutralization sensitive epitopes. Next, please. So although structure guided immunity design is quite successful for SARS-2 and RSV, this task is still very challenging. One HIV vaccine development is one example. Although we have known the epitopes of many neutralizing antibodies, but it's still very hard to design an HIV immunogen to induce the broad and potent immune responses in the human bodies. The results are still not very satisfactory. That means we still have many knowledge gaps about the human immune responses to pathogens. So next, please. So during the second part of my talk today, I will just introduce, briefly summarize our results about the coronavirus spike glycoproteins. It is the key molecule for coronavirus infection and the major target of neutralizing antibodies. Next, please. So here I showed the crystal structure of the receptor binding domain. It's one domain of the spike complex with these receptors. You can see for the SARS, SARS-2, and MERS RBD, they all use the red region we call the receptor binding motif, the RBM, to specifically bind to their respective receptors, SARS to ACE2 and MERS to DPP4, and also SARS2 also binds to the ACE2 receptor. So next, please. So by using the prior-EM, we can also determine and capture the different conformational states of the whole spike trimer. So you can see the one major discovery is that achieved by my group and other groups that for the SARS, MERS, and SARS-2, so in the spike trimers, the RBD can adopt a different conformation, and the down to up conformational state transition is essential for the binding to the receptors. So next, please. So we have a model, means that the spike on the surface of the virus, they have the prefusion and also the receptor binding in active stage. And when the RBDs are all in the down position, but we need at least one RBD in the up position and change the spike trimers to a prefusion, but it's the receptor binding active stage. So after binding to the receptors, it can induce the deassociation of S1 subunit and expose the internal S2 subunits and enabling the prefusion to post-fusion transition stage, which can induce the membrane fusion. That's an essential step for the viral entry. Next, please. So our results and other group results showing that the two key features of the S-glycoprotein contributing to the high effectivity. One is the efficient RBD down to upper conformational transition, and second is the tight RBD receptor binding. Both are required for the high effectivity during the evolution of the coronavirus, jumping from animals to humans and others. So you can say that for the neutralizing antibodies, you can imagine or you can propose that there are several different mechanisms. One is to directly inhibit the binding of the RBD with the receptors. The second one maybe can stabilize or prevent the prefusion to the post-fusion transition. That's also essential for the binding and the membrane fusion. And so next, please. So after the outbreak of the SARS-2, Lin Qi's group and Professor Zhang Zheng's group at the Shenzhen Third Hospital, they collaborated very efficiently to isolate and characterize the neutralizing antibody from the SARS-2 infected patients. So they characterized Lin Qi, how many? 200 or 300 antibodies for the first batch. Okay, they identified the first three antibodies with very high potency. We call it 1F. So the top one is the 1F11. Okay. Okay. So as a structural biologist, I'm very curious to know how where the antibody binds and how it can inhibit the infection. So next, please. So by determining the comp structure or the crystal structure, you can see that these three top antibodies bind to the RBD directly, but with different orientational angles. So all the three antibodies overlap with the ACE2 binding sites. But you can see from here that the 1F11 has the most extensive overlapping with the receptor ACE2 showing in the green. And that's consistent with the functional data showing that 1F11 was the top candidate at that time that has the very efficiently inhibited the infection of the SARS-2. So you can see from the structural information directly that this antibody mainly use the mechanism for the receptor competition to inhibit the infection. So next, please. So for the NTD, it's another domain in the spike that is not directly involved in receptor binding, but scientists also identify several antibodies with highly potency in the infection. By determining the structures, we can also know that these antibodies bind to the NTD. So one possible mechanism is that these antibodies can inhibit the prefusion to the post-fusion transition stage that can have their inhibitory activity. So next, please. So I just mentioned the S2, that's the internal power subunit. Actually, there is also antibodies that targeting the S2 part that had broad activity, although not very potent. By determining the antibody structure with the helix peptide from the S2, we can see how the antibody bind to a helix in the S2, and the neutralizing mechanism maybe is also involved in the prevention of the pre-fusion, the post-fusion transition stage. Okay, next, please. Actually, we know that there are many SARS-CoV-2 variants after the emergency. Actually, Lynch's group also found that the 111 also retained inhibitory activity against the many SARS-CoV-2 variants. So you can see from the table here that three major mutations. One is the let me see, K. It's the lysine 417 to the asparagine, and also the asparagine 501 to tyrosine, and also the glutamate 448 to the lysine. These three mutations disrupt the most of the antibody at that time for their efficacy. So by next, please. By comparing the complex structure of the antibody with the RBD wild type and also RBD harboring these three mutations, we can see that these mutations actually did not disrupt the interactions of the binding. Actually, the antibody shows the resilience in targeting the RBD. Okay, next, please. So I will quickly go through this the following several slides. So we also collaborated with Lynch's group to characterize the nanobodies that also target the spike for the inhibition of the infection. So the nanobody has several advantages. It is small, and also it can access to the epitopes that is inaccessible by the conventional antibodies, and also the nanobody can be administered by inhalation. Okay, next, please. So also Lynch's groups isolated and characterized many nanobodies, and also we grouped them into G1 to G4 groups, and we chose the 3, 2, A, 2, and 4 in the G3 group because it is very broad and potent in inhibiting the SARS-CoV-2 Omicron subvariants at that time. And also it can inhibit the diverse subecal viruses. Okay, next, please. So also by determining the complex structure, we can clearly know the epitopes of these very special nanobodies. It has a very unique epitope, and that is not accessible by the conventional antibodies. And also with the structural information, we can also explain its mechanism for the broad and potent neutralization. Okay, next, please. Okay, to summarize, and with the efforts of many groups during the COVID-19 pandemic, so we know a lot of the epitopes of these antibodies. Most of them are targeting the RBD, and also several of them targeting the NTD. So with all this information, we have a clear and also a relatively complete image of the neutralization epitopes of the RBD. That's very important for us to understand the neutralization mechanism, and also provide very invaluable insights for the vaccine design, and also maybe for the future our preparedness for the next pandemic in the future. Okay, thank you very much. And I would like to thank my group and also our collaborator, Lin Qi, Xiang Ye, and Ding Qiang, and also the CTC group. And also thank you for the agency providing the money for our research. Thank you very much. Thank you.
Thank you very much for your interesting presentation. And we are going to the fifth presenter, will be Professor Dr. Kian Ding. He is an associate professor at Tsinghua University, and his research group specializes in studying RNA viruses and that pose a significant threat to human health. Okay, Professor, you have 15 minutes. to present your.
Thanks for
Yes.
your kind introduction. Actually, very happy to be here to introduce the work in our lab. Actually, we are pretty interesting, establish novel cell culture model, try to understand the basic science about how the virology, as well as the interaction of the virus with the host. So today, I would like to give you two cases we developed. Actually, how does I think this kind of system can be used in cell culture, in antiviral, as well as the vaccine development. Next. And the background of our research, actually the context of the recent emerging as well re-emerging viruses. We can see from this figure that lost virus infection, and the next one is about different kind of outbreak during the past 20 years. Probably some of us actually start in China. And however, due to technology restriction, we still have a reliable approach, try to predict, what's the next, as well as when the X disease will outbreak in the context. Next. And today, I'll first introduce one as the first case about the still coronavirus, but it's different from SARS-CoV-2. Today, it introduced about another one. This virus has been identified in China in 2017. It's in Guangdong province. And originally, they think that it's come from the bat. However, the pigs can be infected by this virus and develop syndromes such like diarrhea, and even can leading to the death of the pigs. And Rothbari group and use reverse genetics approach to rescue this virus, and it has showed evidence that this virus definitely can infect human cells in vitro. So with this indicating that we have the potential to have a spillover from animals to humans. Next. In order to introduce you about, before to introduce the tools we developed, I would first like to introduce the basic virology about coronavirus. This virus is an envelope virus. They have a positive strand RNA virus genome with a 30 kilobase RNA base. And they divide it into non-structural protein as well as structural protein. We know a lot about, spike protein, just as Xinquan introduced. And a structural protein, it means that, associated with our particles. It's so important for vaccine development and responsible for virus entry. And the non-structural protein is also very important. It's only it's occupied 2/3 of the entire genome, and it's very difficult. It's very important for virus genome replication. And we can see they have proteins here, and they also has RNA-dependent polymers here. This is definitely very attractive drug target for antivirus development to blocking virus replication. Next step. So usually in virology, we will really like to talk about the life cycle of the virus. And you can see the virus can interaction with the receptor. For SARS-CoV-2, of course, it's ACE2. For other coronavirus, they have used different receptor. Today, the SARS virus with the receptor, which still don't know the receptor right now. And the spike protein will interaction receptor, then will the virus will get into the cell, and they will release its virus genome. As I mentioned, its genome is polymerase RNA virus, and they have a release genome for translation to have viral protein. And with the help of the proteins, will dissect into individual proteins to achieve this function. And for example, the RDRP will be responsible for replication and transcription for viral genome. And the structural protein will be responsible to packaging progeny viral genome, have the viral particle then released into the cell. Next slide. And in the lab, we usually divide the virus life cycle into three different steps. And then the purpose is that we want to have a tool to study each step. For example, in the entry step, we usually use to evaluate antibodies, vaccines, and we call this entry. We usually use a pseudotype virus system to study the process. And in replication stage, we'll just delete the virus structure protein and keeping the non-structural protein there. They can have the replication process that we call the replicome system. As for later assembly as release, you just overexpress the structure protein, then structure protein can self-assembly and release in the cell. Then we use this kind of three kind of system to study this virus life cycle in the lab and make sure it's very specific for each step. Next one. For each kind of system, they have a loss advantage and also have a loss disadvantage. For example, live virus definitely uses BSL-3. However, this kind of pseudotype replicon VLP is just a BSL-2 because it's only mimicking one kind specific step in the virus. They don't have a live virus production, so it's safe. And as I mentioned, so the life cycle for the live virus is the entire life cycle. For pseudotype, you can only mimic a viral entry. Replicon is only replication for VLPs like that. And however, sometimes we will want to specifically introduce to study the virus entry, we will use the pseudotype of virus. Next slide. The pseudovirus means that they have they can produce new virus and viral vectors in combination with the foreign viral envelope. It means that you can just use different viral envelopes, then you'll have different kinds of pseudotype viruses. And we always, the backbone we're using is HIV or VSV vector or either kind of retrovirus vector. So it means that in the surface of the virus, you can put on different viral envelopes and you have different kind of pseudotype virus. Next step. And I will kindly introduce you with one example about the VSV backbone we are using in the lab. This vector is widely used because it's very safe and is also previously used for a VSV Ebola virus vaccine development and have been approved as a very safe vector. Next one. And what we're doing in the lab, you just clone the viral genome and put in the backbone and also help with the NPLG is can help the virus to form RNP complex. It's very, very essential for virus to initiate replication. You transfer this plasmid into the cell, and then with the help of this helper plasmid, the viral genome can be replicated. They will have a genome and they can packaging getting out of the cell. Then you have a replication competent VSV. This life cycle VSV. Next slide. And what we are doing in the lab is we replace the G protein. G protein actually called glycoprotein of the VSV, just like the jacket, if you wear a jacket in the outside. And as I mentioned, there's a pseudotype of a concept. You just replace the G protein with other kind of viruses. For example, here we use the SCDS coronavirus spike protein. It means that you can create a novel virus. This novel virus still can be replicated. And however, the difference from the Y type one is that in the surface, they can already bearing another kind of virus. Then we transfect these things into the 293 T cell. Then we're packaging the virus. It's very successful, and you can see you can also engineer reporter in the viral genome. It can be really very easy to monitor virus infection. And you can have the virus, the packaging can replicating. And interesting, we found that according to the passages along, you have the enhanced virus infection. Next one. And we're curious why, then passage 6, why the viruses have enhanced infectivity. With sequencing the viral genome, we found that pretty interesting. They have the spike protein in the C terminal. They have 11 residues deletion. And if you delete it, the virus infection can increase 2 logs. And morphology still looks like the VSV. And very specifically, we use the antibody specific target SCDS spike. It can neutralize this infection. But it cannot neutralize the SARS-CoV-2 pseudotype virus. It means that the entry is dependent on SCDS spike. Next one. And what's the mechanism? You want to understand why, as a basic scientist, we want to understand why the deletion of 11 residue can promote the virus infection. And we think that after deletion, it can lead into the spike protein more in the cell surface. So this is the reason why it can promote assembly of the VSV pseudoparticle getting into the cell. The next slide. And interestingly, you can find that another very important protease in the host cell is TMPRSS2. Is a protease can help to processing virus spike protein. And if you overpress TMPRSS2 into the cell culture in vitro, can definitely promote this VSV SCDS coronavirus infection. Next slide. And another, I think, very important implication is that it can, because it's the virus, right? It can bearing the static spike. We think maybe it's a good candidate to develop as a vaccine. Then we prove this concept. We prime and boost this mice with this recombinant chimeric virus. As you can see in the C panel, it can induce very nice neutralizing antibody, and this antibody can also block the authentic virus, as shown in panel D, SCDS coronavirus infection. So it's a very nice candidate. The next slide. I don't want to waste time to summary. I will very quick to introduce the second story. Next one. As for another is for the flavivirus, as we say, the one we choose is Povacin virus because this is a really emerging arbovirus concerning human public health. Next one. This virus belong to Flaviviridae family, and it has a very single positive strain RNA genome, and very compared to coronavirus is only 10 kilobase. And this tick transmit can lead into fever, loss of coordination, or even encephalitis. And in this family, lost very important virus, as we mentioned, lost in Indonesia today here is the dengue virus or Zika virus or something. Next one. And we call, in this system, we use a replicon system. Replicons, I mentioned that can only mimic virus replication. It means that you can delete virus structure protein, for example, here is core, pre-am, and E protein, and replace with the other reporter. It's very convenient to monitor virus replication. Next one. As we mentioned, we constructed this Povacin replicon. We replace the C, pre-am, and E protein with BSD, which kind of antibiotic resistance gene, as well as Go cell receptors are very easy to monitor virus replication. As you can see, we clone virus genome and we put into the Q7 cell and put in the blastocydin selection. Then you have the Q7 cell harboring very active viral replication. We call it we call it Povacin replicon cell. And the replicon cell can support, have a very high abundance of virus RNA genome replication into the cell. Next one. And we use that kind of system to try to evaluate with kind of antivirus can block viral replication. Then we developed a very small high throughput screening. You can see a lost compound I put in the green color here because they have very nice inhibitory effect and also have very less cytotoxic effect. The next one. The critical things, we want to evaluate whether it can be used to develop for evaluate antibody or kind of vaccine strategy. Here we use a single round virus production system, as I mentioned in the left panel. You can imagine in the cell you already have the virus genome there, right? So, however, in this context, you can transfect with other flavivirus structure protein. This structure protein can still function. It can packaging the virus genome and have the virus particle released into out of the cell, and it can even infect another cell because they have structure protein. However, in the virus genome, they still don't have structure protein, and the virus can still replicate in the cell, but they can never package and then production anymore. So it's a pretty safe system. And use kind of the system we transfect into the Povacin replicon cell. If you transfect the Zika virus structure protein, you can have Zika virus pseudoparticle. If you transfect with Povacin or dengue virus structure protein, then you have the viral particles having the surface is bearing different kind of structure protein. Then, as shown in the B panel, this recombinant pseudotype viral particle can infect the cell very well, and this definitely depends on transfection with the core as well as the pre and ME protein. Next one. And kind of interesting because Povacin virus as well as Zika virus, they belong to Flaviviridae family. Then we got our lab is a neighbor with Lynchy actually. Lynchy developed a lot of antibodies against different kind of virus. This Zika different kind of molecular antibody is Zika, targeted Zika E protein. And interesting, we found that some monoclonal antibody can very effectively neutralize Zika because it derived from Zika. However, for the Povacin single particle, it cannot neutralize them anymore. It means that even they belong to the same family, the same genus even, but they still show different kind of epitope. So according for this, it means that we have to develop a Povacin specific approach and or vaccine to block this virus infection. Next one. I would like to summarize the second one is that we established the Povacin replicon system in culture by reverse genetic technology, and we found that adaptive mutation. I skipped it here. You found that the virus in replicating in vitro, they also need to adapt the cell culture in vitro. They need to have some mutations there, and this replicon system can be used for antiviral discovery for sure. Another additionally, I think the replicon-based, based a single round infection viral particle system can be established to assessment across reactivation of different kind of antibodies. According to our data, we highlighting, this kind of different flavivirus definitely have a distinct antigenic profiles. Next slide. I would like to take this opportunity to thank the college and the students in the lab, and thanks for Linqi to supply a lot of region for this research. And authentic virus infection definitely is done at Fudan University, and this is a grant to support the research. Okay, thanks for your attention. I would like to stop here.
Thank you very much for your kind and interesting presentation. And now we have the last presenter for this afternoon. I would like to introduce Dr. Sin Liu. She is a research assistant professor in the Center for Immunology at the School of Basic Medical Sciences, Tsinghua University. She obtained her PhD in immunology from the Cornell University in 2014. And her research focused on the molecular mechanism underlying the differentiation, maintenance, and functional regulation of long-lived plasma cells and the application of these basic theories in the vaccine development. Okay, it's yours.
Thank you for the kind introduction, and I would also like to thank Dr. Zhang Linqi for the invitation. So at Tsinghua, we actually have a sizable community of basic research scientists that's committed to the discovery of new targets for drug and vaccine development, and I'm one of them. And so today, I'm very happy to share with you our recent studies on the molecular mechanisms regulating long-lived humoral immune memory. So needless to say that antibodies are crucial defense molecules induced by vaccines, and this is best illustrated by the summary here, the graph here summarizing the efficacies of different SARS-CoV-2 vaccines. And you can see that the efficacies are highly correlated with the serum levels of protective antibodies. Next, please. And so how do we induce higher antibody production? So in order to understand that, we need to first look at the humoral immune response induced by these vaccines. And so I'm showing you some data from the real world data. So we're looking at the protective antibodies induced by the SARS-CoV-2 mRNA vaccine, and you can see that after the primary immunization, you see a slight increase of these antibodies, and then it decline, and then the second immunization would induce a huge increase of these antibodies, which will steadily decline, but the fraction of these antibodies will persist long after the second vaccination. Next, please. And so this is actually a typical humoral immune response. And to understand how we can increase the antibody production, we need to understand the cells, the memory cells that's underlying these responses. So the spike of antibodies after the second vaccination is actually contributed to by the memory B cells that are induced by in the primary response, and then quickly respond after second vaccination. And the persistence of antibodies long after the second vaccination are mainly, these antibodies are mainly made by the long-lived plasma cells that are residing in the bone marrow. Next, please. So both the memory B cells and the long-lived plasma cells are coming from germinal centers. So these are microdomains that are found in the secondary lymphoid organs where the B cells can actually evolve their antibodies. So germinal centers are partitioned into the dark zone and the light zone, and in the dark zone, the B cells will actively proliferate and mutate their antibody genes, and then they migrate to the light zone where they would be scrutinized by the follicular helper T cells, and which will drive their differentiation into memory cells and long-lived plasma cells. Next, please. And so we actually spend quite a bit of time to look at how memory B cells are differentiated from germinal centers. Next, please. So memory B cells are quiescent cells that are activated and antigen. There are cells that were previously activated specific for the antigen and return to quiescence. And so we think that some of the germinal center cells, they would become a quiescent cells again and commit to the memory B cell fate. So in order to analyze these cells, we took advantage of a cell cycle reporter that was previously published. So in mice transgenic for this cell cycle reporter, the cells in the G1 phase of cell cycle would express a red fluorescence protein. And when the cells enter cell cycle, they will degrade this red fluorescence protein and express a GFP. So as expected, you would see that in germinal centers, because the B cells are highly proliferative, most of them are green. But then you will spot some of the red cells, or those are the cells that return to quiescence already within germinal centers and committed to the memory B cell fate. So with the tools to identify these precursors, we can then actually analyze their transcriptomes. And what we found is that a transcriptional regulator called GFP318 is highly upregulated within these memory precursors within germinal centers. Next, please. So to further understand the role of GFP318 in memory B cells, we took a loss of function approach. And so, and found that they are actually required for memory B cells to participate in the recall response. So as you can see here, compared to the wild type control, the memory B cells in the mice with GFP318 removed from their memory B cells will have a comparable antibody response in their primary response. But their antibody response are greatly impaired after second immunization. And this is because GFP318 is critical for the integrity of their mitochondria. And so in the knockout mice, the mitochondria is so crippled that they cannot sustain the rapid proliferation induced by secondary activation. Next, please. And so in collaboration with Dr. Yugo Tan, who's also here today, we were able to introduce exogenous GFP318 into the CD73 negative memory B cells. They're not capable of the recall, participating in the recall response. And we programmed these memory B cells to make them now capable of reactivating again. Please. So next, I would like to share with you another story that discovery that we recently made about long-lived plasma cells. These cells are responsible for sustained antibody levels after long after the vaccination. Next, please. So we know that different vaccines for different pathogens induce different lengths of protection in humans. Some of them could last for a lifetime, while others could probably protect for only several years. And this differences is mainly accounted for by the persistence of long-lived plasma cells. So these cells are terminally differentiated and specialized antibody-producing cells that's living in the bone marrow. As you can see by the electron microscopy picture here, these cells have like really expanded ER structures and highly capable of secreting antibodies, but they are really rare in the bone marrow, which makes the analysis of these cells really difficult. And they lack the surface B cell receptor complex, so that it's impossible to identify these cells by their antigen specificity, and also they lack surface markers, so which makes the analysis of these cells really difficult. Please. And so in order to solve that problem, a couple of years ago, we designed a mouse line that allows us to track the cells, because long-lived plasma cells are really defined by their longevity. So by building up a mouse model, which we inserted a CRE-ERT2 cassette under the promoter of BRIM1, which is the master transcription factor for plasma cells, we're able to and cross these mice to a reporter line. We're really able to track the cells across time. Next, please. So the idea is that when we treat these mice with tamoxifen, we can induce RP expression in all the plasma cells. But over time, the short-lived ones would die out and replaced by non-fluorescent cells, and the long-lived ones will remain really RFP positive. Next, please. And so by performing single-cell sequencing analysis, we would compare the cells that are really long-lived to the total plasma cells. And in this way, we could identify the plasma cell subsets that are really long-lived. Next. And by this method, we were able to track and we were able to identify the surface markers for the long-lived plasma cells and really track their persistence over time. And as you can see on the left side here, we're able to identify cells by their surface markers that are really long-lived. And in doing so, we're also able to sort out these cells and take a closer look at these cells by electron microscopy. Next slide, please. So as you can see that the long-lived plasma cells are on the left side here, the ECM high and 6 negative population, and you can see that these cells actually have a much more enlarged ER structure compared to other plasma cell subsets, which means these subset of cells are not only long-lived, but they're highly functional. So by doing this analysis, we have identified a subset of plasma cells that are both functional and long-lived. And we think that, so we're continuing the analysis of these cells and to look into how these cells are differentiated, where they live, how their functions are regulated. And we think that the understanding of these cells are really hold a key for future development of vaccines that have durable protection. And so, I hope I have introduced you to you some of our studies on both memory B cells and plasma cells, and our group have devoted a lot of time in understanding these two populations of cells, and we have also published several papers, and there are much more going on that I might not really have the time to show you today. Next, please. And so with that, I will I'm part of the Laboratory of Dynamic Immunobiology that's established and led by Dr. Hai Qi, who's also the Executive Vice Chancellor of Tsinghua Medicine. And so we are a group of more than 40 people, including postdoc students and technicians, and I'm highlighting a couple of people in the leadership here, including myself. And I will also like to acknowledge the people who have contributed to the work I've shown you. And many of them, some of them are continuing to work with Hai and I to discover more about the cells. And thank you for your attention.
Thank you for your presentation, and please give your best applause to our presenters. Okay, due to limited and tight schedule, so we will I only will facilitate to person to ask our presenters. So, any question? Okay. One. Okay. We have two.
Thank you. I am Stephanie. I'm from Ecolab International. We are one of the research organization in Indonesia. And I have a question, especially for Windri. As you have already mentioned before, that we will have the biobank, and then there will be a data sharing, and then also database. Maybe my question is, how will we can access to the database, and then how is about the it said that CRC will be the matchmaker between the CRO and also the hospital. How can a CRO involve in the theme of the precision medicine And then maybe my last question is about since the genomic data is quite sensitive, so how is the mitigation plan for keeping the security and privacy of the subject data? Thank you.
Okay, I'm just gonna straight to answer the questions. So the first one is regarding data sharing. So yeah, so we are currently we are building the platforms for the data sharings. So hopefully it will be ready in, I think, beginning of 2026. So what you have to do is that we will have the system. So there will be like such a requirements, like you can send us the proposals and then we will review the proposals, and then if it's met all the requirements, then we can give you the data that you ask. And then since the data should be in Indonesia, so it will be in our cloud environment, so you can play in the cloud. So that's for the first questions. And the second questions will be about the roles of CRO, right? So your role is very easy. Just bring lots of clinical trials to Indonesia. That's it. Because now I don't think there is the clinical trial for gene therapy, right? So yeah, so you can sell us Indonesia. We already have the CRC. Now the process to do clinical trials is more simple. I think it can be shown by the vaccine TB-72 that will be studied, hopefully, I don't know, in 1 or 2 weeks. So the Bill Gates MRI, they finish all the documents, I think, in a short period of time. And the third question is regarding, what's the third question?
About data security and
data security. Okay, so we already think about that. So don't worry, there won't be any accident like the previous one. So we identified all the samples. So we have the specific numbers, and then it won't trace back to the participants. And for the safety, we have a backup as well. So our team already think about this. And, you know, we have the protection data privacy law as well. So, and before the BGSI form, I think we already have the study of the LC, the ethic, legal, social implications. So, yeah, so hopefully everything will be in place. Thank you.
Thank you very much, Wendy.
Second question.
Good afternoon, everyone. My name is Indra from Biopharma. So I have a question for Professor Wang and Dr. Lin. So for Professor Wang, we know that in the COVID-19 vaccine from Moderna and Pfizer, they already mutate the spike antigens into a stable format. I wonder how far the sector guided vaccines can help us to develop a vaccine that quite resistant to genetic variability like SARS-CoV-2. Because we know that there's a lot of variants and perhaps the vaccine will not be quite effective for the variants that emerge in the later of the pandemic. And the second question for the other question for Dr. Lin. Studying B cell is quite challenging, especially when we are trying to replicate what we have done in mice and translate it into humans. Do you have any strategy for that maybe we need to careful in order to translate your finding from mice into humans? As we know, taking a sample from human, especially when we like to study long-lived plasma cell, is quite challenging as well. And the second question is, I wonder what kind of insight by studying what triggering to make the B cells evolve into long-lived plasma cell and memory B cell that we can learn from that process into vaccinations.
Can I repeat your question? You just asked it to me, so to make sure that I understand correctly.
Yeah.
So you want to know how the structure information especially for the SARS-CoV-2 because there are so many variants. So
Yeah.
how the structure can help the future vaccine design or the SARS-CoV-2 vaccine design or
Yeah, just not only for SARS-CoV-2, but for the pathogen that rapidly evolve.
Okay. Yeah, that's a very good question. So for the SARS-CoV-2, there are many, but now that there are many variants after the initial emergency and many mutations can disrupt the activity of the available antibody at that time. You know, so for the vaccine. Right now, so for the structures, we can provide very basic information, maybe telling people how the mutations can disrupt the antibody response, very specific antibodies. Yeah, but for the vaccine design, the idea, the perfect image is that we can utilize all the structural information to design a new immunogen that can induce broad protection. But you know the SARS-CoV-2 because it's an emergency. So for the following vaccines, people can just introduce that mutation in the vaccine design without using the structural information. But for other pathogens, I just mentioned just to show two cases of how the structural information can be integrated into the successful vaccine design. Now, actually, this is just the beginning because the pathogens and immune design actually is very challenging. I just showcased the HIV case, you know, so because it has been started for so many years. And we knew the actual epitopes of so many antibodies, but it's still very challenging and not satisfactory to really develop a useful or effective immunogen. And so actually, antibody is just one aspect of the human immune responses. You know, so for many T cells and others, you know, so. I think the key is still we have many knowledge gaps. So from the basic viewpoint of basic science, actually I cannot give a very satisfactory direct answer. I just want to say that we know more and we can do more. So we are still at the stage of gaining try to collect more and more information from basic science like the Dr. Chi and Dr. Liu just mentioned about how the B cells, you know, can react and how the halftime or something like that, you know. So we have to collect the basic knowledge from many disciplines to tackle that problem. Hopefully, we can achieve that goal, but maybe it's impossible finally. It's still possible that we yeah, because it's the evolution between the fight between the pathogen and our humans. That's also the how to say that, the driving force for the evolution of both humans and the pathogen. That's my personal view. Okay, yeah, thank you very much. Yeah,
thank you, Professor.
I think that is like a really good question in terms of like mice studying human immunity in mice and human. So I think mice certainly provides us with more freedom to do a lot more things than you can do with human cells. Like all these fancy mouse models that we could use to answer a lot more questions. But also certainly, I think there is also this limitation that you don't know what's going on in the mice, whether that applies to humans. So at the end of the day, you still have to go to the human system, which will be more challenging. And we're trying to do that right now. So that is really work in progress. And your second question is that how we could use what we learn with memory cells and plasma cells to make a better vaccine. Am I understanding that correctly? So We can do that to some extent right now because we know that, for example, like in the case of CFP318, we know that the molecule is really helping the cells to cope with the oxidative stress. So which means if we actually decrease that stress during booster vaccination, we could actually enhance the reactivation of memory B cells without doing all these, genetic modifications. And we are still on the road of learning more how we can actually enhance that. That's also work in progress. Thank you.
Okay, thank you. We have one question from Zoom to Dr. Inta. So the question is why the emphasis of the genomic test for obesity is on vitamin D and antioxidant deficiencies.
Okay, thank you for the questions from the Zoom. So we already also do the services, start the services for nutrigenomics, and the clinicians in our hospital have interest in a specific related with vitamin D and antioxidant. It's because of the treatment for obesity relate with those deficiency might hopefully become a modality for adjunctive treatment for obesity based on a genomic profile. Hopefully it answered the question. Thank you.
Okay, thank you very much. This is the last answer from our presenters. So please, once again, give your best applause to our presenters. Okay, I will return to the MC. Thank you.
Thank you so much, Dr. Dani, our amazing moderator. Ladies and gentlemen, please give applause to our speakers and moderator for this dynamic, fruitful discussion. So I would like to invite the speakers and also moderator, you may please be upstanding in front of the table as we will continue with the photo session as well, same as the previous speakers on our previous plenary forum. So please photo video, be ready. We have to capture this moment before we are continuing to our last plenary forum of today's symposium. So please face the camera, speakers and moderator. Yes, allow me to do the count. Wait a moment. Yes. Yeah, we still have a photographer over there to capture this moment. 1, 2, 3. Is it enough? Okay, from that phone. All right, at the center. 1, 2, 3. Okay, thank you. Please give applause once again for our speakers and also moderator for a very insightful presentations. Yes, we will prepare the stage first, ladies and gentlemen, for our next plenary forum. Well, ladies and gentlemen, we have heard together the insight from each speakers on the third of plenary forum. They have shared their experience, their perspective and knowledge about facing the current trends and future directions of genomics, particularly in disease prevention and management. We do hope their insightful presentation can give a significant impact to all of us here in developing of vaccine and genomics. Well, distinguished guests, ladies and gentlemen, now we are ready to continue to our last plenary forum of today's symposium that will discuss about standards and challenges of vaccine safety and efficacy evaluation. So before I invite the honorable moderator to moderate this last plenary forum, I would like to hear a big round of applause from the floor. Can I hear applause from you, ladies and gentlemen? Yes, I just want to make sure that you are still with us and also our online participations. Thank you so much for staying with us as now we will continue to our last plenary forum. For that, ladies and gentlemen, I would like to invite our moderator. Please be ready on stage. I would like to invite Acting Director of Center for Biomedical and Health Genomics. Please welcome on stage Ms. Indri Ruslamiati, MSc, APT. Please welcome. Yep. Welcome again, Ms. Now I will let Ms. Ingrid to lead the amazing session. I will hand over the microphone to her. Stage is yours.
Okay, thank you. So I'm back. So I hope you're not getting bored with me. So I think finally we will reach the last sessions. So the first sessions will be the standards and challenges of vaccine safety and efficacy evaluations. So as we know that the safety and efficacy is the key, because now the Ministry of Health is transforming from the curative into the preventive, and then the vaccine is one of the tools for the preventions. So therefore, without further ado, I will invite the first for all the speakers. So I will have 4 interesting talk for this evening. So one is Ibu Dia Puspitasari, S Farm, APT, and Biomed. So please, Ibu Dia. Okay, so give applause for Ibu Dia. And the second speaker will be Dr. Lee Yu Hua. Dr. Lee, come on, get up to the stage. And the third speaker will be Professor Dr. Ernie Juwita Nelwan, PhD Solo, SPPD, and go on. Okay, so please take to the stage Professor Ernie. And the last speaker will be Dr. Guan Xiaoli. I hope I pronounced your name correctly. Okay, so please come to the stage. Okay, so sorry for all the speakers because the organizer already tell me that you will only have 10 minutes to give a talk. So please make it as short as possible, yes? Okay, good. Okay, so I think we will go to the first speakers, Ibu Dia Puspitasari. So she's working in Indonesian FDA, or called Badan POM. And she is graduated from University of Indonesia since the bachelor until the postgrad. And now she is the team head of biological product registrations. So I will give the floor to Ibu Dia. So please, Ibu Dia. Okay, thank you. Okay. Good afternoon, everyone. Thank you for being here. And today I would like to talk about the ensuring vaccine safety and efficacy in Indonesia. I'm Dia from Indonesian FDA. I'm team head of biological product registration in the Directorate of Drug Registration, Indonesian FDA, or BPOM, Badan POM. And then we in this presentation, it covers 3 main points. The first is about the development of vaccine and the registration procedure of the vaccine, and then also how BPOM will support the investment for the local production of vaccine in Indonesia, and as well as for the evaluation to ensure the vaccine safety and efficacy in Indonesia. Next. This is the BPOM overview. Maybe some of you don't know about BPOM very well. So I would like to introduce BPOM first. Next. BPOM have the 76 regional offices across Indonesia, and we have the vision that drug and food are safe, quality, and competitive to achieve the establishment of an advanced Indonesia to be sovereign, independent, and have personality based on mutual cooperation. Why I deliver this vision? Because it is to make sure that the product that marketed in Indonesia should be safe and assure in the quality of the product. Next. And then this is the regulatory system in Badan POM or BPOM. The oversight of the quality, efficacy, and safety of the vaccine is integral part of that cannot be separated between pre-market and post-market. And then we as the regulatory authorities we assure from the clinical trial of the product. So we issue the clinical trial authorization to make sure that the clinical trial is conducted under the GCP, good clinical practices. And then we have some regulation to support the regulatory system of BPOM or Badan POM. And then for product, it should be, we have to ensure the aspect of the efficacy, safety, as well as for the quality. And for the quality, it should be under GMP, good manufacturing practices. And then all the clinical trial data, as well as for the quality, it is as a part of consideration for issuing the marketing authorization in Indonesia. So after marketing authorization, throughout the product life cycle, there is the monitoring post-market after the product is after the marketing authorization of the product issuance. And then for vaccine, especially for vaccine, there is the issuance of the lot release for every vaccine or batch per arrival. And then since the vaccine susceptible to the temperature, so the distribution will be guided in the good distribution practices. And also for assure the quality throughout the life cycle, so BPOM also do the product sampling and lab testing for product in the market. And then for vaccine, we also do the pharmacovigilance. We monitor the adverse event following immunization. This is collaboration also with Komnas and Komda KIPI, as well as with the Ministry of Health. Next. This I would like to deliver that BPOM has reached the maturity level 3. It means that our authority is stable, well-functioning, and integrated regulatory system. Okay, next. For keeping update with the advance of the technology and information, we digitizing for vaccine control. Since from pre-market until the post-market throughout the life cycle of vaccine. And then these are the online submission, like for registration for GMP, GDP, export, import, and as well as for the clinical trial. And also for the surveillance, we have the integrated reporting for post-market control, including lab testing report, e-reporting for pharmacovigilance, and BPOM mobile. Next. We move to the vaccine development and registration procedure in Indonesia. Next. Vaccine is a biological preparation that is used to stimulate the body's immune response against the disease, as we know. And then next, these are the vaccine development in Indonesia. So the development of vaccine start from the early development, this is in vitro, and then continue to the preclinical in animal model. And then this is under the laboratory scale. So it can be under GLP, good laboratory practice, for early development and preclinical. And this is for production process development of analytical method validation, optimization, characterization, and product consistency. And then after early development and preclinical, it can be continued to the clinical trial study. We have phase 1 clinical trial, phase 2 clinical trial, phase 3 clinical trial. And for phase 1 and phase 2, it can be produced in the pilot scale, but for phase 3, it must be in the commercial scale. And for the product that used in the clinical trial must be under GMP, good manufacturing practices. And then during the clinical trial, the principle of the GCP should be applied, good clinical practices. And for phase 1, it's in healthy subject. This is a general for the development. And then phase 2, target population, and 3, a large scale of subject. But in the term of vaccine, the subject must be healthy subject in the clinical trial. And then after we do the assessment from all the data, including the efficacy, safety, quality, as well as for the product information evaluation, we consider the risk and benefit assessment, and then we issue the approval or marketing authorization. And then we do the post-marketing surveillance after the product launch to market. Next. Next. Okay. This is the regulation on registration and marketing authorization. It's based on Health Law number 17, 2023. And then the regulation number 5, 2021. And as well as regulation BPOM on criteria and procedure of drug registration. So for all the registration procedure, according to the per BPOM 24 2017. It's like our holy book for this per BPOM 24 2017 for registration. And public protection toward unexpected risk medicine by providing assurance on the quality, safety, and efficacy of medical product marketed in Indonesia. So we should assure the quality, safety, as well as for the efficacy of the medicinal product, including the vaccine. And for pre-market control, this is the aspect that should be evaluated before the product marketed to Indonesian people.
One more minute.
Okay. Okay, this is our registration procedure, the submission, online submission, and then evaluation process for all aspects. And then there is the decision making. This is based on the benefit and risk assessment. And we have national committee to discuss about the benefit and risk ratio of the product, including vaccine that will be marketed in Indonesia. And then the issuance or marketing authorization. But the issuance marketing authorization, it is depend on the data that the applicant submitted to BPOM. Next. This is the vaccine evaluation pathway in Indonesian FDA, but I want to highlight for the 50 working day and 100 working days that for 50 working days, this is for first investigational new drug registration for pharma investment in Indonesia. So if you have a pharmaceutical industry and want to invest in Indonesia for the first registration, you have the accelerated review in 50 working days. And then 100 working days, I want to highlight that the drug development that including doing the clinical trial in Indonesia, it can achieve the accelerated review in 100 working days. Meanwhile, the regular review is 300 working days. So for investment in Indonesia, we have the accelerated review for the product. Okay, next. This is the dossier format. We can accept ICTD as well as for the ICH. Okay, next. This is evaluation on safety and efficacy of vaccine. Next. Okay, for assurance in efficacy and safety of the vaccine, we according to the non-clinical and clinical evaluation. For non-clinical evaluation, immunization studies in appropriate animal models are conducted because they may provide valuable proof of concept. So why the applicant or the pharmaceutical industry must do the non-clinical study because to provide the proof of concept information later to support a clinical development plan. And then for the non-clinical data, pharmacology, pharmacodynamic, as well as for the immunogenicity, it is established immunological characteristic of the product and guide the selection of doses, schedule,
route of administration to be evaluated in clinical trials. And then for the toxicity, potential toxic effect of the product are evaluated with regard to target organ, dose, routes, exposure, duration, and frequency of exposure, and potential reversibility. And there is some additional toxicity assessment, such as developmental toxicity studies, genotoxicity, and carcinogenicity studies. Next. And then this is for clinical evaluation. Clinical evaluation, not only in the pre-licensure or in the pre-market, but also post-licensure. So for pre-licensure, to describe the interaction between the vaccine and host immune response, and to identify safe and effective dose regimens and schedules, to estimate vaccine efficacy by directly measuring efficacy and or to provide evidence of vaccine efficacy based on immune responses, as well as to describe the safety profile and to assess co-administration with other vaccine if this is relevant. Since in the immunization program, children will be given by a series of the vaccines. And then for post-licensure, it is essential to monitor vaccine safety in routine use and studies designed to address specific safety issues that were identified as potential concern from pre-licensure trial may need to be conducted. It may be appropriate to conduct studies specifically intended to estimate vaccine effectiveness. Next slide. Not only non-clinical and clinical data that we evaluated, the risk management plan document, it should be submitted as part of the registration dossier. Why we request this risk management plan document? Because it can explain how risk will be minimized in patient and how those efforts will be measured. So in the risk management plan document, it will describe the pharmacovigilance plan for the in the post-market area. So this is very important for the surveillance after the product released to the market. Next. This is the support of BPOM for the investment on providing local vaccines. Next. Badan POM is responsible for the regulatory assistance. So if you develop the vaccine, so we provide the regulatory assistance from development since early beginning of the development until the product launched to the market. And then technology transfer. If you want to transfer the technology, maybe from China to Indonesia to the particular pharmaceutical industry, we do the assistance in the technology transfer. As well as for marketing authorization, as I said before, that for investment in Indonesia, we have the accelerated review for 50 working days and 100 working days. Meanwhile, in the regular pathway, 300 working days. Next. This is our effort to support development of domestic pharmaceutical industry. And this is our regulation and standard support. Next. Okay. For downstreaming the research product, it's nothing if there is no collaboration. Okay, so if you have the research product that very good, but if there is no collaboration between the academic, business, and government, it's nothing. So we think that the collaboration with the academic, business, and government is really important for downstreaming the product research that will be marketed to people. Next. This is way forward from my presentation. The first that Badan POM assists every stage of development until issuance of marketing authorization in Indonesia. And then Badan POM encourage the new drug development, including the vaccine development through regulatory assistance. And marketing authorization is based on proof of quality, safety, and efficacy according to standards. And Badan POM provide guidelines and regulation involving researchers and pharmaceutical industry. And in the context of downstreaming of research product, collaboration between researcher, industry, and government is needed. Thank you for your attention. This is, yeah.
Okay, so give a big applause for Ibu Dia Puspitasari from Badan POM. So we will go to the next speaker. So Dr. Lee Yuhua. So she will give a talk about the development of the quality control of vaccine for human use in China. So she's from NMPA. So I believe that she was a former director of the Arbovirus Vaccine Laboratory and also has so many experience in doing research and development, especially that included in the National Pharmacopoeia, and you are also the members of the National Pharmacopoeia committees. Right?
Thank you for your introduction. And first of all, I thank Professor Zhang. He invited me here to communicate with all of us. Today, my topic is about development on the quantity control of the vaccines for human use in China and from the National Institute for the Drug and Food Control. Okay, next one, please. Okay, today I want to outline my topic including the outline and the strategy of the quality control of the vaccine in China and the control test for the NDA and the not release and the supervised sample. And also I will briefly introduce my investigation on the quality control. Okay, next one, please. And in China, the vaccines including the bacterial vaccine and the viral vaccine and the genetically engineered recombinant vaccine. And also nowadays, the nucleic vaccine also are in development. Next one. Okay, in China, the vaccine manages the mainly about the pre-marketing, the vaccine registration should upload and the vaccine manufacturer should be certificated and before the vaccine before the vaccines go to the market, then the lot release should be carried out. And finally, after the vaccine going to the market and the vaccine should be supervised. Okay, next one, please. And of course, the quality control should be done according to the series of the laws or the regulation, the related regulation, including the mainly about the drug administration law of the People's Republic of China and the vaccine administration law. Also, the regulation of the model release of the biological product. Of course, the other regulation should be applied with. Okay, next one. Of course, in China and how we can control the quality control of the vaccine, we should consult or we should carry it out according to the entire life cycle of the vaccine that means from the pharmaceutical development and the technical transfer to the commercial manufacture to final product or even discontinued. This is the whole life cycle of the vaccine should be managed. Next one, please. Of course, the target of the quality control of the vaccine is the safety and the efficacy and the controllability. Of course, how to reach the target of these three quality control properties? We should done series control tests. Okay, next one. Just now, I talked the quality control is done by the whole life cycle, and from that means from the bacterial or the virus strain or seeds. For this part, the virus and the bacterial strain should be lawfully and be subject to the approval of the NRA. And also the various states should be managed based on the various systems that including 3 classes of the various states. That means the primary, master, and working various states. And for this system, several elements should be done, including the reports, origin, history source, and the passenger number, and biological characteristics. And several tests should be tested, and the meaning points and the key points should be concerned about this, something like this. Okay, next one. For the animal cell substrate, several control also should be done. First, the cell substrate be sourced lawfully and be subject to approval of the NRA, and the cell bank is also managed based on the cell bank systems that is a master cell bank, working cell bank. If this cell is established, the new one, and the primary cell bank should be controlled. There are several work are included about the reports, origin, history, source, and passenger number, and some biological characteristics also should be paid much more attention. And there are also control tests on the cells should be done. Okay, next one, please. And because of the poor life cycle of the vaccine should be managed. So that is from the auxiliary materials and its experience. The experience should be controlled according to the law and regulation and the national standards, that is the Chinese pharmacopoeia. And yeah, and the safety risk of the advantageous agents contamination that may be potentially brought of the biological or auxiliary materials derived from animal, or they are. So their quantity are strictly controlled, and the controlled content should be as follows. Okay, next one. And from this, the whole production process, we should strictly control this, the system elements. For example, the facility and the production management should comply with the current the GMP, and if the lot of the vaccine comply with the test, or the comply with the control test, but this lot of the vaccine is not meeting the GMP, that this lot is not satisfied and is not accepted to the market. And for the manufacturing process and the control test should be performed according to the approved by the NRA, and from the raw materials and the final products, the whole manufacture process and the control test shall be carried out according to the registration standard and the national standard. Also, it should comply with the laws and the technical requirements and regular specification, such as the Chinese pharmacopoeia. Okay, next one. Next one, please. Okay, yeah. The bacteria and the virus, especially I want to pay your much attention about the activation process. If the product is in activated vaccine and the activation process should be established, the key parameter including the protein content and the activation time and the active agents content and so on. This is a very, very critical point should be paid much more attention. And for the final batch and also should be prepared according to the approved the former and the fixed antigen, content in order to ensure the not consistency. Okay, next one, please. And
1 more minute.
okay, for the quality control, we should all comply with the series, the requirements and the standard. Okay, next one from the salary and experience. And next one, please. Okay, I want to introduce my GE11 or our GE11 vaccine. Okay, next one. Okay, next one. Okay, this is in China and or in the world, the life alternative to the Japanese encephalitis live vaccine is very important and play very important role in controlling of the GE. And this is a schematic and first of all, the preparation of the GE live bark and then the stabilizer to formulate and lyophilize it. Finally, we got the GE attenuated vaccine. Okay, next one. This is a production process, and we use the SP for hamster kidney cell to prepare the cell and indicate the virus and cultivate the virus. Finally, about the 17 hours to harvest the virus shortly and to prepare the bark and the some stabilizer to guard the final bark and filling and freeze drying camping, physical observation and after labeling, packaging, and we got the final product. Next, please. And this is how to control the Japanese encephalitis quality of the GE11 vaccine. For all of these persons, there are many control tests to be done to control the quality control. Okay, next one. And in 2013, and the GE pre-qualification, and nowadays there is exported to several countries, and nowadays we are expanding the use of the GE11 vaccine, the attenuated strain as a backbone to develop dengue, Zika, and some virus like this. Some virus vaccine like this because of the GE SR1442 is a very safe and very efficient, and the neurovirulence is very stable. And after so many years and the large the doses has been used in the real world. Okay, next one. And just I show you for the inactivated vaccine, how we control the quantity of the inactivated vaccine. I take the rabies vaccine. This is a production process of the rabies vaccine, and the vaccine, some items of the quantity control is the standard. The standard of the quantity control is much higher than that of the WHO. Okay, next one. There is also many control tests are carried out to control the rabies vaccine quality. Okay. Okay, next one, please. Okay, and this is I will introduce briefly. We how can we do the lot release? And just I talked we do the whole production process. Okay, this is a document for each the bank of the vaccine, each batch of the vaccine. We will document and next one and will test. This is a checklist how we control by the documentary. We use the checklist to control the whole things. Okay, next one. And if there are some the deviation or some other explanation, we should ask to end it here. Okay, next one. And the stability and the lot consistency, we will use the trend analysis. Okay. Next one, please. And this is also we test some batches of the not release samples, and for the stability and abnormal toxicity, and for the new manufacture, the first 3 lots should be tested, and also if there are some production and are changed or they deviated, and the whole test should be done. Okay, next one. Okay, next one. This is a certificate and the notice of the not release of biological products. The certificate only both checklist and the sample test are satisfied, and we issue the certificate for the release of the biological products, and we issue the notice of the not release. Next one, please. And this is MR, the molecular of the MR vaccine, and we control the MR vaccine based on the molecular elements. Okay, next one. Okay, this one. This is a process of the MR, and including design antigen and IVT and capture and purification. Okay, next one. Next one. This is a particle, and this is a machine to encapsulation. And the next one. And for the quality control, we quoted to the guideline of the Chinese CDE and the WHO guideline. Also refer to the USP and the EP, and the main points of the quality control are about the bacteria, seed bank, and the next one, and the plasmid, and the next one, and the BART, because this is inter process, the product should be controlled strictly, and including the identity and the content and the purity, or some residual materials, including the DNA or the dsRNA, and okay, next one, and is the quality control of the mRNA vaccine really is complex. Okay, and for the final product. Okay, next one, please. Okay, for the final product, this the appearance or the subvisible particle and the sequence for the identity and the length of the sequence and the integrate or the encapsulation and the content and the RNG size, and some items like this should strictly to control. Okay. Next one, and just now, and Professor Zhang also studied and Richard the COVID-19 adenovirus as a vector, and for this kind of the vaccine, we also pay much more attention the virus seed and the harvest. And the next one. And for the bug and the final. Okay, next one. And the final product, we pay much more attention about the identity and the express the antigen protein or the infection virus type and virus particle, and also including the appearance and some visible particle. Okay, next one. And it's ready. Okay, and for the research or the investigation to investigation on the J11 vaccine to improve the quality control, we did series work, because this the J11 vaccine strain SF1442 is from the SF14. Okay, next one. And there are differences. This is a critical amino acid for the SF14.2 and SF1442. Okay, next one. And this is a critical structure of the SF1442, which is really different from the variant strain of the SF14, because the different structure is a basis, structural basis to support the attenuated neurovirulence of the J11 vaccine. This is a molecular mechanism. This work was done in 2014 with cooperate with Germany. Okay, next one. Next one, please. Okay, next one. This is how different between the parent strain and attenuated strain. Okay, next one, please. Next one. Next one. Okay, this work is cooperated with Michelle, Professor Michelle. Okay, next one. And maybe someone worried the J11 vaccine was used in large scale, whether or not it is influenced to the environment we emulate the propagation of the J11 vaccine in pig and mosquito, and the result was satisfied, and there is no any influence to the environment. Okay, next one. Okay. Oh, I will introduce the Chinese NRA was passed the WHO NRA the evaluation 3 times, and each time was satisfied. Okay, thank you for attention.
Thank you very much, Dr. Lee. That was a really nice presentation. So we will move to the third speakers, Professor Ernie Juwita. So she will give a talk about unveiling the challenges of vaccine safety and efficacy evaluation in research and development. So Professor Ernie, the floor is yours. Yes, 10 minutes, please.
Okay, thank you, Mrs. Chairman. First of all, I would like to thank the organizing committee for inviting us. So I'm Ernie Nelwan, and I work at the Faculty of Medicine, University of Indonesia. My talk will be more to share about our experiences because for the last maybe 5 years, I've been conducted several vaccine trial, including the one that just finished, the malaria vaccine trial, the first conducted in Asia within the last 30 years, I think. And also some others vaccines study related to the safety and of course the efficacy evaluation. Next slide, please. So I represented my group. So this is not only the Faculty of Medicine. Actually, we are also collaborated, like Ibu Dia, the first speaker, said that for the vaccine, we need to do the collaboration. So our vaccine trial involving the Indonesian Army and also the Oxford Unit Clinical Research that based in Jakarta in the Faculty of Medicine. So as you can see here that when we talk about vaccine trial, safety and efficacy as our objectives, but that actually is on the top of a lot of process before that. As you can see at the bottom, we have the submission study. We listen to the second speaker also, Professor Lee, that there's a lot of things we need to be sure and also assured during the preparation process and also after that we have the result. And then safety and efficacy is in a two different stage of the process of the study itself, because vaccine safety, we can already get the data during the process, but for the efficacy, of course, we have to wait until we measure the indicator of perhaps antibody, or we open the blinded participants that been involved in our study. Slides. So I would like to share our malaria vaccine trial result. They just been open to the public, but not yet published. So our data is confidential. To the Faculty of Medicine, also to the Indonesian Army 2 days ago, and also to the Ministry of Health. Slide. As you can see here, the initiation of a vaccine trial could be long time before we got the result. Our vaccine trial started in 2018, and then COVID-19 pandemic, and then the changing in the Army authorities slides. And then finally, we get an approval in 2022, while the study itself needs to be conducted within 2 years. So you can see that a long process for getting the result of safety and vaccine efficacy. Slides. So I would like goes directly to the result. So we managed to include 3,345 soldiers that going to be deployed to the highly endemic area of malaria in Papua. Slides. And okay, I do not get the right PowerPoint, but it's okay. So this is the trial that's been regulated in Jakarta, and we vaccinate the army in Sumatra. This is the area of free malaria, and then this battalion deployed to the highly endemic area in Papua. So as you can see that the site could be separated with different responsibilities within sites. So slides. Okay, so this is what I just mentioned. So we are involving the different sites. Next slide, please. And we are conducting parallel 2 vaccines comparing with placebo. So we have 4 arms. So the 345 participants was divided into the 4 arms group. And as you can see that we need to manage to vaccinate all the soldiers according to the protocols. And we need to be sure that during the deployment, as you can see in the bottom, the purple area, that for getting the safety data, we need to following them up every 2 weeks to get an active and also passive information about their condition. Slides. The next 3 to 4 slides will be reporting what we collect, the data that we collected during the trial. So this is the solicited adverse events and unsolicited adverse events. We already classify and we collected the data. Slides. And you can see between vaccine jab, first, second, and 3, because it's a booster vaccine study. There's no difference, and a very, very, very small number of reporting solicited adverse event. Slides. This is the sites of injection adverse event. So after you put the needle, whether there's any complaints. As you can see, also the number is very low. Next. And this is how we compare it between the vaccine and the placebo. So red mean a lot of reported of the events, but green and yellow means it's less reported. And you can see is between the vaccines and between the placebo, it's comparable. So it can be concluded as safe. Slides. This is the same because as I mentioned, we have two parallel vaccine studies. Slide. Next. And also what's important for the vaccine is we need to document it, the adverse event. So this is what we collect. We need to measuring every single complaints, and we need to be sure that this is reported to the BPOM within 24 hours after the report. Slides. Still the same. Next. Yeah, so then that's how we get the safety data. So this is really a lot of works, and we need to be sure that that's documented carefully, and then also involving parties such as not only us, but also the BPOM and also the EC committee. Slides. This is the vaccine efficacy that we got from our trial. Next slide, please. Maybe I'm not going to go to detail, but I want to explain here that, yeah, to get that all data that we need to do this. So we collect more than 11,000 bloods to be sure that we can tell whether the vaccine is works or not, because we are measuring the malaria, is there or not, and also the SAA, and also we make a lot of reports for the EC and other parties, and also for our collaborator in the United States as a sponsor, and also we do the clinical observation every 2 weeks, as already mentioned. Slide. Okay, next is about dengue vaccine evidence that already resulted into the guidelines in Indonesia. Slides. So this is how Indonesia not involved in these dengue vaccine trials, but we are I think we are the first country that approved this vaccine to be used for the people. And then as you can see, because we are not really involved, then we only can get the data from what been shared by the pharmaceutical company. As this next. And this is the efficacy of the vaccine. Excuse me, this is the SAA. The first one is the solicited and unsolicited. Next. And this is the vaccine efficacy. Then next slide, translated into the recommendation. So we can also use data from others that then we have the discussion to say whether this can be implemented in our setting or not. Last study, next. At the moment, we also have the collaboration between the Indonesia and China about the meningococcal conjugate vaccine, and this is on preparation for the data analysis because we just received the MTA like a month ago. Next slide. Yeah, so there's a lot of steps that need to be paced and prepared. But as a researcher and as the experience of doing the trials, I think this is all doable as long as we need to be able to maintain the timeline for the study, because sometimes sponsor need the precise timeline to be able to conduct or not conducting the study. Slides. Yeah, so for potential collaboration on vaccine research and development, I think Indonesia is ready, and yeah, all these steps that mentioned here in my last slide actually can be managed, and yeah, we get already results from several studies, maybe including a COVID vaccine trial that's perhaps Prof. Erlina already mentioned before, because it's also conducted in Jakarta and involving Cipto Mangunkusumo Hospital as one of the settings. I think that will be all, and thank you very much for the attention. I give the time to the moderator.
Okay, so thank you, Professor Ernie. Yeah, we are very short in time because I think there are another agenda that should be going on 5 p.m. So before I apologize to all the speakers that we have to limit the time from 15 minutes to 10 minutes. So is there any questions? Okay. So if no, then I think I give a big applause to the speakers, Professor Ernie dan Ibu Dia. So I will give the floor to MC, Master Ceremony, and thank you very much.
Thank you, moderator. And speakers, please remain on stage. Yes, we have to also capture both of speakers and also moderator. Please be on stage and we have to capture this moment. Yes. Right. Now, please face the phone camera. All right. Thank you so much. And allow me to do the count. 1, 2, 3. Another pose? Yes. Yes, please. 1, 2, 3, smile. All right. Thank you, moderator and speakers. May I please return to your seat, and also thank you so much for your insight during the presentations, ladies and gentlemen. And I have a good information for you. We will show you on screen the barcode. So don't worry, you can also receive all the presentations for all the speakers from the 1st until 4th of the plenary forum. So we will show you later, ladies and gentlemen. You can scan the barcode that we will show on screen to receive all the presentations from all the speakers. Well, distinguished guests, ladies and gentlemen, offline and online participants, by the end of the 4th of plenary forum, we now come to the end of our symposium of this evening. I'm Putri Cahaya as your master of ceremony signing out, and we believe this initiative will enhance our knowledge and capacity, especially for vaccine and genomics, to achieve betterment of our generation. So once again, on behalf of the organizing committee, we extend our sincere gratitude to each and every one of you, all the participants who are joining us here on site at the Trans Resort Hotel Bali, and also online participants. Thank you so much, everyone. We do hope you get a significant impact through our symposium this evening. So we do hope you enjoy the rest of your stay in Bali and have a productive day. Once again, I'm Putri Cahaya signing out. Enjoy, everyone, and see you on the next event. Thank you.
Dalam skema triple helix dalam menjalin sinergi antar stakeholder di bidang vaksin di Indonesia. Volare bergerak dalam mengkolaborasikan aspek-aspek riset dan pengembangan manufaktur, penyediaan, harga dan pembiayaan, distribusi, logistik dan rantai supply, penerimaan dan serapan pasar di bidang vaksin.
Thank you.